Hello, good morning, everyone. And you are very welcome to the third edition of our workshop NeXT-Chem. This workshop is, as you know, is dedicated to the young researchers, master students, PhD students, and also to the postdoctoral students. This workshop was started, the first edition was 2 years ago, in frame of a project for excellence financing. And I'm very happy that yesterday our Ministry of Research announced that this year we launch again this kind of project, which is very good for us to improve our excellence in research and to produce more results and to be more visible for the international community. This year we opened this workshop to students also from other organizations of research, not only for our institute, because till now it was like this, and now we want to grow up this workshop and to give the possibility to other young people to present their results. Also, we are very honored by the fact that this year 3 international personalities in our field of work accepted to present invited lectures. Is Professor Arben Merkoçi from Catalan Institute for Nanoscience and Nanotehnologies, also Professor Silvana Andreescu from Potsdam University, New York, United States, and Professor Luc Picton from Université de Rouen, France. And we thank very much to these partners of our partners that they were so kind to present to our young researchers their results and to encourage them to continue on this road of research. So you are very welcome to this workshop, and I hope that these 2 days will be very interesting for you, and you will meet each other and you will make exchange of ideas, and maybe to create new partnerships for the next years. Thank you very much again, and I want to ask to Professor Radu Fierescu to say some words about this workshop, because he was in charge with all the organization of these activities, and to keep in contact with participants and to organize everything. And I want to thank to him also. So please, Radu.
Thank you very much. I want to thank to our colleagues, both in the organizing committee and in the technical committee, who worked very much, and they will work today and tomorrow to help all the presenters. We are very happy, as our general manager already told you, we are very happy that we have several colleagues from other universities and institutes with us today and tomorrow, and I do hope that in the next years this event will be an important event at the level of PhD students, which will be recognized by doctoral schools and will be considered for their PhD studies. Thank you very much.
Thank you, Radu. And also I want to ask Professor Andreescu to say some words, because also he was very much involved in this organization, and also will be the chairman of the first part of the day, the workshop day. Please, Professor Andreescu.
I'm very happy to send the greetings of behalf of Romanian Chemistry Society to all participants in this third edition of NeXT-Chem. This symposium is a new series of old former symposium which was held in our institute in the years 80. And I'm very happy that it has a very big success. Every year it grew up, and now we are receiving students from our institute and university, not even from our institute. The intention of this symposium is to train the students to be able to give a scientific talk and to sustain a scientific debate about a subject. This is very useful, and for this, Romanian Chemical Society joined with our direction of the institute and with scientific council, and we hope that we will develop this year more and more than former edition this symposium. I wish to all participants a big success. Thank you very much.
Thank you very much, Professor Sârbu. And I think even we have several minutes, we may start with the presentation of Professor Arben Merkoçi. But first of all, I want to say some words about him, about his activity, and to introduce him to you. I will put also his short CV on the screen. Just a minute. So I'm very happy that Professor Arben Merkoçi accepted to make this presentation today. I met him 20 years ago in frame of FP5 project, a very nice project about toxins, determination of toxins in milk. In that project, Romania, it was a partner together with Barcelona University, where Arben worked then, and was coordinated by University of Rouen at Vergata. And since then I followed his career, very impressive career, and now he's professor and director of the Nanobioelectronics and Biosensors Group of Institut Català Institute for Nanoscience and Nanotehnologii, which is a part of Barcelona Institute of Science and Technology. So after his PhD at Tirana University in Albania in ion selective electrodes, he worked as a postdoc and senior researcher researcher in field of nanobiosensors and lab-on-chip technologies in Italy, Spain, United States, and since 2006, he's working in Catalan Institute for Nanoscience and Nanotehnologii. Professor Merkoçi research is focused more on nanotechnology and nanoscience-based cost-efficient biosensors. In the last years, he developed the paper plastic-based nano-based biosensors, which are his research is a reference for all the groups working in this kind of sensors. He has an impressive Hirsch index is around 70, with more than 300 peer reviews papers, and he supervised 30 PhD students at least, and he's a co-editor in chief of the prestigious journal Biosensors and Bioelectronics, and member in of editorial boards of other journals. And it's very interesting that he's a co-founder of 2 spin-off companies, and as you know, here in Romania also the government pushed us to spin off our research results to make this, to found this kind of companies. And maybe he will also tell us 2-3 words about this experience as a co-founder for spin-off companies. So thank you very much, Arben, for your presence here, and please share with us your results. Thank you very much. I don't know if you hear me. Sorry.
Sorry, can you hear me?
Yes, now, yes.
Yeah. So thank you very much, Dr. Mihaela, for the nice introduction, and of course for the invitation to you and the organizing committee. So may you stop sharing your screen so I will start my screen?
Yes, of course.
Can you see my screen?
Yes, we see.
So thank you very much. It's really a pleasure for me to be here today, and especially because of you, Dr. Mihaela, for the invitation, and of course for the fact that we have been working together since many years. So it was like 20 years when we were partner in the project coordinated by Professor Paleschi in Rome, Rosa Promit project for toxins, and you are one of the key persons, researchers, scientists in Rome working in this project, and really it was great to know you and collaborate with you. And since that time we have been in contact, and I'm really very happy that we are together now in this workshop, and with the willing to collaborate and share expertise, and be probably partner in the same project again together, because we had a great experience that time with this European project. So I'm going to show you what we are doing in Barcelona in relation to nanobiosensors or nanomaterial-based sensors for diagnostic applications. When I say diagnostics, I mean not only health-related monitoring, diagnostic, but also environmental monitoring and safety and security. But the special thing in these applications are that we are trying to work with simple materials, simple platforms, trying to integrate different kind of nanomaterials into paper, plastic, with interest for different applications. So, sorry, I have to share again because I forgot to do the sharing of the voice. So, sorry, just 1 second. So I'll stop sharing and will do it again. Sorry again. Sorry for this. Stop sharing because I have to do share sound because I have some video. So can you see it now?
Yes, we see.
Okay. So I'll have a short introduction first about our center. I work at Catalan Institute of Nanoscience and Nanotehnologii, which is a joint research center between different entities. This is why you see here different logos. This is our center here, and you can see here, let me show also the pointer. Laser pointer. So this is why you see here different logos that belong to different entities to which we belong, because we are part of National Council of Research, CSIC of Spain, but also we are part of a regional network of research centers and finance supported by both national government and regional governments, and also Autonomous University of Barcelona, UAB. Since several years, our center, you see here, ICN2, is part of this big center, Barcelona Institute of Science and Technology. So what we have done is gather together 7 research centers so as to increase the visibility and collaborate. And this is really a very nice idea because we are the centers of with different expertise, and we can collaborate with us. And this is really a very nice formula that very much recommendable also for other places. And here is where we are. So I think that some of you have visited Barcelona. This is Barcelona city, and behind these mountains there is the campus of Autonomous University of Barcelona, and we are somewhere here, very near to the city. I put here also some hospitals, biomedical research centers, because we have a lot of interest in collaborations, and Barcelona is very famous at the European level also for biomedical research. So it's really very nice place also to collaborate and work. We are at the campus of International Excellence, UAB, and being a campus of International Excellence, we have nice facilities from business area to technological part, but also to mention that we have also synchrotron in the campus, which is very important for developments and, of course, science and technology transfer, innovation services also, with nice facilities also in terms of micro nano fabrication. So in collaboration also with Microelectronics Center, we have next to our center. Our center is, as I said, is devoted to nanoscience and nanotechnology, and we are trying to develop, and we are doing well in working in nanoscale frontier research, but also we are very much interested to for the technology transfer. I'll show you also something about our companies, spin-off companies, and of course public outreach, so collaboration with different players with interest for the science we are doing. So being a center in nanotechnology, we are have different research lines, and you can see here several of them, which are very interesting, ranging from biosensors and bioelectronics, for which I am responsible, to other colleagues working with different aspects related to nanotechnology, nanomaterials, their properties at nanoscale level, including modeling, theory and simulation, spintronics, surface properties, and all these with interest for different applications. So our main pillars of application include from energy to life science to ICT. And all these supported by different transversal activities that goes from theory and simulation, nanomaterial growth, nanofabrication, characterization, and metrology, which gives support to all these activities. My group and what I'm going to show is related mostly to life science. So this is what we are more interested in my group. And in this context, we have interest a lot in overall in diagnostics, as I said, not only clinical analysis, but environmental monitoring, but also we are interested in developing of materials with interest for therapy sometimes, and then putting the things together in the so-called theranostics, so therapy and diagnostic connected together, and also in the environment context, so detection, for example, of contaminants, destruction of contaminants, and putting some platforms together, which is really very, very interesting. So going now to a little bit to the area of health, which is one of the main application areas, you know that there are a lot of problems, a lot of challenges for nowadays, problems related to aging of population, chronic diseases, spending on health, or pandemics. Nowadays we're living this COVID-19 pandemics and all this, and we are now facing this, the importance of diagnostics. And of course, when we speak about diagnostics, we could say that biosensors are very, very important and are a very, very important alternative for use as diagnostic device. Overall, in places with low resources, extreme conditions, we need this kind of devices. And you see here different, for example, kits or sensors, for example, like glucose biosensors that you can find in any pharmacy in the world, or even nowadays sensors to detect COVID, like lateral flow. But for all these devices to be useful, to be applicable in real situations, real clinical scenarios or environmental scenarios, it is very important to fulfill, we call this the criteria of reassured. It means that they should be able to do sometimes real-time detection. They should be very easy in terms of sample collection or easy of specimen collection. Affordable, this is very important to end users, so otherwise nobody is going to buy this if they are very expensive. This should be sensitive, avoid false negatives, specific, very important, so selective, user-friendly, so easy to be used, because you can use this even at home, rapid and robust, otherwise has no sense because they would not work properly. Equipment free sometimes, so you can just even with naked eye detect something like lateral flow, for example, similar to pregnancy tests, or you have seen now to detect serological test for COVID. And deliverable to end users. So these devices should go to the people that need this for use. So this is very, very important. And going a little bit to biosensors, I put here the biosensor for glucose that people knows. So, but you can have any kind of biosensor for any kind of application like this one, where the most important part is this sensor. So the biosensor, which is disposable and connected to a simple reader. It can be even a smartphone. There are two important parts of the biosensor, receptor and transducer. So receptor, it means any kind of biomolecule or synthetic molecule that is able to detect in a specific mode any kind of molecule or species you want to detect. For example, in case of glucose, we have there glucose oxidase, an enzyme, or if you want to detect a protein, you need to put here as receptor antibody, or you can put a DNA strand or RNA, for example, if related to COVID, for example. Once the interaction, so the specific interaction occurs in this surface, in contact with the sample, a signal is produced, and there is the transducer who is getting this signal. It may be electrical signal, optical signal, mass change signal, and then connected to the system, to this small reader, a signal is produced, and we have the signal treatment readout. So the user get a simple response, yes, no, or whatever value that is with interest for diagnostics. So this area is known, and since several decades, people working by sensors. What we are trying to do now is trying to push nanotechnology and nanomaterials to improve this kind of biosensors, or even to develop new biosensors. So nanomaterials are very different. So we have different kinds of nanomaterials. We are working from micelles to liposomes, gold nanoparticles, quantum dots, graphene, very, very interesting. I'll show you some examples, nanotubes. And why? Curiously, the range of the size of nanomaterials, usually from up to 100 nanometer, fit quite well with the size of several biomolecules, which are the building blocks in most of the biosensors. For example, glucose, protein, DNA, viruses. So we are precisely in the same range of sizes of even the virus. So COVID is in this area of size. So it's natural to consider these nanomaterials as building blocks to work together with these biomolecules and build any kind of biosensors, taking advantages of interesting properties of nanomaterials like surface, a large surface, physicochemical properties, either electrical, optical, and many other properties. In fact, biosensors nowadays can be everywhere, and for example, these may be as medical devices, as wearable, even in the smartphones, smartwatches. There are some sensing there. Smartphone, smart TV, everything smart. You see, you can have sensors in any part. Hello? Are you hearing me?
Yes, we are here. Sorry.
Okay. Yeah. So biosensors can be everywhere. So as you can see, even for example, we had these some sensors integrated in a buoy system. This was also a project that was coordinated again by Professor Paleschi in Rome. Trying to put some sensors in the buoy and to use these to detect some contaminants in the ocean. So in the smart cities in the future, biosensors will be everywhere, and this is very, very important. So the field of sensors and biosensors is quite important for the future also, and for the present, of course. The nice thing is that nowadays, with the merging of the IT and all these communication technologies, smartphones are everywhere. Everybody has a smartphone, so these can be very easily connected to a biosensor. So we can transform a smartphone to a nice device to measure, and you can see here several examples. For example, a smartphone connected to lateral flow, a smartphone that is converted to a microscope. Or you can see here some old phones connected and used to do some electrical measurements. The nice thing here is also this Google glasses from Google. So they use these glasses. For example, you see here this guy is measuring the lateral flow and the signal. So the image is captured by this camera and treated and sent to the doctor office, for example. So there are several challenges for the field, and as I said before, we need to solve problems. We want these devices to be efficient. Sometimes these devices need to be disposable or implantable. So depending on the applications, we are trying to solve problems and apply these in different scenarios. What is important for biosensors, and if you want to be successful in this and even to open a spin-off company, is to keep in mind the cost, performance, manufacturing capability, and mass production. If your technology is not fulfilling these criteria, it's very difficult that your device, your technology goes out of the laboratory sold to the market. So we are trying to build and use these simple materials, as I said at the beginning, and to try to integrate nanomaterials and build very, very simple devices with interest for different applications in different areas, being diagnostics, one of the most important. And of course, in this area, we are working since many years, and here is a summary of the different reports from our group in the area of the use of nanomaterials. So since the use of nanoparticles as labels, for example, in bioanalytical systems, or the use of nanopores, nanotubes, graphene, even the use of nanomotors. I'll show you some examples, lateral flow, and also putting the things together in the theranostic or sensory mobile platform with interest for different applications. So let me show now in the next part of my talk some examples related to the developments we have had in our group since several years. I'm not going to enter in details. I want just to give you a flavor of the possibilities, the different developments of these different platforms. First of all, I will show you some examples related to nanoparticles, how we use this as electro-optical labels. Then I'll show you the use of some lab-on-a-chip nanochannels, because for biosensors, you need also sometimes to do per-concentration, filtering, automation sometimes. Then I'll move to a very simple platform. Simple is the best, and I'll show you how we integrate nanomaterials into the paper, nitrocellulose, and build very simple devices, some nanomotors to capture even single analytes. And finally, I'll speak about graphene, so why we are using graphene and some advantages and applications. So starting with nanoparticles, I'll show you the first example of the use of nanoparticles reported since several years. So in the cancer, so you know, cancer diagnostic is very, very important. According to World Health Organization, by 2030, the number of people that may die by this disease may reach 11 million. But 30% of cases are preventable. In the case 1/3 of cancer cases is subject to treatment in case of an early detection. So you can understand the importance that the early detection of cancer has in saving the lives. And according to clinical doctors, there are different scenarios, but two are the most important for cancer diagnostics. So the detection of fixed cancer cells, or the detection of circulating tumor cells, the one that escape in the bloodstream. And of course, they are with a very, very low concentration in the blood. So we are working with the two scenarios. First, with the fixed cancer cells, what we did is we identified first the proteins that are expressed on top of these cancer cells. We use some gold nanoparticles modified with antibodies against these antigens, and putting these cancer cells on top of some electrodes. These are screen-printed electrodes. Of course, we use some control experiments, or some cells that do not express this kind of antigens. You see that in the case of cancer cells, the gold nanoparticles are anchoring, so going to the cancer cells. Why not in the control cells? What we do for the detection is we use the so-called hydrogen evolution reaction. So if we have gold nanoparticles on top of the electrodes and apply potential, in fact, it's a negative potential, the hydrogen evolution is occurring. So measuring the current produced during this hydrogen evolution reaction and relating this amount of the current with nanoparticles, and in this case with cancer cells, we're able to quantify, of course, identify and quantify these cancer cells. So this is a very simple method that gives you an evaluation of the quantity of these fixed cancer cells that are on top of this electrode. But as I said, the most important is to detect also the circulating tumor cells. And for this, what we use is some magnetic beads. So in fact, we captured first the cancer cells through magnetic beads modified with antibodies, and then some other golden particles with some other antibodies were again capturing, interacting with the cancer cell. So you see here, this cancer cell is captured by these magnetic beads, and you see these white dots that correspond to gold nanoparticles. Afterwards, we collected these onto a magnetoelectrode and measured again the hydrogen evolution reaction. The nice thing here is that through magnetic beads, we're able to separate cancer cells, and then through magnetic beads, through gold nanoparticles, we're able, through the hydrogen evolution reaction, measure the quantity of these cancer cells. So the detection limits were nice, but still not that good to have these for use in early diagnostics. So we are told by the medical doctors that we should detect 1 cancer cell per 5 milliliters of blood. And this is really very, very difficult. And so far, nobody has achieved to detect such very low levels of cancer cells. So what we did also in this area is we tried to incorporate also this nanoparticle-based detection system even to PCR. You know, PCR is very, very interesting method, very sensitive. And what we did is we connected these golden particles with DNA strands. We put also magnetic beads, so we have all the amplification occurring, and then the amplicon, so the amplified strands bearing also the golden particles, is now detected with electrical system. This is a patented system that is in use now by a company in Barcelona. Also, we could do the same with this detection with PCR directly on top of the electrode, but in this case we use impedance spectroscopy, and we're able to distinguish between the different DNA sequences related to some plant viruses, and applied this in collaboration also with the company. So, as I said, it's very important in biosensors also to do some pre-concentration, because the samples sometimes are very, have a very heavy metrics, and you need to filter, you need to automatize. This is why you use also the so-called lab on a chip. So we have soft lithography process, but also stamping, simple plastic stamping of chips. These are standard technology that are based on the master fabrication, PDMS casting and bonding, so as to create these channels. You can see here, for example, so we are able to create different kind of channels and even integrate electrodes inside. And I'll show you why we do this. So after creating this structure, so microfluidics according to the application, so different channels, different entrance and output, we integrate electrodes, we use screen printing technology or inkjet printing. So they are very interesting because we can also print different nanoparticle-based inks, so that are with interest even for wearable devices. So we are printing using inkjet printing, even field effect transistors that usually you print this with clean room. You see here, this is a flexible, transparent, printed onto plastic field effect transistor that we used for some protein detection, or even printing this onto the paper using this simple inkjet printer that we are working since many years. So let me show you how this lab on a chip system works, and you have here an example. So this is the chip with inlet, outlet. We put the magnets, and we have the 3 electrode system integrated inside this. It's very important first to calculate and to optimize the magnetic field we want to apply, because the plug of magnetic beads that will be created here depend on the magnetic field. After optimization of the magnetic field and all the orientation of the magnets, we used some magnetic beads modified with antibodies, and then did the usual immunosandwich with 2 antibodies and nanoparticles, as I'll show you in this next slide. So we used some magnetic beads that are commercially available, DINA beads. We modify these with antibodies, and as you can see here, this is a cut through of the channel. So once we introduce the magnetic beads with antibodies, the sample is coming, so is it pre-concentrating, then the secondary antibody, and finally the nanoparticles that are used as labels, and to get the signal very fast. So the system now is pre-concentrating, and we use this to detect some biomarkers related to Alzheimer. In this case, it was apolipoprotein, which is one of the indicators for Alzheimer diagnostics. Another way to reach this filtering, for example, is by using some porous membranes. We use this alumina-porous membrane that have a very nice porosity, very controlled pore size and thickness. So we put this on top of the electrode. You see here onto this screen-printed electrode, we put these membranes. Inside these membranes, we put different kinds of receptors. We work with antibodies, tamers, DNA. What you can see here is that this platform is serving now as a filter. So it is the case here of a whole blood sample that is detected for some biomarkers. And as you can see here, the red and white blood cells remain on top because they cannot enter these pores, but the proteins that you want to detect can go through, and they can block the channel. What we do is through the use of an electrochemical indicator that is diffusing through this channel, we see that upon the recognition, this channel is blocked. So we correlate the entrance of this electrochemical indicator with the presence, absence of the analyte we want to detect. We have a kind of on-off system. So we play around this and we applied also this, for example, in the study of some cancer-related cells. This is neuroblastoma case where these cancer cells were secreting some metabolites, and to use these to detect through the use of antibodies inside these channels, and use this system for some drug studies, so interaction of some drugs with cancer cells, because they were inducing the segregation of these peptides, and we were able to detect this with interest for drug development. And here are some data that we did in collaboration with hospital in Barcelona. So this was with interest for drug development, as I said, but thanks to the use of these nanopores, we were able to move these to some real nice applications. Now I'll show you that it's very important in biosensors to have cost-efficient platforms, and this is why we are also working with paper. Why paper? Paper is nitrocellulose, based on nitrocellulose, and as you know, this is low cost, easy to manufacture, recyclable, biosustainable. The fact that the paper has a porous matrix is very interesting because given the capillary forces, some movement of the fluidics can occur, so you don't need energy. So it's a kind of zero energy device. So the liquid is running in the paper in a very natural mode. If you cut the paper and you change the size, the shape of the paper, you can change also the microfluidics. I'll show you what we do. And also, we can integrate very easily into the paper different kinds of nanomaterials, nanoparticles, quantum dots, transforming the piece of paper into a plasmonic paper, for example, or other kind of optical detection system. The idea behind this is why not to use very simple system, as simple as a pH paper used to detect pH. So just putting this strip into the liquid and based on the change of the color to detect something. And there are different formats for these, known as dipstick, LFA, micropad. So we wrote a review on this, so you may have more information about the different paper-based systems with interest for different applications. And also here you have in this other review, Chemical Society review, you have some more information about the different nanomaterials that we can use in different applications in relation to point of care system and more with the paper-based sensors. Recently, we wrote also a nature protocol where we have more information, for example, for lateral flow device, so how to build a paper-based system for detection, even COVID or any kind of diagnostics, with interest for different applications, mostly in diagnostics, going from assay design and all the components to the detection. Let me show you first how these assays work, and this is the case of lateral flow device that we are working more. Here is the case of nanoparticle-based lateral flow. So you see here we use gold nanoparticles modified with antibodies and nitrocellulose with different porosities, so they are in different pads like sample pad, conjugation pad, two antibodies, different primary and secondary antibody, and then an absorption pad. Let me show you this video that explains better what we are doing. Sorry, I have to. So you see that once you introduce the sample onto the paper is serving as filters. So everything is running, so big particles are staying on top, and then the proteins you want to detect are running through the lateral flow. And you will see that once you enter the sample and probably add some buffer, because you need some buffer to reveal and to move the sample along the paper, you can see that the golden particles and antibodies will interact now with the proteins. They will move along the lateral flow, and they are going to interact with antibodies. They are going to do the same as we have in the LISA system, but now everything is in the paper. And in a couple of minutes, you will have the revealing, so the appearance of the control line and the detection line. So this is really very, very simple, very interesting, because give a very efficient result very fast. So we have very nice results here. You see, for example, the control line is the same almost, and then you have the different intensities of the lines that correspond to gold nanoparticles, and these are related to the concentration of the analyte. To make this device more efficient, we have been working with different strategies to make this more sensitive. For example, what we did is we connected to gold nanoparticles some enzymes, for example, HRP, and by using some substrates that are mostly using in the immunostaining techniques, we were able to amplify the color. You can see here, for example, using TMB or using some other substrates, we're able to get much more improved color. You see here from the bare to the modified with enzymes, you see a much more stronger color. And this is really very interesting. Another strategy we use is by changing the architecture of the paper. And you see here that from the standard to the bigger sample pad or bigger sample and conjugation pad, the microfluidics is changing. And we calculated this microfluidics. And what does it mean? If you change the microfluidics, you are changing the time during which the incubation between the antigen antibodies is occurring. And this in terms is related to the change in the sensitivity of the system. As you can see here, we changed the intensity of the signal, and we could find some parameters that give better sensitivity or broad range of response. So you can play around the geometry of the paper. But instead of changing the geometry, we played also with putting some pillars. So we printed some wax pillars onto the paper, and you can imagine that if you have these wax pillars which are hydrophobic, you again bother the microfluidics. So printing different configurations of pillars, we found out that the microfluidics was changing, and again we were able to tune up on demand the performance of the lateral flow thanks to the change of putting these pillars. This device is very interesting, and we can detect even DNA, and here is a case of DNA detection using lateral flow. You can see here, for example, this is a case of Leishmania-related DNA, where we put some amplified DNA into the paper and detect this through gold nanoparticles. You see here images of the paper and precisely of the gold nanoparticles. When we have, for example, this amplified system here with a secondary antibody, you see here the change of the signal and the nice response for different quantities of Leishmania parasites. These kind of devices also are in use by our partner in Barcelona, a hospital for cancer diagnostics, as I said before, using this lateral flow as alternative to radioimmunoassay for some different applications. Also, nice thing is that with paper you can play also with different configuration. Here is a vertical flow assay. So instead of lateral, we have this vertical flow. So the same principle, but now putting the paper into these cartridges. And this was applied for prostate cancer biomarker, PSA, for which it's very important to detect this in the urine. And what we do is we take, for example, 5 milliliter of urine and taking this through this shearing. The nice thing is that now we are pre-concentrating because all the sample is going through these cartridges. So all the analyte is trying to be captured in this conjugate and detection path. And then we have the same operation principle, giving a nice response for PSA in urine with interest for clinical applications. And we went further, even to the detection of heavy metals. For heavy metals, it's difficult to have antibodies, but we could have these through the collaboration with our partner in the United States, who developed an antibody for cadmium complex with EDTA. And through this complex and using immunocompetitive method, we are able to detect up to 0.1 ppb cadmium, which is really fantastic. And you can imagine that changing the receptor, instead for cadmium using a receptor for lead, or even, for example, for uranium, we did also the detection of uranium in water, again with our partner in the United States, and we sent these devices in the United States, and they applied these in connection to a smartphone to the detection of traces of uranium in some underground waters, showing the efficiency of the method. Another interesting paper material is paper, cellulose nanopaper, which is produced by a specific bacteria, giving a very, very nice multifunctional biomaterial with high hydrophilicity and high porosity with interest for different applications. So what we did is we used these nice fibers. These are nano-sized fibers, and dope these with different kind of nanomaterials. So we put, for example, silver, gold nanoparticles, quantum dots inside this paper, either by synthesizing in situ the nanoparticles or putting this ex situ. You can see here this paper without and this with silver and this with gold nanoparticles. So you see the change of the color. Here is in front of the windows of our institute. So we have now this nice colored paper, and what we want to do is use this as sensor. And you can see here now, after setting up this in two-dimensional cuvettes or microwell plates, like in a ELISA system, we are able to measure, for example, the interaction of this piece of paper with different compounds, for example, metimazole, iodide, cyanide, thiourea. Nice thing is that You see here the change of the color, because what happened is that the nanoparticles are interacting with these compounds, corroding, and due to the change of the size, they are changing the color, the plasmonics we call. So we correlate this intensity of the color with the concentration of these compounds, and to use this as simple sensors to detect these compounds. Of course, this is not selective because it's interacting with different compounds, but then interacting with antibodies, we can correlate this with different applications. Now this company, so the spin-off company we have created, is working in Barcelona since several years with different projects. So now 5 years that is operating with the idea to develop professional self-testing in vitro diagnostics with interest for different applications, and with image support and connectivity with interest for different diagnostics applications. Let me show you now in the coming, I have still some minutes, the use of nanomotors. So this is a very interesting device. We have been working with this since many years in collaboration with Professor Joseph Wang in the United States through a NATO project that I was coordinating. So these devices are very interesting. So what we do is we use the same membranes I showed before, alumina-porous membranes, where we, by using electrolysis, we can electroplate different kinds of metals inside. For example, silver, gold, even magnetic layers, platinum. And after dissolving the membranes, we are able to get these nice cones that contains, in this case, for example, platinum particles. You know that platinum is a catalytic material that can transform hydrogen peroxide to water and oxygen. The oxygen that is coming out from these tubes is serving as a propulsing force. You see here, these devices are moving around in the water, and given the fact that they have magnetic properties, we are just changing the orientation. They move or go around. What we do is we correlate this, connect this with receptors, for example, with bacteria-related receptors like lectins or some hydrophobic molecules, and use these motors to capture different kinds of compounds. And you can see here in this example how these tiny devices going around in the water, swimming, are capturing bacteria, for example, in drinking water, or even in some Jewish water, Jewish liquids, or seawater, or they are able even to distinguish between the different kind of bacteria. For example, can distinguish E. coli in presence of Cerevisiae. You see here this motor is trying to capture E. coli, but not yet. So you see, he's trying to capture it, and you'll see with now with this green is capturing. So these devices are selective. The idea is now to use these after collection with magnetic field, bring these to the sensing surface. So this is the idea behind this. We also use these devices to enhance the interaction between antibodies and antigens, so improving the diffusion, which is very important for the efficiency of the immunoreactions to occur. And we found out that the system that was assisted by nanomotors was more sensitive than the system that was not assisted. So this is a very nice application of these engines. Finally, in the next 5 minutes, I'll try to show you what we're doing with graphene. This is a very interesting material that gave the Nobel Prize to 2 professors in Manchester since 10 years ago, and we are working together in a European project, Graphene Flagship, which is one of the biggest projects. Sorry. So based on this material, we have also created a spin-off company, Graphenica Lab, that is working with graphene. And here is a video showing what this company is doing. So based on the exfoliated graphene. So please see this video, 3 minutes probably.
A dream, a drawing. Technology is on our side to capture what is buzzing around our imagination. It does not matter if it seems impossible. Why not invest in ingenuity, talent, and technology to achieve intelligent, versatile, cost-efficient, and robust solutions? Why not print our future? This way, we can face all the challenges that appear day after day with new scenarios in such varying environments as transportation, logistics, home, office, or the internet. Graphenica Lab has succeeded in starting from the basis of graphene, the finest, lightest, and strongest known material, the best heat and electricity conductor, almost transparent, flexible and elastic, an abundant element in nature, sustainable, and offering almost unlimited fields of application. Starting from a graphite exfoliation process, we can transfer the properties of graphene to any material, making it intelligent and enabling it to become a sensor for any physical, chemical, or biological task. Even for heating, we do this through a proprietary technology, wax printing membranes, accepting a wide variety of water-based inks at a highly efficient cost, and which can be mixed with biological material, opening a door of infinite possibilities in terms of biosensors manufacturing. This system means a world of unlimited creative possibilities, no matter the shape or the size or the final use. Wax printing is possible on almost any type of surface, both flexible and rigid, curved or sharp, performed in-house or in-field, and the results are spectacular. Chemical sensors, physical sensors, biosensors, tactile surfaces or not, supercapacitors, energy harvesters, NFC tags, and more importantly, a practically unlimited range of applications in internet-related fields, including smart transportation, smart home, smart office, smart grid, or smart packaging, as well as wearable devices for diagnostics. Because with Graphenica Lab, we have the technology today that will be indispensable tomorrow.
So this is what this company is doing, and based on the advantages of graphene, there are different advantages, but one of the most important is the high electrical conductivity offered by this material that we are taking advantages. Also, very important is that graphene, in this case graphene oxide, is very nicely processed in suspension form, even in water, which is very important for use in biosensors. Of course, graphene based on the carbon has a very known chemistry, and we can connect it very nicely with any kind of receptor. We are working with aptamers, we are working with antibodies connected to graphene, and use this for different kind of biosensors. One of the most important things we are very much interested is to apply this graphene in very simple devices like paper, plastic, either for optical or for electrical related applications. And you have 2 reviews we wrote in Advanced Materials if you want to get some ideas about how to apply this exfoliated graphene to different kind of sensing systems. One of the applications I'm going to show is related to the quenching of fluorescence. This is a well-known phenomena where a donor and acceptor should be controlled in terms of distance for this phenomena to occur. In fact, for the conventional molecules like fluorescein or rhodamine, for example, for quenching to occur, the distance is around 10 nanometer. But in the case of graphene, this distance is even higher, 30 nanometer. With this in mind, we try to see first what happened with the quenching capability of graphene, and we compared the quenching capability, for example, of graphene toward cadmium selenide fluorescence. You see here almost 100% quenching when cadmium selenide is in contact with graphene oxide. And in fact, in comparison to graphite, carbon nanotubes, or nanofiber, the quenching capability of graphene oxide is almost 90%. With this in mind, we designed a very nice system. We call this digital light response system, where bacteria is detected with very high efficiency. So based on the quenching capability of this graphene oxide toward quantum dots during interaction with antibodies. So this is a very sensitive system that we designed and then applied this is a patented technology that we put onto the paper, and you can see here how it works, just to have an idea through this video, how this operation is occurring. So we first prepared the paper substrate using paper of different porosity and sampling different paths according to the application. Now we apply the cadmium selenide quantum dots onto the paper, and you'll see what happened. So first of all, these cadmium selenide quantum dots that are inserted to the paper are excited by UV visible light. So you'll see now the revealing of the photoluminescence upon excitation. So we have this nice light coming from these quantum dots. And once you introduce the graphene oxide suspension, you see the quenching is occurring. And due to the interaction, because of the antibodies, we are able now to correlate the change of the color with the present substance of the bacteria, and even quantify their concentration based on the measuring of this image of the light coming from this quantum dots. So this is a very nice system, very simple system that we use, of course, after some optimization, and we are able to apply this in some samples with interest for safety and security. Another example is related to the detection of contaminants in water, and here we are using some graphene quantum dots, and using a piece of paper, we are able to detect some interaction in very simple mode. Let me show you also this other video that explains how simple it is and how nice we can detect these different contaminants. First of all, we print onto the paper by using wax printing the different wells where we are going to introduce the graphene quantum dots. You see here again, we have graphene quantum dots onto the paper, and afterwards, again, we'll see the revealing of the photoluminescence. So we have a very nice signal first coming from the graphene quantum dots. So when we introduce the sample with different kinds of contaminants, you see the quenching is occurring. So we see that the sample has or not this contaminant based on this interaction. These are some PP stacking we are inducing with the quantum dots and the sample containing this contaminant. This is integrated in a smartphone, so this piece of paper is now read through this smartphone and getting the result. So this is really very interesting. We are working with different European projects for all these projects I showed you, with different partners, even recently with microbiome-related project with interest for liver cirrhosis detection, early diagnostics. Finally, I'd like to summarize that, as you have seen, there are a lot of possibilities coming from integration of nanomaterials with different kinds of platforms to build simple devices with interest for the detections in diagnostic area, in the clinical area, in environmental monitoring, safety and security, food testing, and many other applications. So I'd like to let you know that we wrote also several reviews on this topic, so we have a lot of information in this if you want. And of course, I would like to thank all my collaborators from different countries. I wish that some of you would like to come, and you are welcome. So just write me an email. If there is any possibility for you to do a research stay in our group, you are very, very welcome. We are a very international team with people from around 15 nationalities working together, because the science needs collaboration and the science has no frontiers. So you are welcome. So thank you very much for your attention. And if you have any question, I'll be very happy to give response. Thank you very much.
Thank you very much, Arben. It was an impressive presentation, and thank you for giving us a flavor of this very nice field for me, biosensor and sensor field. I hope that also other colleagues will be attracted to this field. Thank you very much. And I have some questions, but first of all, maybe some other colleagues, some other participants want to ask you questions. So I give them the opportunity to ask questions. Please, if there is a raise a hand.
Ana Maria Gurban, if it is possible.
Hello.
Thank you for this presentation. It was very, very interesting for me. I would like to ask you for the biosensor design for cancer diagnosis and detections. When you immobilize the cell, the tumoral cell, how you are performing the immobilization, and if you can use the same biosensor for two or how many assay?
Yeah, this is a very important question. In fact, the work we did with cancer cell was done simply to show that when we just put the cancer cells on top of these electrodes without any kind of immobilization, just putting a sample that containing these cancer cells on top of these electrodes, and based on the interaction, of course, with it was a simple adsorption, nothing else. We are able then, through the use of gold nanoparticles, to detect, after gentle washing, of course, we are able to detect these cancer cells. But what we did with the case of circulating tumor cells, we use the magnetic beads, and we are able to capture the cancer cells first through magnetic beads, and then separate these by magnetic separation, and then put the collected magneto sandwich onto the electrode. So there are two different approaches. And these are standard approaches. Yeah, thank you.
Thank you. Thank you very much for your answer.
Please, sir, if there is another question.
I have two questions. First of all, I want to say that it was a very interesting talk, and I observed a very large range of principle and application of this work. My question is, in this large range, did you try to work also with small molecular imprinted polymers?
Yeah, I didn't show, but we have also a lot of works with molecular imprinting polymers. We have been working for some antibiotics detection, and we are currently working in this microbiome project to synthesize our own MIPS using even some nanoparticles. So this is really very, very interesting. MIPS are very, very interesting material and technology for biosensors. And we are using this in connection also to impedance spectroscopy. So using some electrical measurements upon recognition done by these molecular imprinting polymers.
Okay, then perhaps we could establish some relations in the future.
Yeah, we will be very much interested. So
in this field, yes,
yeah,
with our director, which she is also working in biosensing. So perhaps we can put something together. And the second question is more practically. I saw that you developed a spin-off. This spin-off, it's private, or it's a joint venture with the university? Which is the property of this spin-off?
This is a joint venture. In fact, in these two companies we have, this is a joint venture between private partners, and also university, our institute, two institutes in fact, Catalan Institute of Nanoscience and Nanotehnologii and ICREA, because I belong at the same time to two institutes, and the two institutes are also partner in these spin-off companies, because yeah, the idea behind this is to make possible that our technologies are useful and go in the market, and we have a big support by our institute, and both institutes are very much interested for these companies to work and to be efficient. So during some times of incubation, let's say, inside the institute, they are now independent, of course, because this is a totally private activity now, although the founders are public, but they have their own activity. And we are trying to collaborate, of course, and have some projects together, but they are independent companies now.
And the staff is part-time from the institute and other from the spin-off, or the staff which you use, the staff, personnel?
The staff are full-time employed there. So they are, some of them, my previous PhD students, postdoctoral students, and some new people hired from different places. They are full-time employed there. And we are just now collaborators, so consultants, and I am the head of the research, of course, director of the research for both companies, but my position is as an external, so, but they are doing their own job.
Thank you very much, and congratulations for the talk.
Thank you very much. You are very welcome.
If there is another question. In this case, I have several questions, Arben. First of all, do you think that from your experience, the impedance spectroscopy is reliable for mass production sensors, and to use is friendly to use for I don't know, domestic application?
No. In fact, this is very important, and in fact, impedance spectroscopy is a very sensitive method and very much affected by any kind of reproducibility of the sensor. So if you do not have a technology to print a very reproducible sensor, this is very, very sensitive. You know, impedance spectroscopy is very sensitive to the changes in the surface, any kind of, in addition to the surface, of course, if you do not control very well the non-specific absorption and everything, so it's very sensitive. But once you control the technology of the fabrication, and in fact what we are doing is we are using inkjet printing instead of screen printing. Inkjet printing is a very, very much more reproducible technology with a very nice surface, and using nanoparticles as a sensing surface. And based on that, and based on the use of some simple protocols, we are also connecting these impedance-based measurements with a smartphone, so with a special program. We are offering, and we are in fact working for COVID, also for some other application with this project, European project. So I couldn't show you today because we still need to publish this. But I mean, we are very happy with impedance spectroscopy, controlling the quality of the electrodes, and of course all the procedure, and then the connection with smartphone. So this is really very nice and very sensitive.
Thank you.
Because it's it's a direct measurement, label free. So this is one of the advantages for real applications.
Thank you. And about the PCA sensor, do you succeed to put on the market this sensor, or is still ongoing?
No, this is not in the market, so probably our company is, but it depends on the collaboration, the contract. But we just demonstrate this in the lab that it was working with some synthetic samples, spiked PCA into urine, and it was working fine. But it's a matter of it depends. So for the moment, it's not in the market.
And the last question is about spin-offs.
Yeah.
So, how much time do you think that spin-off need to invest till the mass production for sensors? Let's say in this field for sensors. Do you think that in 5 years spin-off will succeed to have a mass production, or maybe 2 years, or what do you think?
Yeah, I think there are different ways because depending on the kind of the spin-off, and there are different formulas. So because spin-off companies generated from universities, research centers like us, the idea is to work with new technologies and develop certain technology readiness level. You know, TRL levels of the technologies are different. They are from 1 to 9. And depending, so as these are very new technologies in our spin-off companies, the idea is to develop, to increase the TRL until certain level. But then you need a big company that will take charge of the mass production. So a small spin-off company is not able to offer product for the market. It can offer open demand, some samples that are really at with very good performance, but once they are sure that this device is working and is working in a real environment, is very reproducible, then there will be contracts with big companies, if interested, of course, to mass produce this. So this is one of the formula. Of course, if you have a technology that is able to mass produce and offer to the market, of course, yes. But in our case, it's related to technologies that need a very, very high TRL. And we are not able to go to 9, TRL 9 to offer directly to the market. But we go, let's say, to 5, 6, and then once we are at 5, 6 TRL, then attracting big companies and then transferring to them. And what our companies are doing.
Yes, our feeling is the same because we have a big debate here about spin-offs, and we think that this is the problem. We cannot develop a spin-off and to go directly to the market. We have to just to be innovative, companies, and then to transfer the technology to big companies.
Yeah, it depends. So I think there are different formulas. So yeah.
Thank you very, very much, Arben, for your presentation, and for sure new partnerships will be initiated after this presentation and after your participation to our workshop.
Yeah, I am very happy, really, and I thank you again very much. It was a pleasure, and I wish I'll visit in person your institute, and you are welcome also to visit us. And I'm really sure that we will have a nice collaborations together. So, and I'll be very happy to have some of you in our lab working together and developing things together. So, thank you very much. It was really a nice pleasure.
Thank you, and see you in October, in the end of October, to the Biochem International Symposium.
Hopefully it will be in person.
Yes, we try to do this at least hybrid symposium.
Okay.
We wait for you.
Yeah, I'd like very much. I have had the occasion to come in Romania several times in some other collaborations. A beautiful country, and I miss Romania, so I'll be very happy to come again. Thank you very much.
Thank you. Thank you.
Okay, have a nice day.
Bye. I think, dear participants, I think that you enjoyed the presentation of Professor Arben Merkoçi, and now we can start the first part of our workshop, and I invite our chairman for this part, Professor Andreescu, to lead this part and to present all the participants and to start the presentations. Thank you and good luck.
Thank you very much. And we will begin just now the first session of our symposium. Is a session about multifunctional materials and nanocomposites. The first talk will be given by our young colleague, Iulia Elena Neblea, and it's about controlled release systems based on material cellulose and polyethylene glycol diacrylate for biomedical applications. And the team which performed research is Iulia Elena Neblea, Ana Mihaela Gavrilă, Tanța Verona Iordache, Anita Laura Radu, Andreea Miron, Ana Lorena Ciurlică, Marinela Dumitru, and Andreescu. So it is a registered presentation, so please put it to nu știu ce ecran, doamne, că screen, final al naibii. Nu venea în cap. Așa.
My name is Iulia Elena Neblea, and today I will present the control release system based on bacterial cellulose and polyethylene glycol diacrylate for biomedical applications. My main subjects for today will be focused on the synthesis of hydrogels, materials used, and some results obtained along the study. Hydrogels are hydrophilic materials composed of a 3D polymeric network that has the property of retaining a large volume of liquid due to the presence of hydrophilic groups in the macromolecular structure. These materials are currently a very popular topic in areas such as agriculture for control release of fertilizers, or in medicine for dermal treatments, contact lenses, different control release systems, or in tissue engineering. For example, various treatments with injectable hydrogels based on hyaluronic acid and collagen. Some common polymers that can be used for the synthesis of hydrogels are either synthetic, like hydroxyethyl methacrylate, acrylic acid, polyethylene glycol, polyenisopropylacrylamide, polyvinyl acetate, or natural, like chitosan, arginate, gelatin, chitin, and others. The main purpose of this study is the synthesis and characterization of hybrid hydrogels based on polyethylene glycol diacrylate and bacterial cellulose with potential applications in wound healing devices. For the preparation of the materials, we used a synthetic polymer, polyethylene glycol diacrylate with a molecular weight of 700, bacterial cellulose as biopolymer, a redox initiating system composed of potassium persulfate and sodium metabisulfite, and the final product was subjected to a series of control release tests using cefalexin, an antibiotic administered for the treatment of bacterial infections of skin or soft tissues. The synthesis was first performed by preparing a suspension of polyethylene glycol diacrylate and variable concentration of bacterial cellulose in water. The suspensions obtained were intensively homogenized in an ultrasonic bath. Next, we added the initiating system and kept the vials in one water bath for 24 hours. At the end of the reaction, the vials were broken and the hydrogels were cut into desired pieces. The purification step required to keep the hydrogels in distilled water for 7 days, and the final step was to dry the samples in the oven. In the images below, you can see the hydrogels with variable cellulose content before the purification step. The next step was the characterization of the hydrogels. The same images below capture the morphology of the hybrid hydrogels thus obtained. The arrows indicate the bacterial cellulose fibers that are randomly oriented and form a compact 3D network that tends to absorb more water. Sample 0BC indicates a smooth and compact structure compared to other samples and present bigger pores. We also observed that the smooth structure of the synthetic polymer is covered with bacterial cellulose fibers, indicating that grafting of polyethylene glycol has taken place on the surface of the fibers. The last image of a 100 BC sample indicates an agglomeration of fibers determined most probably by the high concentration, but also the lack of homogeneity. The FTIR spectroscopy showed that the hybrid hydrogels indicate all the characteristic bands for both the synthetic polymer and bacterial cellulose, without major differences due to the variation of concentration. In this way, we observed the specific peaks of hydroxyl group vibration, characteristic vibration of the polymer chain, characteristic bands of carboxylate groups, and also the vibration of the groups specific to primary and secondary alcohols in polyethylene glycol structure. Thermogravimetric analysis indicates the influence of cellulose concentration on the thermostability of hydrogels. As the concentration increases, the thermostability of the hydrogel slightly increases, a fact highlighted by the improvement of the starting temperature for thermal decomposition, which is characteristic of the diacrylate and cross-linked polyethylene glycol molecules. When speaking of hybrid hydrogels, the decomposition stages specific to both synthetic and natural polymer are observed. The first stage indicates water evaporation, the second is determined by the decomposition of polyethylene glycol diacrylate and bacterial cellulose, and the third represents the decomposition of the remaining polymer backbone. The values for total weight loss do not indicate major differences between the samples, and also the decomposition peak for studied samples is slightly shifted towards left due to the presence of the bacterial cellulose. The swelling degree of the samples was also investigated. The study was performed at equilibrium in distilled water and at physiological temperature. Here we observed the influence of bacterial cellulose concentration. The swelling degree increased along with the quantity of bacterial cellulose from 0 BC and showing a peak at 60 BC and slightly decreasing towards 100 BC. Study of cefalexin was also performed. A solution of 2.5 milligrams cefalexin in PBS was used to load the hydrogels. The study was conducted also in PBS for 72 hours at physiological temperature, reaching a maximum release after 24 hours. The listed chart indicates the total concentration of cefalexin released over time for each of the studied samples. We observed that each sample released more than 0.2 milligrams and that 40 BC sample releases the largest amount of drug, indicating an optimum ratio between polyethylene glycol diacrylate and bacterial cellulose. Lastly, our study describes the successful synthesis of hybrid hydrogels based on bacterial cellulose and polyethylene glycol diacrylate. As main conclusions, I would like to say that the hydrogels had a similar behavior in both FTIR spectroscopy and thermogravimetric analysis. The swelling degree studies favors the use of bacterial cellulose in higher doses, and the control release study indicated a constant release of drug, reaching a maximum in 24 hours. This fact actually encouraging the hypothesis of using these materials as targeted healing devices in wound therapy. In present, our hydrogels are currently undergoing biocompatibility and cytotoxicity studies, and as future perspectives, we would like to continue the study by using other active ingredients for controlled release, which might lead to an utterly innovative solution in the field of biomedicine. Thank you so much for your attention.
Thank you very much, Julia, for your presentation. Now the discussion is open. Please ask the author about the results. If there are no questions, I have a question. In your opinion, which bacterial cellulose concentration is recommended for this slow release application? Which is your opinion?
So my opinion regarding this aspect is that we need to use as smaller doses of bacterial cellulose as possible, because 100, as we used in this study, 100 BC bacterial cellulose is too much, and it creates agglomerates in the material. So the best concentration, as we saw in the test, is around 60 BC, 60% or 40%.
Okay, and do you know something about the cost? If bacterial cellulose is much expensive or less expensive than diethylate polymer?
I don't know exactly the cost of bacterial cellulose. This one was produced by our collaborators from University Polytechnica of Bucharest. So I guess the cost of maintaining a culture, a bacterial culture for producing bacterial cellulose is a little bit higher, maybe closer to the polymer price, the synthetic polymer price.
Okay, okay. Thank you.
Thank you.
There are any other questions? If not, thank you again, and congratulations for your presentation. And we pass to the next presentation. It's a presentation which will be given by Andreea Afilipoae. It's also a registered presentation, and the title is Mechanical Properties of Polyamide with 2D hybrid reinforcing agents. And the team which performed this research is Andreea Afilipoae, Zina Vuluga, Augusta Raluca Gabor, George Mihail Teodorescu, Monica Cărăulașu, Rodica Zăvoianu, Octavian Pavel, Alexandra Elisabeta Stamate, Mariam Minculescu, and Mihai Cosmin Corobea. As I saw, the main part is from NeXT-Chem, and some other are from University of Bucharest. Please put on the screen the presentation of Andreea Afilipoae.
Good morning, everyone. I will start my presentation with materials and methods. Polyamide represents a class of thermoplastic polymers with various properties and applications. However, it has some drawbacks, such as low thermal stability, poor dimensional stability, or low electrical conductivity. Graphene is used to overcome some of these drawbacks thanks to its exclusive electrical, optical, mechanical, thermal, and chemical performance. Bio-based polyamide is obtained from natural sources, the main source being castor oil. As seen in literature, polyamide's properties can be improved by using different fillers, such as glass fiber, in several directions like mechanical modulus, dimensional stability, thermal stability, impact properties, and so on. In this context, finding new classes of fillers with lower impact on the environment and good potential for mechanical properties of polyamides can be an alternative for greener nanocomposites in the main applications areas. The purpose of this study was the investigation of mechanical properties of polyamide 1010 with a new class of two-dimensional hybrid fillers obtained through melt processing. A full bio-based polyamide 1010 was used as the polymer matrix with a hybrid two-dimensional nanofiller. This consists in molybdenum hydrotalcite and different concentrations of graphene oxide or commercial graphene nanoplatelets. These composites were obtained in dynamic conditions through melt processing, and the resulted samples were analyzed from a mechanical point of view using dynamic mechanical analysis, tensile and impact test. In this graph, it can be noted that the Young's modulus increase depends on the percentage of graphene oxide. An increase of the modulus over 45% of the sample with the highest percentage of graphene oxide demonstrates the reinforcing effect of the two-dimensional hybrid agents. The same trend was observed in the case of tensile strength, which generally increases with the graphene oxide content of the two-dimensional hybrid agent, except for the 15% graphene oxide sample in which the tensile strength decreases. This phenomenon can be explained by the formation of agglomerates. In the case of the 2D hybrid agent containing graphene nanoplatelets, the highest increase in mechanical properties was observed, over 78 of the Young's modulus and over 62% of the tensile strength. We observed a trend of drastic decrease in axial strain at break for all samples with 2D hybrid agents, evidence of their reinforcing effect and increased rigidity of composite materials. Also, there is a decrease in the value of impact strength compared to the NeXT sample of over 30%. The results obtained by DMA analysis correlate very well with the results obtained after the tensile test. An increase in the value of the storage modulus was observed, along with an increase in the percentage of graphene oxide. The reinforcing effect is also observed from the tan delta by moving the temperature corresponding to the maximum peak to the right. The conclusion of this study consists of these previously presented reinforcing agents that show a promising way for the reinforcement of bio-based polyamide 1010. As observed, these 2D hybrid agents proven their efficiency as reinforcing agents by increasing Young's modulus and tensile strength. These results show that polyamide 1010 reinforced with these 2D hybrid agents can be useful in the automotive industry. Thank you for your attention.
Thank you too for your presentation, Andrea. And now it's possible to put questions or to make comments about this presentation. Please, you are invited to do this. If you don't have a question, I have one. I want to ask Andrea, which is the reason that you used graphene oxide instead of graphene as such? Which is the advantage to use graphene oxide?
For increase the compatibility with.
Did you try also to work with graphene and after that with graphene oxide, or directly because you know from literature that is better with graphene oxide?
I also graphene.
You work with graphene also? Yes. No, I think that you don't
And the results are lower.
Are lower. Okay. So you prove that it's better to work with graphene oxide. Very
Yes.
good. Thank you very much for your presentation. And now we pass to the next presentation, which will be given by Monica Cărăuleașu. The title is on the bio-based polyamide with flame retardant properties. And the team is Monica Cărăuleașu, Zina Guluga, Cristian Andy Nicolae, George Mihail Teodorescu, Andreea Filipoae, Rodica Zăvoianu, Octavian Pavel, Alexandra Elisabeta Stamate, and Mihai Cosmin Corobea. The team is also from CECHIM and the University of Bucharest. And now I want to ask to our organizing team to put on the screen the presentation of Monica Cărăuleașu. Please. Nu mai merge, nu?
Good morning, everyone. Polyamide was proven among polymer thermoplastic material as one of the most important engineering thermoplastics. They have mechanical, thermal, and chemical properties such as high strength, stiffness, and good temperature resistance. Graphenic type of fillers and layered double hydroxides were proven as interesting choices for improving polymer properties. Polyamide 1010 is one of the noteworthy commercially produced engineering polyamides. Bio-based polyamides are a new class of polymers which can be obtained from renewable resources. The purpose of this study was to improve fire behavior of polyamide materials with a new class of halogen-free flame retardants based on hybrid structure containing both graphene and layered double hydroxide structures. Some flame retardant agents may induce strong decrease of the thermal stability of the polymer material. Therefore, in this study, we verified in a complete picture both thermal and flame retardant properties of the composites with two-dimensional hybrid fillers. As materials, we used the bio-based polyamide 1010 matrix and hydrotalcite with molybdate anions with different graphene oxide concentrations, or hydrotalcite molybdenum with graphene nanoplatelets to obtain the composites. The final materials were processed under dynamic condition by melt processing followed by injection molding. Thermal analysis was done with thermogravimetric and differential scanning calorimetry analysis. The flammability of polyamide 1010 composites was performed by means of the limiting oxygen index and vertical burning test according to the ACTM D3801. From the thermogravimetric analysis, we observed that the fillers used show modification in the decomposition process. For neat polyamide 1010, the decomposition is formed by two stages, a main stage preceded by a slight shoulder. Regarding the inorganic fillers, one decomposition stage is observed which is positioned between two stages highlighted by neat polyamide 1010. In relation to fillers based on graphene nanoplatelets, the mechanism is still in two stages, but there is a slight improvement of the thermal stability highlighted both by the increase of onset temperature and by the increase of temperature at the maximum decomposition rate. The melting and crystallization behavior of polyamide 1010 composites were determined by differential scanning calorimetry analysis. The material was heated up to 240 Celsius degree, maintained for 2 minutes, then cooled to minus 40 Celsius degree, maintained for 2 minutes, and reheated up to 240 Celsius degree, the cooling heating rate being 10 Celsius degree per minute. All measurements were performed with helium with a 25 milliliter per minute flow rate. It can be observed that the glass transition temperature increased in the presence of fillers from 37 to 42 Celsius degree. On the crystallization curve, with the addition of fillers, the crystallization increased. The fillers acted as nucleating agents, where hydrotalcite molybdenum with graphene nanoplatelets fillers was the most efficient on the non-polar units. On the second heating, there were two melting temperatures corresponding to an unstable phase, respectively to a stable phase that can be found in the literature. In the presence of hydrotalcite molybdenum with graphene nanoplatelets, there is a reduction of the unstable crystal phase of polyamide 1010 with melting temperature at around 190 Celsius degree. In this case, a significant increase of the weight of crystalline stable phase is observed. The limiting oxygen index value of neat polyamide 1010 was found to be 26.3%. The addition of hydrotalcite molybdenum with graphene oxide in the polymer matrix increased significantly the limiting oxygen index value. The best improvement was found for the sample with hydrotalcite molybdenum with 15% graphene oxide with limiting oxygen index value close to 30%. The results are in good correlation with the result from vertical burning test. According to this test, neat polyamide 1010 and the composite with graphene nanoplatelets burned and produced heavy flaming drips which ignited the Croton indicator. So, neat polymer and the composite with graphene nanoplatelets were classified a V2 level. By contrast, the vertical burning measurements highlighted the significant improvement in flame retardant action of hydrotalcite molybdenum with graphene oxide fillers in terms of the reduced flammability of the melt dripping, classifying these composites at the highest level V0. In conclusion, improvement of the flame retardant properties of polyamide was possible by using hydrotalcite with molybdate anions and graphene oxide fillers. In the case of composite with graphene nanoplatelets, we saw two steps of decomposition mechanism as neat polyamide 1010 compared with the others with one step of decomposition. The best results for limiting oxygen index was found for the sample with hydrotalcite molybdenum with 15% graphene oxide. Therefore, the flame retardant properties was brought by LDH structures. Thus, the V0 level was obtained for composites with graphene oxide compared to the composites with graphene nanoplatelets with V2 level. Thank you for your attention.
Să mai aude ceva? Da?
Thank you very much also for the presentation. It's an interesting one. Monica, if she has
I didn't hear you. Maybe you have a low connection.
I will put again the question because if you have an explanation why composites with graphene oxide are have a greater oxygen index than that with graphene.
I think that graphene oxide, because it has a bigger concentration of oxygen and maintain the combustion compared to graphene that it don't have oxygen.
So you put a compound which has oxygen. This is
Sorry, I can't understand. It's a low.
Again, I had difficulties with my connection. So please, if you can now.
So I will answer.
Okay. Yes, please.
Can you hear me?
Yes, now I can. My connection is unstable. This is why we have difficulties. Please.
The sample with graphene oxide formed inert gases and form a barrier against combustion.
Okay, I have not but I'll speak without video because it's impossible to connect video. So I will. Now you hear me?
No, it's interrupted.
Do you hear me? It was interrupted again. The connection is not stable.
I can hear.
So, George Mihitorescu, also, it's... Poți să te duci la Ștefan să-i zici ceva? Că nu se aude. Da? Now, I will make again the presentation. Team is George Mihail Teodorescu, Zina Buluga, Cristian Andy Nicolae, George Mihail Teodorescu, Andreea Filipoae, Rodica Zăvoianu, Octavian Pavel, Alexandra Elisabeta Stamate. Nu, am citit greșit. George Mihai Todorescu, Zina Vuluga, Florin Oancea, Mihai Cosmin Corobea, Andreea Filipoae, Monica Cărăulașu, Cristiana Di Nicolae, and Raluca Gabor. All are from NeXT-Chem. Properties of recovered polypropylene from waste mass waste. Please make the presentation.
Good day, everyone. In recent decades, and especially in recent years, the production of surgical face masks has increased considerably. This object is heavily used in hospitals, and as we all know, since the start of the pandemic, everyone had to wear face masks most of the time. This level of consumption leads to considerable quantities of waste and pollution for the environment. The objective of this study is to analyze and compare the mechanical, thermal, and nanomechanical results of polypropylene recovered from masks to capitalize this waste and to find an application for it in an industry. Therefore, two samples, one made only of masks and one with mask and mask cords, were prepared in dynamic conditions by melt processing, extrusion, and injection molding. This was done in order to obtain test specimens that were furtherly analyzed from mechanical, thermal, and nanomechanical point of view using the mentioned devices. From a mechanical point of view, we noticed an increase in impact strength with almost 13% and 40% for axial strain for the composite with mask cords compared to the neat masks. This indicates that the mask cords function as an elastomer within the composite, improving its elastic properties. As a result, both Young modulus and tensile strength present decreased values for the composite with the mask cords, indicating lower stiffness. This behavior is confirmed also by DMA analysis, showing a lower value of storage modulus for the composite with the mask cords compared to the neat masks. We also decided to compare the results obtained from our analysis with values from data sheets of industrially used polypropylene, both homopolymer and copolymer. From a thermal point of view, TGA analysis shows increased thermal stability for the neat polypropylene from masks compared to the composite with mask cords. This proves that the mask cords behave like an elastomer and interacted with the polypropylene from masks. Dynamic scattering calorimetry analysis shows a negligible increase of the melting temperature and a decrease of melting enthalpy for the composite with mask cords. From a nanomechanical point of view, nanoindentation of our samples showed an increase of reduced modulus with 38% and hardness with 30% for the composite with mask cords. These results correlate well with the obtained data from mechanical analysis. However, nanoindentation also showed that neat polypropylene has a higher degree of elastic recovery compared to the composite with mask cords. Nanoscratching indicates that the introduction of mask cords in neat polypropylene leads to a reduction of both surface roughness and coefficient of friction, meaning that the composite will have a lower wear degree. The conclusions that can be drawn from this study is that worn masks can be used in order to obtain a viable polymer matrix for further use in an industry. Mask cords can be used as an elastomer agent when they are melted and mixed with the masks, and last but not least, further investigations are required in order to better understand this polymer matrix and to propose a viable application for it. I would like to thank Team 12 for all the help given and you all for your attention.
Thank you very much. I hope that you can hear me because again we have questions? Yes, sir. Again, we have troubles. Do you hear me now? Do you hear me now? If there are some questions to George.
I can't hear you clearly.
Okay.
Pretty weak.
Okay, there are some problems with the connection. So my question is the following: do you think that it's possible to recycle the polypropylene from the mask? There are some very big difficulties, or it's possible and we have to work on it? What is your opinion?
It isn't difficult. The only thing that we have to take care of is, as you know, some face masks have a much tougher part on the nose. When we put the face on the mask, there is a slight metallic part or plastic, depending on the mask, that needs to be removed. So when recycling the masks, we should only use the face mask and the mask boards without the hard part. So no, it shouldn't be difficult.
Okay, this is a good news, because there are a lot of waste masks, and it will be a big danger for the environment in the future.
Yes,
so
especially
it's a good news.
now.
Thank you.
For sure.
Thank you very much. Okay, now we have another panel, and the title is Spectral Characterization of Macroheterocyclic. Do you hear me or not?
Yes, it's okay.
Again. Yes. So, nickel, zinc, SPC, MC, and the team is from Universitatea Clarkson, Sofia Press.
Hello everyone, my name is Liviu Olteanu. I'm PhD student.
I think that now we can make the presentation, please.
Doctoral School of Materials Engineering, and I will present a paper with the title Spectral Characterization of Macroheterocyclic Compound, Nickel tetramethylpyridylporphyrin, Zinc tetrasulfophthalochanin, Microcrystalline Cellulose. Macroheterocyclic compounds, such as porphyrins and phthalocyanins, have unique physical and chemical properties. The formation of such adducts involves intense interactions between two differently charged microcycles, this interaction being strongly influenced by the nature of the metal inside. The investigations of spectral properties of this macroheterocyclic compound are achieved by FTER and Raman analysis. Many efforts have been done in the area of porphyrins and phthalocyanins, supporting them on different substrates, metallic oxides and cellulose, knowing that longer photochemical stability is essentially important for their longer activity as luminescent materials. Also, their deposition on microcrystalline cellulose have been investigated in order to stabilize their triad structure. Microcrystalline cellulose can form hydrogen bonds both within its own structure and with other molecules that may remain attached to the polymer chains by localized interactions. In order to obtain more stable and more efficient photocatalysts, some porphyrins and phthalocyanins could be entrapped into the polymer chains of some cellulose derivatives, such as microcrystalline cellulose. The anionic porphyrins and phthalocyanins are rather closely packed in the submicroscopical pores of microcrystalline cellulose. It has been shown that cellulose is composed of amorphous and crystalline. When microcrystalline cellulose is added to the solution, cellulose-to-cellulose hydrogen bonds are replaced by cellulose-to-solvent bonds due to the strong interaction with glycosidic chain segments, thus providing the swelling of the polymer. The porphyrin or phthalocyanin complexes could be absorbed into microcrystalline cellulose by entrapping between the glycoside chains and the crystalline area of microcrystalline cellulose for low concentrations of complexes, or by entrapping in amorphous domains for high concentration of these complexes. Negatively charged porphyrins and phthalocyanins complexes could form molecular associates as dimers and oligomers even in diluted solutions. The well-defined structures of microcrystalline cellulose prevent the aggregation of the phthalocyanin molecules because of their location in the intracrystalline voids and galleries of the supports. Also, microcrystalline cellulose, being strongly hydrophilic, supports the co-adsorption of water in the galleries and cavities, which improves the diffusion of reactants and products. This is major reason for the lower catalytic activity of the phthalocyanin supported on microcrystalline cellulose. Here are presented the chemical structures of nickel tetramethylpyridyl porphyrin in the left and the zinc tetrasulfophthalocyanin chemical structure in the right. By coupling porphyrins with phthalocyanins, heterocomplexes useful for photodynamic applications could be obtained. Their association is based on electrostatic mixed dimers by pairing porphyrin and phthalocyanin bearing substituents with opposite charges and have higher efficiency and stability. Porphyrins are a class of microheterocyclic organic compounds composed of 4 modified parallel subunits interconnected of their alpha carbon atoms via methane bridges. The heteroaggregate between nickel tetramethylpyridyl porphyrin and the zinc tetrasulfophthalocyanin can adopt a close contact face-to-face mutual configuration in order to optimize the pi-pi interactions between aromatic cores. The spectral evolution of a solution of nickel tetramethylpyridyl porphyrin in dimethyl sulfoxide upon addition of zinc tetrasulfophthalocyanin is shown in the figures. Nickel tetramethylpyridyl porphyrin, zinc tetrasulfophthalocyanin, and the microcrystalline cellulose have been used as pure materials without any supplementary procedures from Aldrich Company, while their heteroaggregate has been prepared in the laboratory after a standard procedure. The infrared spectra were measured using an FTIR spectrometer Vertex 80 with ATR, and the Raman spectra were recorded with a portable Xanthus 2 TM Raman analyzer equipped with 2 laser sources and 2 detectors. Here we have the FTIR spectra for nickel tetramethylpyridine porphyrin with the red line and the zinc tetrasulfophthalocyanin spectra in the middle, and the FTIR spectra for the heteroaggregate in down. The IR spectra of aromatic condensed benzene rings of metal phthalocyanins and porphyrins include the most extensive set of stretching and bending vibration bands, stretching vibration of carbon-hydrogen bonds 3046, 3060 cm-1. Relatively low intensity and skeletal carbon-carbon vibration 1580, 1610 cm-1, with intensities are varying for moderate to very low. The frequency of 1580 cm-1 is caused by stretching vibration of the carbon-carbon bonds condensed with the Pyrrole cycle. Intense vibration in the region of 947 cm-1 are totally symmetric stretching vibration of carbon-carbon bonds of aromatic ring. Plane bending vibration of carbon-hydrogen are in the ranges of 1283 and 1289, 1158, 1167, 1091, 1096, and 1033, 1060 cm-1. Out of plane bending vibration of C-carbon metal bonds of metal phthalocyanins and porphyrins appear in the range of 720 and 780 cm-1. The broad, medium intensity porphyrin deformation band at 500 cm-1 is shifted to a higher frequency in the metalloporphyrins. The most intense band in the far infrared spectra of metalloporphyrins appears at 350 cm-1, and the adjacent weaker band at 380 cm-1 can be attributed to the metal-free ligands and metal-nitrogen bond. Vibration of the carbon-carbon-nitrogen group appear as a low-intensity band at 1396, 1407 cm-1, and a very strong band characteristic of all phthalocyanins appear at 3020 and 1365 cm-1. At porphyrins, a high-intensity band is at 1500 and 1530 cm-1, corresponds to vibration of nitrogen-bringing atoms. Stretching vibration, including vibration of the pyrrole and the benzene residues, are in the range of 1420 and 1480 cm-1. The band intensity varies from low to very high. The band at 1594 and 1608 cm-1 and 1573, 1582 cm-1 can be assigned to carbon-nitrogen stretching vibrations. Different for the IR spectroscopic method, which only depends on the chemical composition and phase of the samples, the resonance Raman spectral features also depends on the excitation wavelengths. In the image, I present the Raman spectra for the microheterocyclic compound. In conclusion, to develop new sensitizers based on microheterocyclic compounds. In these papers, we studied the nickel tetramethylpyridyl porphyrin zinc tetrasulfophthalochanin microcrystalline cellulose compound. Spectral properties of the studied materials indicates that this could be a perspective for many applications due to their versatility to change their central metal valence and to attach new substituents. Under this context, this microheterocycle compound is a special compound with proper properties Their capacity of association have been identified by FTIR and Raman spectral techniques, which identified their internal bonds, their capacity to add new axial ligand, and to create larger and versatile spatial structures. Thank you for your attention.
Thank you very much for your presentation. Now we have the possibility to ask or to make comments about presentation. Please. If there are not other questions, I will ask something. You said that in conclusions that a large range of application is possible. Can you give me such examples of application that you have in view in your research?
I think I'm Radhika Ion. I think the student is not there. If it is possible, I could answer just to
yes, of course.
Presentation. We intend to use such a complex system, especially in photovoltaic cells, photovoltaic conversion of solar energy by using DSSS cells. He already prepared, produced the cell, and now if we succeeded this new system, we will use this system to create a good converter of solar energy. At Valahia University, especially in the institute of this university, it is a strong group creating different devices for solar energy conversion. So he will be ready to produce such a application.
Okay, thank you. I think that it's a very important application in view of Green Deal and other European direction. So yes. I think that it's one of the interesting direction also in Romania to study such compounds. Okay, thank you very much.
Okay, thank you.
So we now we will give the next presentation, which will be performed by Maria Eliza Pușcașu. The title is Additive Manufacturing of Zinc Oxide Carbon Nanotube Based Structure for Wastewater Treatment. The team is Maria Eliza Pușcașu, Ștefania Chiliac, Raluca Elena Irimescu, Anca Elena Slobozeanu, and Alexandru Cristian Matei. They are from National Institute for Non-Ferrous and Non-Metals. Please make the presentation.
Hello.
Hello.
Can you hear us?
Yes, yes, very clear.
And do you see the presentation?
Yes, also. Very good.
Success. Okay, good morning. My name is Chiliac Stefania, and today, along with my colleague Pușcașu Maria Eliza, we'll present you a study about the additive manufacturing of zinc oxide carbon nanotubes based structures with potential application in wastewater treatment. During the presentation, we will talk about a few topics, such as a short introduction, the aim of the research, the synthesis of the composite powder and its characterization, the paste preparation, the additive manufacturing of the zinc oxide carbon nanotubes 3D structures, conclusions, future prospects, and bibliography. Industrial production, intensive agriculture, mining, and urban utilization have led to an increase in water use, which have greatly impacted the quality of water available worldwide. Untreated domestic wastewater and industrial effluents contain a variety of organic and inorganic pollutants, wastewater treatment methods with high efficacy that will require less processing time and the production of non-toxic byproducts in the water are urgently required. And for this reason, developing composite nanostructured material for this field represents one of the research trends nowadays. Zinc oxide nanoparticles had shown great promise in the area of water and wastewater treatment due to its high bed surface area, strong oxidation ability, and good photocatalytic capability. The main drawbacks of zinc oxide nanoparticles are related to its limitation to absorb light only in the near UV region, and its wide band gap, which implies a reduced ability to use solar energy efficiently. On the other hand, carbon nanotubes can be modified easily by functionalization of the material surface, and it presents large specific surface area, high chemical stability, and the capability to absorb a great variety of contaminants. Carbon nanotubes' poor dispersion ability and his problematic separation process prevents the use of this adsorbent material by itself. So reinforcing zinc oxide with carbon nanotubes could be an interesting strategy to overcome their limitation. The aim of our research was to obtain 3D porous structures from hydrothermal synthesized zinc oxide carbon nanotubes nanocomposite powder with an additive manufacturing technique for potential application in wastewater treatment. The first step of our study was to obtain the zinc oxide carbon nanotubes composite powder by the hydrothermal technique. For this purpose, functionalized carbon nanotubes powder was dispersed in water, brought to a specific pH with sodium hydroxide, and ultrasounded for 15 minutes, resulting carbon nanotubes solution. Zinc nitrate hexahydrate was dissolved in water and gradually added under magnetic stirring to the carbon nanotube solution previously heated to the 50 Celsius degrees. After precipitation with sodium hydroxide, the resulted suspension was poured into the Teflon reaction vessel and introduced in a 1000 milliliters autoclave for hydrothermal synthesis at 200 Celsius degrees. After the hydrothermal treatment, the nanocomposite powder was filtered and washed with distilled water up to pH 7, then dried in the oven at 100 degrees. The FTIR spectra of the zinc oxide carbon nanotubes composite demonstrates the formation of some bonds between the functional groups of carbon nanotubes and oxygen in zinc oxide, and also the presence of characteristic vibration of zinc oxygen bonds. The X-ray diffraction spectra of the zinc oxide carbon nanotubes nanocomposite samples shows the formation of crystalline phase of zinc oxide as the major phase, and also the presence of carbon from carbon nanotubes chains. Also, using the Scherer equation, the crystalline size calculated in the 101 direction was about 61 nanometers. In the end, the same image of the zinc oxide, the carbon nanotubes sample, demonstrates that this kind of powder can be used for the 3D printing process because it has almost spherical nanometer sized particles required for this method.
The synthesized hybrid zinc oxide carbon nanotube powder was used in order to prepare printable paste. Paste preparation implies the mixing of the powder with a combination of organic additives in order to obtain a homogeneous and printable paste. The mixing was done using a planetary centrifugal mixer. If the obtained mixture had a satisfying viscosity, the paste was inserted in a 10 milliliter cartridge. All the prepared paste were subjected to 2 minutes of mixing with a speed of 2000 rotations per minute and 2 minutes of defoaming with a speed of 2000 rotations per minute. The composition of the non-printable paste can be seen on the slide. The used additives were not compatible with the hybrid powder, reason why the paste were either not homogeneous or wasn't extrudable to the nozzle. Also, in the paste preparation process, we also tested small quantities of paste in order to determine if they are printable. And even if some paste seemed printable, it was not well dispersed by the printer. We tried a various number of organic additives combination in order to obtain the paste with the best behavior for the extrusion-based 3D printing. The paste composition you are seeing now on the slide were printable to various printing parameters, being changed either the printing pressure or the printing speed. In the end, we determined that the paste used to obtain the 3D9 object present the best printing characteristics and a better drying of the green body, which had less cracks than the others. In order to create the 3D object, a square cuboid with dimension of 10 or 5 millimeters were designed with a CAD software named SolidWorks. After the design was done, the cuboid was sliced and printing parameters were set in the 3D printer software. The inner pattern of the design sample had a line-based hedge type with a continuous strands and a distance between strands of 1.7 millimeters. The rotation angle between two successive layers were set on 45 and 145 degrees respectively. Zinc oxide carbon nanotube 3D structure were obtained using the paste, which had the following components: PAI, HPMC, and Twin 80. And the 3D printing was done by a robocasting technique using the 3D bioplotter starter system. In order to obtain the 3D structure, a carbon nanotube zinc oxide homogeneous and printable paste was obtained mixing the hybrid powder with the organic additive mentioned before. The pastes were inserted in a 10 milliliter cartridge, and then using the planetary centrifugal mixer, a defoaming step was applied in order to remove the bubbles from the syringe. Zinc oxide carbon nanotube 3D structure were obtained by extrusion-based 3D printing techniques by dispersing the paste from the 10 milliliter cartridge through a needle tip of 0.4 millimeter diameter, according to the CAD parameter. A printing speed of 8 to 50 millimeters per second and a pressure range of 2 to 4 bars were applied. Scanning electron microscopy image of printed samples is depicted in figure present on the slide. The fiber thickness varies between 420 and 528 micrometers in accordance with the nozzle diameters of 400 micrometers. Strand thickness is between 390 and 430 micrometers, and the distance between strands varies in the range of 1.66 and 1.70 millimeters. The results are in accordance with the printing parameters, which were set before. Results of the EDS analysis of the printed sample performed in only one spot shows the presence of carbon, oxygen, and zinc elements. In conclusion, the conclusions of our study are as follows. Nanostructured powders with the particle size and morphology required for the printing method were obtained using the hydrothermal synthesis. Carbon nanotube zinc oxide 3D structure were fabricated by robocasting technique using hydrothermally synthesized nanocomposite powder and polymeric binders. Various organic binders were tested for the preparation of paste using the 3D printing process. And the quality and proportion of organic additive influences the printability of the nanocomposite paste. Future research has to be conducted in order to characterize the 3D structure. The printing process has to be optimized, and finally, we have to study the potential of zinc oxide carbon nanotube-based 3D structure in wastewater treatment application. We want to acknowledge the financial support of the core program PN 19190101 and ERANET MANUNET NIMP project. And finally, we want to thank our colleagues from Advanced and Nanostructure Material Laboratory from IMNR. This is our selective bibliography, and thank you for your attention.
Thank you also for your very nice presentation, a very interesting field of application and complex research. And now you can put questions. You. I want to ask you, why is the reason that you use nanoparticles? Is not enough to have bigger particles? Because I think that it's more difficult to make nanoparticles. Do you have a reason to use nanoparticles or not?
Yes, we have a reason. Because of the nozzle we are using, it has 400 micrometers. We have to have as small as possible particles. Otherwise, the nozzle can be clogged and the printing will be stopped and the 3D body will not be obtained. It's harder to obtain nanoparticles, it is true, but it's necessary for the 3D printing process. The smaller they are, the better.
Okay, and what kind of pollutants do you intend to destroy?
I'm sorry, I can't hear you.
What kind of pollutants do you intend to destroy with your system? Organic pollutants or what kind? Do you hear me now?
Yes, both organic and inorganic, I think.
Okay. Thank you very much. Thank you very much again.
I have a question, please.
Now
sorry.
We have another presentation. It's,
sorry, we can't hear you very well.
I will turn up my mic. Okay, I have a question for you, please. I don't know if you or your group established, is there a potential for the transfer of this technology towards, there are many Romanian companies working in environmental protection. Are any of them interested in such a technology?
I think they are. We haven't established this yet, but we think they are. It's a very potent way on wastewater treatment, but we still have to study this aspect. Okay,
thank you very much. It is a very important aspect, to find a producer, eventually company to implement this technology in real life conditions.
Yes.
Okay, thank you.
So if you hear me now, I will present the next talk. It is a talk from a team from NeXT-Chem. The presentation will be performed by Mircea Filipescu. The research is studies on parameters influencing the mechanical recycling of some renewable polyesters. And the team is Mircea Filipescu, Mihail Dracne, Nicoleta Dragomir, and Doina Dimonie. Please give the talk.
Hello, my name is Mircea Filipescu, and I will present you the study that has as subject studies on parameters influencing the mechanical recycling of some renewable polyesters. The presentations includes an introduction, some aspects about the degradation of polylactic acid, experiments, materials, methods, characterizations, results and discussion and conclusions. With the growth of the interest towards the use of bio-based material, the importance of the mechanical recycling and the decrease in the incineration activity become more and more obvious. In a circular economy, the mechanical recycling is a good solution to reduce the dependence on the fossil resources. The aim of the present study was to analyze some parameters which are influencing the mechanical recycling of some polyesters and new grades polyesters modified according to own formulation by studying the influence of the drying as a pre-recycling treatment on the PLA, on the polylactic acid meteorological properties. When we talk about degradation of polylactic acid in solid state, we think about the small amounts of water that diffuse into the material and attack the ester bonds in the amorphous phases and generates the decreasing of the molecular weight, the formation of carboxyl and hydroxyl chain and groups, and the diminishing of the functional properties. Polylactic acid destruction has the effect of decreasing the average molecular weight, nonlinear loss of mass, of weight, and implicity of functional properties. Hydrolysis of the ester bond takes place randomly and depends on characteristics of the environment, like temperature, humidity, and polymer characteristics, like material thickness, porosity, surface architecture, macromolecular characteristics of the polymer, crystallinity, molecular weight, optical purity, chemical composition, and wettability. Thermal degradation of polylactic acid is attributed to polymer chain scission, and depolymerization reactions. Degradation of compounding is produced by humidity, temperature, shear regime, oxygen that generate thermohydrolytic mechanism and or backbeating, which results in various types of functional groups. Melt degradation depends on the molecular weight of polylactic acid. Hydrolysis is the predominant mechanism of polylactic acid degradation at a temperature between 150 to 215 Celsius degree. The most important parameter for monitoring degradation is the decrease of molecular weight and the formation of functional groups. The materials used were primary polylactic acid, code PLAFP, modified polylactic acid 1, code PLAFPM1, modified with inorganic filler, and modified polylactic acid 2, code PLAFPM2, modified with thermoplastic elastomer. The influence of the moisture content on the rheological properties of a type of polylactic acid using the films production and of the two variants obtained by its physical modification based on original recipes were studied. In this respect, the meteorological properties of undried and dried polymeric materials were compared. The equipments used were Brabender and roller for classical melt compounding, Götfert rheological extruder for shaping as filaments and granulation, air circulation oven for drying. For characterization was used an equipment for extensional flow of the melt, respectively, indexer which provided the following melt properties: shear force, shear rate, fluidity, melt flow index, melt resistance to flow, dynamic viscosity, melt density, melt sensitivity to change flow condition. The study was performed under the following conditions depending on the type of material: PLAFP granules at 200 to 240 Celsius degree, PLAFPM1 granules at 185 to 255 Celsius degree, and PLAFPM2 granules at 190 to 210 Celsius degree. After analyzing the graphics of PLAFP material, which represent the dependency of shear stress function of piston load and temperature, the following conclusion can be made. The drying process does not affect the values of the shear stress. The shear stress does not depend on the processed material or on the humidity content. Drying does not affect the level of shear stress. Shear demands are developed in the range of 20,000 to 100,000 Pascal. The level of shear stress is dependent on the load and independent of the temperature value. After the assessment of the graphics of PLAFP material, the following conclusion can be made. Shear rate depends on the type of material processed. The moisture content affects the value of the shear rate. The shear rate depends on the moisture content of the processed material. Under the same flow condition from the melt, the shear rate is about 3 times higher for the dry sample. According to the E formula, the shear rate is higher the lower the molecular weight is. Because the shear rates are higher, the molecular mass of dry material is lower, so the material has degraded. The degradation factor value will show the influence of the humidity on the shear rate developed at the flow in extension field of the PLAFP samples. Table 1 shows the influence of moisture content under different shear rates. Degradation is higher at low shear rates because the process of hydrolysis of the ester bond is strong. At high shear stress, the hydrolytic degradation decreases and the thermomechanical degradation is accentuated. The temperature dependence of degradation factor shows the decrease of the hydrolytic process when the temperature increases at 200 to 240 Celsius degree. Most likely, the melt fluidity value in case of dry polylactic acid is due to a predominantly thermal degradation associated with another parallel process of faster melt flow of both undegraded macromolecules together with those with lower weight result from degradation. The degradation factors value in table 12 superunitary, thus illustrating a phenomenon of decrease in molecular weight in all the flow condition study. The lower degradation factor values in table 2 compared to those in table 1 may indicate that fluidity highlights in a less extent of the extraction process. The calculation of degradation factor from the rheological properties of the polymer melt is a quantitative method for illustrating the destruction process of compounding polymer by melt techniques. After the assessment of these graphics, the following conclusion can be made. Lower flow resistance in the case of dry PLAFP demonstrates the existence of lower molecular weight macromolecular chain, probably resulting from a process of thermohydrolytic destruction. The degradation factors shown in table 3 are all subunits and demonstrate that the flow resistance of non-dry polylactic acid macromolecular chain is lower than in the case of dry polylactic acid, which indicates a pronounced process of degradation. After the assessment of these graphics of PLAFPM1, it can be said that the drying process does not affect the values of the shear stress. Shear demands are developed in the range of 20,000 to 100,000 pascals. The level of shear stress is dependent on the load and independent of the temperature value. Degradation factors values are lower than in the case of PLAFP as a consequence of the formulation used to make this type of material. According to these graphics of PLAFPM2, the drying process does not affect the values of the shear stress. Shear demands are developed in the range of 20,000 to 100 Pascal. The level of shear stress is dependent on the load and independent of the temperature value. The low shear rate in the case of PLAFPM2 emphasizes the importance of controlling the moisture content, which is the predominant factor in the degradation of the material by ester hydrolysis. There are no very large variation of degradation factors with temperature and load, according to table 5. Degradation factor values are superunitary and highlight the need to dry the material before use. In the case of undried PLAFPM2, the melt flow resistance is similar to melt flow resistance for dry PLAFP material and 1 0.8 times higher than in the case of Android PLA FPM1. In conclusion, the obtained results show that the study of the rheological properties in the molten space is a useful tool in assessing the melt degradability of renewable polyester and of materials based on these grades of polymer. Additional characterizations are underway to support the melt degradation mechanism described in this study. The results show that both PLA and new PLA-based materials must be dried before any procedure involving phase transformation to avoid the degradation during the melt reprocessing and compounding. The efficiency of the renewable polyester mechanical recycling is strongly dependent on the capability of diminish the melt degradation during each recycling number. The pre-recycling treatment of drying of polymeric materials based on renewable polyesters before each processing cycle is a useful instrument to diminish the melt degradation for reaching desired functional properties. Acknowledgements by bibliography. Thank you for your attention.
Thank you also for your presentation. It's a lot of work inside. Please, if there are questions. If not, I have two questions. First, you said that hydrolysis is higher at a lower share rate. Do you have an explanation for this?
Because the time in the extruder is lower.
Oh, this is a possibility, yes. And the second question, you said that the drying process has no influence on the shear stress, but on the other side, the hydrolysis is the mechanism of the degradation. Why do you suppose that the drying process doesn't influence the shear stress?
I didn't.
So I will.
Can you ask again?
I will repeat. I will repeat and to modify some. It is known that the degradation process of polylactic acid is by hydrolysis, but on the other side, your results show that the shear stress is not influenced by the drying. Why is this?
Because it's a property that depends just only by the characteristic of equipment.
Very good. Thank you.
It's not depends on the material.
Thank you. Thank you. So, thank you very much. We pass to the next presentation. The next presentation will be given by Laura Nicoleta Dragomir, and the title is Study of the Interface into New Practical Interest Compounds Based on Renewable Polymers. And the team is Laura Nicoleta Dragomir, Alina Mustățea, Sorina Iftinie, Mircea Filipescu, and Doina Dimonie. The team is from NeXT-Chem and from University of Bucharest. Please give the presentation.
Hello everyone, my name is Nicoleta Dragomir, and today we will discuss about study of the interface in the new practical interest compounds based on renewable polymers. Table of contents refers to introduction, experiments with materials and methods and result and discussion, conclusion, acknowledgement, and bibliography. About introduction, we will start with a short analysis of the raw materials chosen to make these new materials. Start. About strength, we have biodegradability, low cost, low use of petroleum resources. Weaknesses, it's about low physical-mechanical properties, high hydroscopicity, and poor processability. Opportunities, it's about development of new materials with high applicability, and threats, it's about huge agrarian area of vegetables growing. Polycaprolactone, about strength, we have biodegradability, semi-crystalline polymers, hydroscopicity, and low cost. About weaknesses, we have limited and complex production chain. Opportunities, we have development of new materials with high applicability, and threat, it's about low cost of materials. Following the process of obtaining the new materials by mixing starch with polycaprolactone, we observe that an immiscible compound is obtained. See figure 1, which has two polymeric phases, figure 2, specific starch and polycaprolactone. Because we want to homogenize materials, we add a compatibilizer, see figure 3, so changes appear at the interface of the two polymer phases. About experiments, we have materials, method and characterization, and result and discussion. Materials, we use cornstarch, polycaprolactone and compatibilizer. Methods, it's about non-processing compounding, the classical Bravender, and characterization is about highlighting the surface changes on the morphology and internal behavior of new compounds. The result and discussion, like you see in figure 4, 5, and 6, we obtain new starch-based compound with smooth surface and very well flexibility. Using FTIR analysis, we highlighted the influence of the compatibilizer on the chemical bond at the level of the new materials obtained. We identified the specific bands for starch, polycaprolactone, and compatibilizer. The effect of compatibilizer refers to disappear, appear, or shifting of certain peaks specific to certain bonds. The results obtained of the XRD analysis showed that the morphology of the binary compound is different compared with raw materials. It identified that in the case of ternary compound, the morphology appears completely modified. The crystallinity is reduced almost by a half compared to binary compound, fact influenced by the presence of the compatibilizer. SEM microscopy are made from raw materials, figure 8, binary compound, figure 9, and ternary compound, figure 10. In the case of a raw material, specific morphology is observed from starch, polycaprolactone, and compatibilizer with well-defined morphology with a certain shape and distribution of grains. The binary compound, highlighting the formation of the structure that has intergranular space, so the homogenization of materials is low. In the case of ternary compound, the homogenization tendency is observed, the grains are incorporated, the morphology is almost uniform, and the intergranular space are reduced. In the case of EFM spectra, most materials do not have very smooth surface, but the sample with code 69, 71, 1993 were not as having a uniformity. The best results were identified in the case of sample 69 and 93, whose surface is very smooth, a fact supported by presence of the compatibilizer in the ternary compounds. For conclusion, polycaprolactone starch-based materials have been successfully obtained by MEL processing methods. A compatibilizer was used to generate homogenization of two raw materials, determining the control of materials interface. New starch-based compounds were finally obtained. It can be said the interface control is a very good way to obtain materials with specific properties. Bibliography. Thank you very much for your attention and acknowledgements. Thank you and have a nice day.
Thank you too for your presentation. it's also an interesting field of research. And I want to ask if there are questions. No. Then I have a question. Did you assess which is the concentration of starch, which is better to introduce into melt to obtain good results? Did you hear me? I mean, there is an optimal concentration or not? This is my question. So I think that we have no more connection with the team. So we pass to the next presentation. The next presentation will be given by Elena Cojocaru, and the title is Design of Carboxylated Graphene Oxide Containing Chitosan Composite Electrospan Bioscaffolds. And the team is Elena Cojocaru, Jana Gitzman, Grațiela Gredișteanu, Pârcălăboiu, Eugeniu Vasile, and Horia Iovu. The team is from University Polytechnica from Bucharest. Thank you. Please give the next presentation.
Sorry, good afternoon. Immediately, I put my presentation. Can you see my presentation now?
Yes, yes, please.
Okay, thank you. My name is Elena Cojocaru. I am a PhD student at University Polytechnica of Bucharest, and in the next minutes I will present you my research about design of carboxylated graphene oxide containing chitosan composite electrospoon bioscaffolds. The main objective of this work was to obtain the chitosan carboxylated graphene oxide composite scaffolds with nanofibers architecture using electrospinning technique. Electrospinning is a modern and non-destructive technology for electrostatic generating thin fibers with diameters in the nanometer range. The materials with nanofiber structure possess remarkable properties such as a large specific surface area, high degree of porosity, enhanced stability, and mimicking the extracellular matrix, which make them suitable for applications in tissue engineering. The materials used in this study are chitosan and carboxylated graphene oxide. Chitosan is a natural polymer, very difficult to be electrospun due to its polycationic character and high viscosity. For designing the chitosan fibers, it was necessary to mix the chitosan solution with a synthetic polymer with high molecular weight, such as polyethylene oxide, which increases the electrospinability of chitosan. And on the other hand, the carboxylated graphene oxide is characterized by the presence of carboxyl functionalities linked on the planar structure of graphene oxide sheets and presents outstanding physicochemical and biological properties, such as high specific surface area, good antibacterial activity, and biocompatibility. By addition of carboxylated graphene oxide to the chitosan polyethylene oxide fibers, it is expected to increase the electrospinning properties of the material due to its electrical conductivity. The obtaining principle of nanofibrous materials is based on both non-covalent and covalent interactions that appear between the components of the system, as is represented in the figure 1. Then the obtained scaffolds were cross-linked in the presence of glutyraldehyde, leading to the formation of covalent bonds such as imine bonds. Table 1 comprises the preparation of materials used for obtaining composite nanofibers scaffolds and electrospinning parameters optimization. The obtained electrospin scaffolds were investigated using various characterization methods, such as FTIR and Raman spectrometry for structural analysis, SEM for morphological characterization, water contact angle, in vitro degradation, and the MTT and LDH assays for the evaluation of cellular viability and cytotoxicity. FTIR spectrometry was used to analyze the structure of composite scaffolds and to highlight the new covalent bonds that are formed after cross-linking step. All characteristic absorption peaks from chitosan and polyethylene oxide were identified in the FTIR spectra of nanofibrous scaffolds, demonstrating the successful combination of polymers into nanofibers. The appearance of this peak after blending the chitosan polyethylene oxide with carboxylated graphene oxide confirms the successful incorporation of carboxylated graphene oxide in the nanofiber structure. It is also important to mention the presence of a new strong peak at around 1718 in the FTIR spectra of unwashed samples presented in figure 2b, which is related to the stretching vibration of carbon double oxygen bond of free aldehydic bond from glutyraldehyde structure. The absence of this peak from the spectra of washed samples demonstrated the efficient elimination of unreacted residual glutyraldehyde. Comparing the Raman spectra of the composite scaffolds with the spectrum of ultrasonicated carboxylated graphene oxide, it was observed that the ratio between intensities of D and G bands was significantly increased when an amount of 0.1 and 0.2% graphene was used, suggesting a well dispersion in the nanofiber structure. The sharper to the band of the chitosan carboxylated graphene oxide 0.2% indicates a better exfoliation of carboxylated graphene oxide layers. The composite with 0.5% graphene indicates the presence of disordered structure of carboxylated graphene oxide sheets that generated the formation of agglomerates. SEM images highlighted the uniform, smooth, continuous, and bead-free nanofiber structure of chitosan polyethylene oxide scaffold. The composite scaffolds showed continuous nanofibers with irregular surfaces. As a result of chitosan polyethylene oxide carboxylated graphene oxide interactions, the carboxylated graphene oxide sheets marked with yellow arrows can be entrapped into the nanofiber structure or may be disposed along their surface. A decreasing trend was noted in the mean diameter of composite nanofibers as carboxylated graphene oxide content increased. These changes can be attributed to the electrical conductivity of the electrospinning system, which increased with rising the carboxylated graphene oxide content. The contact angle measurement results presented in figure 5 show two aspects regarding the cross-linking treatment and the carboxylated graphene oxide content. Firstly, it can observe that the contact angle of all cross-linked samples is higher compared to the uncross-linked ones due to the cross-linking reaction of glutaraldehyde with amino groups of chitosan. Secondly, the hydrophobicity of composite scaffolds registered a slight increase as carboxylated graphene oxide amount increases in both uncross-linked and cross-linked materials. However, the contact angle values suggest a sufficient hydrophilic character of design nanofiber scaffolds for potential biomedical applications. Figure 6 shows the in vitro degradation rate of uncross-linked and cross-linked scaffolds in PBS solution after 12, 24, 48, 72, and 96 hours. The mass loss is mainly influenced by the chemical cross-linking and the nature of materials embedded in scaffold. As was expected, the cross-linking step improved the stability of scaffolds through the creation of new imine covalent bonds between the constituents of polymeric matrix and glutaraldehyde. It can be observed that the uncross-linked materials registered a higher mass loss compared to the cross-linked ones, suggesting the efficiency of cross-linking step. The in vitro biological response of all electrospin scaffolds was investigated on fibroblast cells after 24 and 72 hours using MTT viability and LDH cytotoxicity assays. After 24 hours, the number of living cells decreased with increasing the amount of amount of carboxylated graphene oxide, and the lowest cellular viability was registered in the composite with 0.5% graphene. Major improvement in the cell viability and a good proliferation potential were observed after 72 hours in all investigated materials. The LDH results showed no important differences in the cytotoxic responses registered for chitosan polyethylene oxide and composite scaffolds with 0.1 and 0.2% graphene after 24 or 72 hours. But in the presence of composite with 0.5% graphene, the cytotoxic response was visibly higher as compared to the control, which could be correlated with the carboxylated graphene oxide agglomerates, as was observed in Raman, which may affect the cell development. In conclusion, the carboxylated graphene oxide based on chitosan composite scaffolds were designed using electrospinning technique. The effect of carboxylated graphene oxide on the structural, morphological, and wettability characteristics, as well as on the in vitro degradation and in vitro biological behavior, was investigated. According to the in vitro cytotoxicity compatibility assessment, the fibroblast culture showed a reasonable proliferation level after 72 hours of incubation in the presence of composite scaffolds with 0.1 and 0.2% carboxylated graphene oxide. Thank you for your attention.
Thank you too. Very nice presentation, important field also. Please, if there are some questions about this presentation. No, I want to know if you measure some mechanical property of this scaffold.
Yes, these scaffolds were measured regarding the nanomechanical properties using the nano-indentation method.
And the results are enough good for application?
Yes.
Okay, thank you very much. We'll pass now to the last presentation of this part of session 1. The presentation will be made by Ancuța Dinu Iacob, and the research is detection of amino acid L-phenylalanine, L-tyrosine, and L-tryptophan with biosensor based on polypropylene. The team is Ancuța Dinu Iacob, Constantina Petrei, Dorin Descălescu, Irina Georgiana Munteanu, Bulgaru, Ramona Oana Roșca, Gunache, and they are all from the Galaxy University Dunărea de Jos. Please.
Thank you. Good afternoon. And I want to know if you see my presentation. You see me?
Yes, yes, please. Go on.
Good afternoon. My name is Ancuța Dinu, and I am PhD under the guidance of the Professor Constantina Petrei. I carried out 2 researches entitled detection of amino acids, L-phenylalanine, L-tyrosine, and L-tryptophan with biosensor based on polypyrrole. You may be asked why I choose the amino acid study, and the reasons for the choice are represented by the fact that amino acids are those that support the healing and development of tissue and cells. Amino acids are those that contribute to the supply and of energy. And the deficiency or excess of any amino acids can lead to disease such as insomnia, chronic fatigue, digestive disorders, hair loss, anxiety. In the context of living with times of many prohibitions and restrictive measures to prevent the spread of the SARS-CoV-2 virus, numerous articles published by specialists in both psychology and medicine show that isolation and quarantine have affected all mankind, emphasizing and increasing the number of depressive states, emotional disorders, anxiety, and sleep. A study published in 2020 following a survey reports the effects of the pandemic on the human psyche, highlighting the increase in cases of depression, anxiety, and stress. So, phenylalanine, tyrosine, and tryptophan are the amino acids responsible for the balance of the nervous system. They have a lot in common, one of them being the very file in which they are used. So in medicine, these amino acids are used for depression, Parkinson's disease, osteoarthritis, rheumatoid arthritis, phenylketonuria, attention deficit hyperactivity disorder. In pharmacy, it is included in the composition of some food supplements in several pharmaceutical forms, capsules, creams, syrup, and in chemistry they are used for testing of sensor and biosensors. Currently exists a neonatal test named Test GU3 to detect the level of phenylalanine in the blood. This test is an old detection technique, expensive and time-consuming. Tyrosine is an amino acid that is naturally produced in the body from another amino acid called phenylalanine. About I talked now. Tyrosine helps make several important substances: dopamine, adrenaline, thyroid hormones, melanin, and tryptophan is also a precursor to neurotransmitter serotonin, the hormone melatonin, and the niacin. In terms of amino acids detection, numerous device achievements have been attempted but have been shown to be of low sensitivity and low precision. And the purpose of this work is to develop a new, efficient, sensitive, and low-cost device for early diagnosis of appropriate pathology caused by these free amino acids and development of innovative therapeutic approach. We use the next materials, so screen-printed carbon electrode, SP150 potentiostat, galvanostat, the AC Lab Express software, and electrochemical cells with 3 electrodes, reference electrode, auxiliary electrode, and working electrode. And the reagents used in this study was purchased from Sigma Aldrich and used without further purification. So the benefits of this device, respectively sensors and biosensors, are high manufacturing reproducibility, easy of use, low cost, no contamination, and are used in various fields of activity: pharmacy, medicine, chemistry, environment, biology, physics, electronics, market opportunities. The objective of this work was to describe the development of a new biosensor with applicability in the determination and quantification of the essential amino acid L-tyrosine in pharmaceuticals. So the proposed biosensor is made of a screen-printed carbon electrode modified with the conductive polymer polypyrrole, doped with 2 inorganic compounds, respectively potassium hexacyanoferrate trihydrate and sodium nitroprusside dehydrate, and on the electrode surface immobilized lactase enzyme. Why I choose polypyrrole? Because this has a number of features and advantages that demonstrated high biosensor performance, increased stability and sensitivity, high electrical conductivity, malleability, and biocompatibility. And why I choose the enzyme lactase? Because it is easy to use, no cofactors are required, and participates in many oxidation reactions for a wide variety of organic substances. The method for modifying biosensor was chronoamperometry, and this method are an electrochemical technique in which the potential of the working electrode is stepped and the resulting current from Faraday processes occurring at the electrode is monitored as a function of time. This is the current versus time curves registered during electrosynthesis of 2 biosensors, respectively polypyrrole with polypyrrole doped with potassium hexacyanoferrate trihydrate, and the second biosensor was modified with polypyrrole doped with sodium nitroprusside dehydride at 0.8 volts during 19 seconds. The chronoamperometric curves corresponding to the number of biosensors modified with polypyrrole and doping agent 6 biosensors were prepared from the same solution in order to the reproducibility of the preparation method. As can be seen, the differences between biosensors prepared under the same condition are very small, the coefficients of variation being less than 5%. And the second method for detection L-tyrosine was cyclic voltammetry. This is the most used method for the study of redox chemical reaction in solution or of the processes that take place on a certain electrode. We choose to present the study of electrode modified with polypyrrole doped with potassium hexacyanoferrate trihydrat and with immobilization enzyme lactase for the detection of L-tyrosine amino acid. This is the results for electrochemical responses of sensors, sensors before modification. And I present respectively the electrochemical responses of biosensors after modification with polypyrrole and enzyme lactase. The scaling rate plays an important role in electrochemical measurement as it contributes to bringing out the redox processes and greatly influencing the voltammetric answers of the sensor and biosensor. The voltammograms were recorded at 10 scan rates, thus making possible the research of dynamic characteristics and of sensors and biosensor performances. The peak intensity in the case of the biosensors were higher compared to those of the sensors, this being due to the immobilization enzyme and the conductive polymer. And the next step is to study the calibration curves recorded in solution of different concentrations of L-tyrosine. This is the lactase enzyme immobilization procedure on polypyrrole doping agent biosensors for electrochemical determination of L-tyrosine. And for the stability of the enzyme, each biosensor obtained was left for 2 minutes in the presence of butyraldehyde vapors. Detection of tyrosine with the modified screen printing electrodes and validation in real samples. So the purpose of this analysis was to compare the results obtained by the two methods, respectively cyclic voltammetry and heat, with the values declared by the producers in the leaflets of the pharmaceutical products used. Validation of these biosensors achieved by its successful application in the analyzed pharmaceutical product contains the essential amino acid tyrosine in various concentrations and from different producers. So in conclusion, the polypyrrole films with the ions doping potassium hexacyanoferrate trihydrate and sodium nitroprusside dihydrate successfully synthesized through chronoamperometry method by depositing screen printed carbon electrode. The biosensors were used in order to detect L-tyrosine in standard solution and pharmaceutical products, and it results the polypyrrole doped with potassium hexacyanoferrate trihydrat and with lactase, enzyme lactase, has the best electroanalytical performance. The electroanalytical method has some important advantages when it comes to laboratory practice, such as its precision, reliability, simplicity, and low cost. This biosensor has been successfully applied to the determination of the essential and non-essential amino acids in simple and multi-component pharmaceuticals. This is the references, and thank you for your attention. This work is supported by the project Entrepreneur Doc. And have a good day. If you have a question.
Yes, thank you very much for your presentation. There are questions, please? If not, I have a question. How did you deposit polypyrrole on the surface of electrode?
With method chronoamperometry with next parameters at 19 seconds and the potential 0.8 volts.
Then it was an electropolymerization, yes?
Yes, yes, by electropolymerization.
Okay, this is clear. And the other question, you put the enzyme before activating the surface of polypyrrole with glutaraldehyde? I saw that. Or after?
I put the enzyme after electropolymerization.
After electropolymerization, but before the reaction with glutaraldehyde, yes?
Yes, yes.
Yes, you try to make after the glutaraldehyde reaction?
No, I not try.
It would be interesting because we try to do, but with our method, to activate before with glutaraldehyde and after that to make the immobilization, covalent immobilization of enzyme. Okay, it's just a suggestion. Thank you very much. It was a very nice talk, in my opinion, very interesting results. And it was the last presentation because now we have a break till 14 hours. At 14 hours, we have a plenary session with Silvana Andreescu from Potsdam, United States. And our session 1 will continue at 15 o'clock. We have still a free presentation. The first one being also from Galați University, Dunărea de Jos. So, good lunch and see you later. Thank you.
Hello again, dear participants, to the NeXT-Chem workshop, the third edition. After lunch break, now we pass to the second part of our first day of the workshop, and we will start with the presentation of Professor Silvana Andreescu. After this morning, we had the presentation of Professor Arben Merkoçi from Spain. Now we have this, we are honored by the presence of Professor Silvana Andreescu, also because she is graduated from University of Bucharest, so is a Romanian researcher. She started here. After her graduation, she received a PhD in chemistry, specializing in biosensors from University of Perpignan, France, and also from University of Bucharest in 2002. And then between 2003 and 2005, she was a postdoctoral fellow with a NATO grant at the State University of New York. And immediately after this grant, she became a member of the Clarkson faculty since 2005. Now she is the Egon Matievich Endowed Chair in Chemistry. She is department chair and professor of bioanalytical chemistry in the Department of Chemistry and Biomolecular Science at Clarkson University in Potsdam, New York. Also, she is a recipient of French government graduate fellowship, of NATO postdoctoral fellowship, a National Science Foundation Career Award, the John Graham Faculty Research Award, the Research Excellence Award, and recently I have seen that she received the Research Mentoring Award at the Clarkson University, which is a very prestigious award for mentoring the students and postgraduate students in research. Her research interests are in analytical and bioanalytical chemistry, bio-nanotechnology, sustainable nanotechnology, and the development of practical biosensors for food, clinical, and environmental monitoring. She has published more than 150 peer-reviewed publications, 21 book chapters, and she co-edited 3 books. She was awarded 3 patents and has delivered many presentations throughout the world. She has an Hirsch index of 51 and with more than 8,000 citations. She has managed research projects totally over $4 million from various agencies from United States. And also she is very interesting that the Clarkson University is an what we call in Romania or in Europe, in fact, entrepreneurial university. So this university is very focused on application and in collaboration with industry. Now she will present a lecture regarding advancing environmental sustainability through custom design materials and sensors. And we thank you again, Silvana, for your presence here at this early hour for you, because now in New York is 7 o'clock in the morning. So we thank you very much for this these times that you spent with us sharing your experience and your results. Thank you again, Silvana, and please.
Sigur, mulțumesc, Mihaela. Bună dimineața, bună ziua, mulțumesc foarte mult pentru invitație și pentru oportunitatea să prezint la conferința aici, la NeXT-Chem, ceva din cercetările noastre în ultimii 5 ani. O să încerc să share screen.
O să opresc eu. Ok.
Great. Excellent. Okay, so can you see the screen?
Yes.
Great. Well, so what I would like to do this morning is really to go over some of our work on designing materials, maybe not so much sensors, that could be used for environmental monitoring and environmental sustainability. So Clarkson University, we are a private college located in upstate New York, in a very beautiful area in Adirondack Mountains. This is a picture of our campus. I think it's a little bit opposite than what you will see in Bucharest and where NeXT-Chem is located. So very beautiful area. Please come visit us. We'll be more than happy to host you. So I've been here since 2005, and over the years, one of the strengths at Clarkson is the development of materials, nanomaterials, colloids, and this is something that I picked up in 2005 when I moved here. And some of the research I've done is really to develop material innovations that could be used for a variety of applications, including sensing, but also environment. So some of the outline of the talk. First, I will go over a brief overview of what it means, environmental sustainability, why should we care about it, the link between materials and sensors. Then I will go over some of the nanomaterial sorbents that have received significant attention in the last couple of years, and the basics of these porous sorbents, manufacturing, deployment, and potential uses in sustainability. And gave you some examples of the materials developed in our lab. And I will focus primarily on water eutrophication, particularly phosphate removal. We've used these materials for heavy metal and removal, PFOS, PFAS. So if times allow, I will show you some of those results. So maybe the first place to start when we talk about sustainability is really the United Nations Sustainability Goals. This has been published in a report in 2018, where the UN set up 17 priority goals. Among those, so no poverty, zero hunger, that has to do with achieving food security and improved nutrition, as well as sustainable agriculture. Good health and well-being, so household and ambient air pollution is of great interest, so monitoring disease and prevention, clean water and sanitation, so drinking water management, more effective way of producing food. Along these lines, it's also decent work and economic growth, industry, innovation, and infrastructure, so create opportunities for employment, innovation and R&D investment, sustainable cities and communities. So here we talk about waste management, polluted air, building and water, and responsible consumption. So chemical limit or minimize chemical and hazardous waste. Life below water, so conserving the sustainability use of oceans and marine resources. One of the points that the UN makes is the issue of coastal eutrophication due to nutrient pollution, which I will develop later on. Life on land, so protect, restore, and promote sustainable use, peace, justice, and strong institutions. So when we think of materials and sensing solutions and what and how we can use this for promoting environmental sustainability, first is what needs to be addressed and what are the target contaminants. And if we look at water pollution in general, we can see a variety of chemical and biological contaminants, obviously pathogens and viruses, but also pharmaceuticals, heavy metals, pesticides, PCBs, the issue of PFOS, PFAS that received significant attention in the last 2 years, nutrients, phosphate and nitrate. This is a huge problem with the pollution generated, this so-called eutrophication. If we think of the needs for addressing the environmental problems, sensing and materials can help with removal, can help with analysis and fast response time, and provide high throughput in low resource settings and insight. Another area in the last couple of years is to empower citizens, so-called digitized and connected technology made available to all users, so things that can be used anywhere, any place, by anybody. And all of this to improve economic efficiency. If we look at the environmental impact, the ability to identify and manage risk, identify and reduce pollution, lower ecological impact, minimize waste, prevent and also solve the pollution problems, health impact, and as far as technology, field deployment, cost saving, and improved monitoring. There are 3 sustainability pillars, and in the context of sensors and material solutions, one will look at the environment, so natural resource use, prevention, monitoring, and management. And this refers to water, land, air, and waste. This is connected to economic impact, so research and development, cost, economic growth, industry, and business, as well as social factors, so education, equity, community, job creation, and standards of living. So when we design these new technologies, we look for something that is affordable, that is accessible, that is environmental friendly, so that we do not create a secondary pollution problem. We like something that is deployable and scalable and can be picked up by businesses so that it can be immediately implemented into the field. And economic and social, so research, innovation, and entrepreneurship is something that we have been working on for some time to identify ways to translate the technologies that we have developed in the lab into the field. So our motivation for this work is really very simple: clean water. Water is the most important resource, but less than 1% of the water supply is available for self-human consumption. So most of it needs to be treated, which requires increased energy and treating cost. Most sources of drinking water show some evidence of contamination. So methodologies to improve performance, cost, and diminish environmental impacts are still needed. So again, the pollutant water and some of the chemicals that are present in this water that undergo treatment. Current technology is to use bulk materials. Most of it still relies on carbon, on activated carbon. And in the last, I will say 10-15 years, there has been a significant growth in the use of nanotechnology in different forms and shape to improve treatment technology and remediation methods. So we are looking at the exploration of nano and mesoporous materials as alternative solvents to produce clean water. So in terms of nanomaterial solvents and what are they? So activated carbon, polymers and resins, activated alumina, these are some of the materials that are conventionally used. In porous materials, so we can see any type of carbon-based nanoporous carbon. Some of them are derived from natural resources such as woody materials, graphene, carbon nanotubes, mesoporous silica. There are a variety of porous polymeric materials, nanoporous oxides, zeolites, dendrimer, and recently we see a lot of work in metal organic framework, MX-zines, and three-dimensional layer materials. So the interest in using these materials has to do with the enhanced surface area, binding sites, and the huge porosity enabling enhanced removal. So the properties of the solvents that we are looking on for these applications, first, we like a large absorption capacity, a fast absorption rate, cost effectiveness, and non-toxicity to the environment. It is also important for these materials to be able to separate another important issue, particularly related to the use and implementation and deployment of nanotechnology, is really scalability. How can we produce this structure in large quantity? Is it economically feasible? And primarily in cases like carbon nanotubes, or we see a growth and then we see sort of a diminished increased interest over time due to the large manufacturing cost and inability to produce these industry scale applications. So the mechanism of absorption in these cases, it involves a variety of monolayer, multi-layer. There is a boundary layer that one will characterize. We look at properties of adsorbent and the adsorbate. So the surface charge is important. And one of the important issues is how can we tailor selectivity, and can we tailor selectivity of these materials towards particular pollutant? Can we make some of them selective for nutrients, some others for heavy metals, dyes, phenolics, PCBs, PFAS, and so on. In the evaluation of adsorption properties, we use adsorption kinetic as isotherm model, Langmuir model, and Friedrich model are typically the most used. And then there are a couple of characteristics on adsorption capacity, removal percentage, adsorption rate, and selectivity ratio. One other characteristic, if we want, it's reusability of this sorbent. So once we bind something onto it, can we also remove it so that we are reusing the sorbents in the context and in the spirit of sustainability. The sorption mechanism can involve physisorption, ion exchange, microprecipitation, and chemisorption. So the work that we have done in my lab primarily is on metal oxide sorbents. There is a lot of literature on the use of magnetic iron oxides that have the ability to remove some pollutants by absorption onto their surface, and then with the use of magnetic force, remove them from the environment. There is a lot of work in titanium dioxide photocatalytic degradation incorporated in different matrices. The type of sorbents that we used are cerium oxide sorbents. We use them as nanoparticles, so nano-sized particles, porous hierarchical nanostructures when we grow them onto the surface of other materials such as silica or polymeric core shell type. And more recently, we looked at cerium oxide incorporated within metal organic framework. And when we do this work, we try to control the size, the morphology, the surface charge, the porosity, and also the supporting material, as in many cases, we prefer to use nanostructuration onto and deposit these materials onto some sort of supports, either silica or polymeric beads, so that they are easier manipulated and used. So one of the questions is why cerium and why we became interested in cerium. Cerium oxide has dual oxidation states, cerium 3 and 4, which means it facilitates redox reaction. It can act as an oxidizer and a reducing agent. We've used this capability for heavy metal removal and detection. Because it's an oxide, it has very high surface functionality, so we can really tailor the surface and the hydroxides at the surface for creating chemisorption sites for toxicants. Another important issue is that these particles are not toxic, but they can become toxic by accumulating toxicants onto the surface. Actually, they are used as catalytic converters in gasolines so that they are continuously released in the environment. There are some other potential uses in biomedicine as antioxidants. We got exposed to these particles when I came at Clarkson in 2005, when many people were using it in chemical mechanical polishing, which is a process for producing semiconducting wafers for the electronics industry. And over time, we use the redox reactivity to create optical and electrochemical sensors, labels for bioassays and inorganic, and as free radicals scavengers or cosmetic sun blockers. So these were some of the additional properties we have explored. I will only share with you today our research on designing these materials as sorbents for environmental remediation, and the type of contaminants we have removed with these materials are phosphates, arsenic, phenolics, and more recently we looked at PFOA, PFAS. So the goal of the research, as we started a couple of years ago, was to synthesize uniform nanostructures based on these materials, study the interaction with environmental contaminants, investigate the adsorption mechanism and the driving parameters as far as kinetic isotherms and surface charge, and then use this fundamental knowledge to design remediation and sensing devices. Most of the work that we've done in the last 3 years was focused on removal of phosphate for preventing water eutrophication, and this is what I will be focusing in today. These are some of the cerium oxide that we have synthesized in the lab. We now have a variety of over 20 types of cerium that are in colloidal form, so we can tailor the size from 5 nanometers to 10, 20 nanometers. We also create them in micro size, 250 nanometers. We have the ability to change the surface layer. Some are stabilized with citric acid or ascorbic acid or oleamine. We have synthesized them onto the surface of silicon dioxide, so basically silica beads, creating this nanostructure, very porous surface onto the silica surface. We have them in powder. We have also produced them in colloidal forms. And more recently, we have synthesized them and put cerium inside a metal organic framework, which gave us this huge surface area and accessibility to the MOFs pores. We've done cellulonization. We put them on paper on different solid surfaces. So we do have the ability to tailor reactivity, tailor coverage, as well as surface functionality and functional groups. This is some of the work we have done to create nanocrystals or cerium, very well-defined sizes of about 6 to 7 nanometers in diameter. Another thing that we've done is to dope them with europium or gadolinium. This is because europium gives us fluorescence properties, so imaging properties, which we used for sensing platforms as well as for imaging of changing in surface functionality as a result of absorption. What is and how we can use these materials in preventing one of the largest and most important issue in water pollution or clean water, so water eutrophication. Nutrient pollution is one of the most widespread, costly and challenging environmental problem. This is caused primarily by excess nitrogen and phosphorus in the air and water. This phosphorus and nitrogen, they are used as detergents, they are used as fertilizers, and also in many industries. So this is the EPA, Environmental Protection Agency in the United States, recognized this as one of America's most widespread environmental problems. When we think of phosphorus, phosphorus is all around us. It's part of the RNA, it's in ATP, phospholipids, and the fertilizer contains a large amount of phosphates in different states. Concentrations in water range from 2 to 23 ppm in wastewater. Higher concentrations that reaches 100 ppb levels, it creates increase in algal bloom production. So what is water eutrophication? So essentially, as phosphates are released into the environment through detergents or through agricultural practices from fertilizers, this promotes extensive algal growth. So you may have seen if in certain lakes or coastal areas, the formation and these green areas of plants that float onto the surface of the water. So these plants are phytoplankton that prevent oxygen to pass through. So as a result, there is a significant mortality rate of the fish population and also organic matter that is decomposed and due to the lack of oxygen. So nutrients can cause this increase in phytoplankton. In the US, we see many areas that are that show harmful algal blooms, and you see significant amounts on some beaches, altering tourism, where these grain deposits occur. So it's not only that it's the issue for fish, but it's an overall touristic and many other industries that are affected by. These are some of the issue of eutrophication, so reduce the ability of fish and other aquatic life to find food and can cause the entire population. So there are areas when the entire fish population dies as a result of eutrophication. This is not only a US problem, this is a map of eutrophic and hypoxic areas around the world. We can see that primarily the coastal areas are affected by these eutrophic zones that show the depletion in dissolved oxygen levels, with 400 dead zones identified worldwide. I did the search yesterday, try to find the extent of water pollution and eutrophication in Romania. This is a report I found that was published in 2019 in Water Resources Management, and showing the extent, the red lines here are nutrient and pesticides emissions. So zones that are affected by eutrophication in the country, primarily in this. So this is the rivers area, and the second graph here is the lakes and coastal water. So the Black Sea is affected by nutrient pollution. So the red zone here is nutrient pollution. Some of the other, the purple is organic pollution, and the green is chemical pollution. So it seems to be a really an important problem in Romania as well. A report from the NASA Earth Observatory on the Black Sea. This is an image that I pulled from the NASA website showing the algae bloom in Black Sea. One of the issues they raise with Black Sea is that because it is isolated from the world's ocean and because it is extensive drainage of network of rivers, the Black Sea has a very delicate water balance. So particular concerns are salinity, water level, and nutrient levels. And they raise the issue of the increased nutrient loads from human resources together with pollution. So the problem as far as eutrophication and what people will call as a phosphorus paradox, phosphate is essential for food and agriculture. Without those fertilizers, the food production will diminish. So it's a valuable resource that it's needed for food production. However, it is a non-renewable resource, so it is extracted from certain mountains and in Asia, and it is expected that these supplies will actually diminish over the next 100 years. In 2008, there was a significant increase in the cost of fertilizer based on phosphate because of the limited supply from those extraction sites. However, while essential, this extensive accumulation leads to this water eutrophication and poor water quality. So this is something that is absolutely necessary, but if used in extensive amounts, it causes significant pollution. So it's the good and the bad. The need is to ensure sustainability of phosphate and food. So ideally, you would like to remove those phosphates, but also recover it for further use as a fertilizer, which speaks very highly of the concept of sustainability on reuse, recover, and implement it again into the chain of technology. So the technology gap right now is to selectively remove and recover this phosphate and also manage these phosphates. So none of the areas that I've showed you as far as eutrophication are monitored properly. And a lot has to do with the lack of methods. The conventional method is still a molybdenum blue method that is used since the 60s. And there is there are no technologies that enable low-cost monitoring of excess nutrients in eutrophic water. So these are some of the research goals and research needs that we've tried to address. The nano-serious sorbents that we have developed, we started from the idea that using simple colloidal particles of 10 to 20 nanometers, it's actually not feasible because of the small size. We can't just dump a bunch of particles in the water and then leave them there into the field. So what we try to do, and we started, is to grow these nanostructured cerium oxides onto the surface of porous sorbent, of much bigger materials that could be packed into columns or deposited on some sort of solid surfaces. And this is one of the first that we have developed in 2018 when we took silicon dioxide beads and we grafted the cerium oxide nanoparticles onto the surface, and we see an increase in the porosity. We also see nanometer size, so small islands of cerium oxide onto the silicon. The synthesis is pretty straightforward. We started with the silica beads, and we then did the reduction reaction from cerium nitrate onto the surface of the beads, creating these nanoparticles onto the surface. It takes about 2-3 hours, 7 hours. So we stir it for 6 hours, and then we only separate it from the reaction mixture. The size of these beads, it's around 570 nanometers. The silica beads by themselves are around 400 nanometers. So we see an increase due to the grafting of this cerium oxide onto their surface. The zeta potential, so all of these particles are negatively charged. We then studied the phosphate adsorption, and the reason we use this cerium is the formation of cerium phosphate onto the surface. So the molybdenum blue method, this is the method that was developed by Stanley in 1967. And even to these days, this is the gold standard that is used to determine phosphate. It is not it's a simple, apparently simple method. However, it is very, very sensitive to incubation time. If you leave it longer with 5 or 10 seconds, then you see a decrease in precision. So it's not an easy method to use by non-experts. So essentially what we have done, we have done a batch study. We've also done some column studies in which we expose these particles to phosphate. We mix them, we then filtrate it, and then we analyze the supernatant for the removal, for the decrease in activity. So if there is no change in phosphate adsorption and there is no adsorption onto the cerium, then we see the same blue color because the phosphate is still there. If we remove phosphate, then the supernatant will show no color or very reduced blue color due to the removal and grafting of this phosphate onto the surface of the beads. And these are some of the results quantifying phosphate adsorption after the removal for different concentration of phosphate after the removal by the cerium silicon beads. The environmental variable, the contact time, the adsorption process it's relatively fast. It's about 16 minutes. We reach 100% adsorption. One interesting thing is, which we did not expect, is that the process is relatively unaffected by pH changes. So here, when we looked at the effect of pH, the sorbent, the sorption capacity is very similar for pH ranges ranging from 5 to 10. So this formation of cerium phosphate onto the surface of the beads is not pH dependent. The kinetic isotherm models, we used both the Langmuir and the Friedrich model, and it shows that the Langmuir model, meaning the monolayer absorption, more favorable describe the phosphate adsorption onto these materials. As far as mechanism of the adsorption process, we look at the affinity of an ion exchanger. We believe the process involves bidentate and monodentate inner sphere complexation, forming this cerium phosphate onto the surface. Which is interesting is that only phosphate, it shows us this removal ability. We exposed it to other possible coexisting ions, including nitrates, chloride, bromide, and none of them show significant adsorption. So it's only phosphate. So this sorbent shows sense selectivity towards phosphate. This sorbent shows selectivity towards phosphate. Another thing that we are very excited about was the ability to recover the phosphate bound onto these beads. And the way we achieved that, we looked at ligand exchange. So we wanted to detach the phosphate, so destabilize the cerium phosphate onto the surface. And we did that by washing with hydrogen peroxide. The cerium peroxide bonds are stronger than the phosphate, and this allowed us to remove the phosphate from the beads and reuse the sorbents for a couple of cycles. These are examples of reusability for 6 consecutive cycles. So not only that the sorbent is selective, but it also gave us an opportunity to recover the adsorbed phosphate and also to reuse this sorbent. So in terms of competitive advantages over activated carbon, we believe the sorbent provides high surface area. It definitely has a very high removal rate, effective at a wide range of pHs. And we achieve 99% effectiveness. The removal rate is 30 minutes to 1 hour. These particles are stable, selective, and they can be regenerated. As compared to activated carbon that is 90% effective, it cannot be regenerated, so most of it is carbonized after it is used. However, we are working with engineers and the practitioners in agricultural fields on how we can implement and translate the technology into the field. And one of the questions that comes often when you work on these technologies, can you mass produce it, and how expensive it is? And this is what we work right now. We produce about 100 to 200 grams of these sorbents easily. However, can we produce them in kilograms amounts? That's something that we need to figure it out for future research. We've used it in column-based separation. These are some of the columns that we use in the lab, and we try to implement this technology as a capture, remove, reuse kind of thing. So when we talk to agricultural experts who are really dealing with this eutrophication problem, the first question is, can we do these columns in 4 kilograms? So as the amount of sorbents that will need to go into an actual column that used for phosphate removal. In the same time, we started to work on next generation sorbent to further increase the surface area, but by using metal organic frameworks. So incorporate these cerium particles within MOF matrices that creates an even higher surface area with accessible cages for maximizing phosphate uptake. So my student produced this cerium nanoparticles inside a MOF structure where the open sites, these open channels, enable passage of phosphate, which is a very small molecule, through it, but we take advantage of this very large surface area that is produced through incorporation of the nanoparticles within the MOF structure. We've also done computational calculations with an experiment with a theoretical physicist to study the binding energy and try to understand why and what is so unique about this cerium phosphate onto the surface of these nanoparticulate
matter. So we've reported this work in 2020 in ACS, Applied Materials and Interfaces. This is some of the characterization of the MOF. So this is the structure of the metal organic framework, and the nanoparticles that are created in situ are about 2 to 3 nanometers. So we have very, very small particles that have a huge reactivity and huge surface area for binding. When we looked at the phosphate uptake results, the absorption is significantly faster than on our cerium silica beads. So we achieve here almost 100% absorption within 4 minutes. So very, very fast. These are samples of phosphate, how the phosphate absorption looks like, and this is the molybdenum phosphate blue color after exposure to MOF, indicating successful removal of phosphate. So there is essentially no phosphate in the supernatant. All of it is bound onto the surface of the cerium MOF. We've noticed the same effect, so it's consistent with our previous work as far as interfering ions, so the same selectivity towards phosphate. It also works at different temperature, 20, 40, 60. We see essentially no change in the removal capacity and also pH independence. One thing that we have tried to reuse these materials as well. Our initial trials have failed because simply when we try to regenerate and wash it with a couple of, with hydrogen peroxide as previously or with strong acid, what happened immediately was essentially that the MOF structure collapsed. So it's not the cerium, but it's the MOF structure collapse. And once the structure collapses, then there is agglomeration of these nanoparticles. So we are still working to see and find ways of reusing the MOFs. We are also working on different ways of grafting these materials onto different surfaces, such as maybe polymer materials or fishing nets, for example. Absorption isotherm, similar as with our cerium MOFs, we see an preferential Langmuir type, so indicating monolayer absorption. We compare the performance with other porous materials. This is what we reported for our cerium MOF. The absorption capacity is 179 milligram per gram sorbent. Our cerium silica shows 91. So we do see significant, so double the absorption capacity as compared to the silica beads. Other sorbents reported in literature, including other MOFs or mesoporous silica, this SBA, they show lower sorption capacity than these materials. So we think there are significant potential of using these cerium-based sorbents for phosphate removal. We then started to use the same chemistry and design sensors, so use essentially the same binding efficiency, but create sensors for measurement of phosphate into the field, with the idea to create a low-cost, either a paper-based or an electrochemical type sensor that could be used in the field. These are some of our initial results where we investigated the cerium chemistry and the cerium phosphate formation as a result of this binding. First results show linearity ranging in the ppb range. We used primarily cerium salts initially to study the reaction, and hopefully we'll be able to move towards some of these more nanostructure materials that provided a very high and very selective binding to phosphate. Another pollutant that we are very interested in, there is a and generating significant interest is the PFOS, PFAS, so these flame retardants that are used in clothing, that are used essentially everywhere. It was found in the blood and urine, so it became bioaccumulated in the body. And we started to look into the potential of these materials to absorb this PFOS, PFAS, which are essentially fluoride, very long fluoride chains with sulfonated or carboxylated ends. These are the very first initial results that we got. Indeed, it shows that the cerium sorbent do provide some binding to these cerium materials. And this binding, most of them binds through either the fluoride or sulfonate. We are still investigating the mechanism of this reaction, and it preferentially towards long chain PFAS. And using a similar chemistry, we now work on electrochemical detection of these compounds. The toxicity of PFAS and the EPA limits that have been set up this year are around 1 nanomolar. And the conventional method is HPLC MSMS. We have a lab here at Clarkson that is certified to do PFOS, PFAS analysis. Every single sample costs one sample $100 to $150. So it's quite expensive. So we have designed this electrochemical sensors, screen printing electrode that have redox materials that may be sensitive to PFOS, PFAS. So this is an example of the calibration curve, and one of the responses to a long chain PFAS showing detection in the low picomolar level. So we are very excited about this work and where this will lead us. So I will skip over some of the heavy metal work and go over the conclusions, so that I allow a couple of questions, a couple of minutes for discussion. So in conclusion, I hope I have convinced you that nanoporous sorbents hold promise for applications in environmental removal and sensing, and particularly this class of metal oxides that have been I will say significantly overlooked in the scientific literature. We see a lot on titanium photodegradation, and we see a lot on carbon-based materials. But we think that cerium is an interesting material with a very unique set of physicochemical properties. We are excited about the ability to regenerate and recover pollutants, particularly phosphate, and create this class of regenerable sorbents. So moving away from just absorbing materials, which creates this huge waste, but rather to a system where we can capture, remove, and recover. There are opportunities to improve performance and deployment. We are not yet there to deploy these devices, but scalability, manufacturability are key for future development of these technologies. I will say that the key and the research that led us here, we've worked with cerium materials for over 15 years now, is to create this tailored surface chemistry and understanding of the interfacial processes. So uniform, stable, dispersed materials that can be that can achieve predictable and also reproducible results. So the materials that we've created are incredibly reproducible. I have undergraduate students working with them. The absorption studies, all the results that we have we have generated in the initial studies have been very robust. And we feel that future research into integration into engineering system and hopefully manufacturing at a large scale are addressable in the future. So with that, I would like to add by thanking my group. The work that I presented today was done by about 3 PhD students in my group in the last 3-4 years. It's them who are doing the work. I am just in my office writing emails and proposals, and so I'm very grateful for everything that they have done for this project. It's a pleasure to have such a large group of enthusiastic students. These are some of the pictures from 2 summers ago where we didn't have to wear masks and we were not quarantined. And I really end with an acknowledgement of collaboration, communication, and diversity. So these are all the representative countries from my students in my groups, going from Ukraine, Egypt, Nepal, Nigeria, Bangladesh, Tunisia. We really like working together and creating beautiful science. So with that, I will end here and will be happy to answer questions and any discussion that you would like to have on these materials and others. So thank you.
Thank you very much, Silvana. Mulțumim foarte mult pentru introducerea în această lume a metodelor de îndepărtare a unor nutrienți care pot să devină poluanți. Pe de o parte sunt nutrienți, pe de altă parte sunt poluanți. Și în cadrul NeXT-Chem și în cadrul celelalte institute și universități din România există preocupări în acest sens, dar cred că direcția pe care ne-ai arătat-o tu poate să fie de asemenea foarte interesantă și atractivă, eventual pentru colaborări în acest sens, mai ales că ai acceptat să faci o prezentare de asemenea o plenară și la simpozionul nostru PRIO-Chem, care va avea loc la sfârșitul lunii octombrie. Deci până atunci poate colegii noștri, participanții de astăzi, se vor gândi cum să abordeze o eventuală colaborare cu grupul vostru, care, suntem invidioși de așa o participare internațională. După cum știi, România e un pic mai complicată, dar facem pași în direcția aceasta spre internaționalizarea domeniului de cercetare și în România. Îți mulțumim foarte mult și acum colegii mei, dacă vor să adreseze întrebări, rog să o facă. Mulțumesc mult!
Just să adaug, numai să adaug la ce ai spus, Mihaela, aș fi interesată în orice colaborări. De multe ori primim request sau cereri să dăm aceste materiale la alte laboratoare, putem să sintetizăm și să trimitem în diferite laboratoare. Am făcut asta pentru mulți ani. Altceva este, în afară de fosfată, avem o mulțime de proiecte on sensing technologies. So sensors for food, sensors for environmental pollutants. I just did not show all of that work today, but I am more than interested to explore collaborative research, and hopefully we can travel soon and I can come and visit NeXT-Chem. I haven't been to Romania in about 3 years. We have a ticket for like 2 years that we keep postponing it because of travel restrictions and so on. But I will be more than happy to explore collaborations, and I think my email should be fairly easy to find. So please do not hesitate to reach out to us, and happy to discuss.
Mulțumim, sigur e disponibil la adresa ta. Ca un comentariu vreau să spun că mi-e interesant că și în Statele Unite există această abordare sau aveți aceeași problemă ca și noi în relația cu mediul economic, când suntem întrebați, e bun, este foarte bine, foarte interesant, câte kilograme puteți să ne dați, cât costă, ce echipament avem nevoie, ca mâine să facem și cât costă să punem pe piață produsul. Văd că și la voi este aceeași situație.
Yeah, I think it only became clear to me in the last few years, and we have a very strong engineering department, environmental engineering in particular here. And as soon as I started to discuss with them, said this is all great and fundamental research is great, and I think you need to have very strong fundamental research for any type of technology. But can it be translated? Can we put this in the, we can't even test it in the field because the first question they ask is, can you do this in 4 kilograms to pack our existing filter and replace whatever they are using right now? It's very difficult also for us and for me to hire a PhD student just to go from synthesizing 100 grams to 4 kilograms, because I think it will be a very tedious work and not necessarily rewarded because we can publish any of that. I don't see how we can publish a work going from so it needs to be industry related, industry driven, and there needs to be funding for it. And I think there is a gap between fundamental research and applied research. And unfortunately, at least in the United States, we have agencies that are funding fundamental research, like the National Science Foundation, and we have agencies who fund deployment, but not necessarily the in-between. So that's something that I'm sure Romania is facing the same sort of issues. There are small businesses who are trying to bridge the gap and investigate ways on selling maybe small quantities of these materials or investing on as their business model to create a product that can be potentially delivered to multiple for this sort of applications. But the eutrophication is something we got into it because there is a need. We see it in the news, especially in New York State and the water, the Great Lakes. We see it like almost every month there is something about the tourism, the fishing industry. It's really a huge problem, and I think it's going to become even worse. The only ones that has emerged in the last 2 years is the PFOS, PFAS. I don't know whether in Romania this is something that agencies started to look into it, but all the funding essentially has been shifted to this issue of PFOS. There is the eutrophication is a little bit lower, but this PFOS, it's becoming huge. If you look in the literature, that's almost every month Chemical Engineering News has an article about it.
Thank you very much again, Silvana. Let's see if in the autumn we'll be able to make face-to-face symposium. Anyway, see you in the autumn, and good luck with your proposals and with your work and your research group also. Congratulations for your work. And
thank you so much. It was my pleasure to present here today. I'm looking forward to the event in October, and I'm looking forward to meeting you all in person. In person as soon as we can travel again. Thank you, and enjoy the symposium and the workshop that you put together. Have a great day.
Thank you. Goodbye.
Bye.
Mă iertați, dar aveți microfonul oprit.
Sorry. So we pass now to the second part of the presentation of our participants, and I invite now Professor Rodica Ion to chair this part, and I wish good luck to all the presenters this afternoon. Please, Professor Rodica Ion.
A vorbit cineva cu Rodica?
Păi am înțeles că domnul Fierescu a luat legătura cu dumneaei și așa s-a fost stabilit.
Doar la secțiunea de patrimoniu, deci ar trebui să continue domnul Sârbă.
Deci continui eu atunci.
Da, la secțiunea, e în continuare secțiunea de materiale, deci continuați dumneavoastră.
Da. Bine. Then we continue with the second part of session 1, multifunctional materials in nanocomposites. We have a presentation by Alexandra Virginia Băunegru-Mereșescu with the title Development of Biosensors for the Hydroxycyanamic Acid Analysis. And the team is Alexandra Virginia Mereșescu, Constantina Petrei, Irina Georgiana Bulgaru, Munteanu, Ramona Oana Gunache Roșca. The team is from University Dunărea de Jos from Galați. Please give the presentation.
Good afternoon. You can see my presentation?
Yes, it's okay.
Thank you. I am pleased to present an important of our work, namely development of biosensor for the hydroxycyanamic acid analysis. In the following slides, I will present some information about hydroxycyanamic acid, electrochemical method like cyclic voltammetry, tyrosinase-based biosensor, and my experimental study about development of novel electrochemical biosensor based on carbon nanofiber, gold nanoparticle, and tyrosinase for the detection of ferric acid in cosmetics. Hydroxycyanamic acid are known as natural antioxidant. Ferulic acid is one of the hydroxycyanamic acid found in a lot of fruit and juice and some medicinal plants. Ferulic acid is...
Nu se văd slide-urile. Nu se văd slide-urile. Dați-i show. Slide show, ca să apară slide-urile. Așa, da.
It's okay.
It's okay.
Okay. Ferulic acid is often used in anti-aging cosmetic formulation because it is able to stimulate intracellular antioxidant defense system. Also, ferulic acid, according to concentration, may be efficient in dermatological conditions such as atopic dermatitis. Therefore, determining the concentration of ferulic acid is important in food and cosmetic industry. In the past few years, electrochemical analysis has proved promising in point of application prospect in various fields, like quality control in food, pharmaceuticals, or cosmetic industry, due to high sensitivity, low detection, and relatively low cost. One of the most used voltammetering technique is cyclic voltammetry. By this technique, information can be obtained regarding the mechanism of the electrochemical reaction, the identification of the species present in solution, and the determination of the diffusion coefficient of the electroactive species. The figure shows the three generations of amperometric enzymatic biosensors. They are classified according to the method of electron transfer used to quantify the biological reaction. In this picture are represented several immobilization techniques, such as physical adsorption, cross-linking, or covalent bonding. All these methods have multiple advantages, but also limitation. The immobilization process should be reproducible, simple, with a low cost, and this shortens processing time. Tyrosinase is a metalloenzyme able to react with phenols and is useful for various applications. This enzyme is suitable for evaluating the electrochemical property of materials, which is why it is often used to modify sensors. Also, carbon nanomaterials and metallic nanoparticles are able to improve the performance of tyrosinase-based biosensor. In the experimental study, we developed a novel electrochemical biosensor based on carbon nanofibers, gold nanoparticles, and tyrosinase for the detection of ferric acid in cosmetics. More precisely, we studied the electrochemical behavior of this novel biosensor, and we quantify ferric acid from free cosmetic products. The results obtained were compared with spectrometric method in the infrared range. For the experiment, we used several substances, including ferric acid, potassium chloride, potassium ferrocyanide, and tyrosinase, and the necessary equipment was a potentiostat galvanostat, electrochemical cell with the free electrode, spectrophotometer Bruker alpha-FTIR, and other laboratory tools. The free analyzed products were chosen based on their composition. The first product was the ordinary serum with resveratrol and ferulic acid 3%. The second was sebiotonin cream for eyes and lips with ferulic acid and aloe vera. And the third product was the makeup removing emulsion Detoxas Lavital active. In the electrochemical analysis, different amounts of each cosmetic product were used, and they were dispersed in a 15 milliliter phosphate buffer solution. To prepare the biosensor, the tyrosinase was immobilized on the surface of the screen-printed electron-based carbon nanofiber modified with gold nanoparticle by the drop and dry technique, followed by reticulation with glutaraldehyde. In order to perform this procedure, a tyrosinase enzyme solution was prepared in PBS solution with a concentration of 50 microgram to microliter. The biosensor was kept at the fridge until it was used. In preliminary analysis, we compare the electrochemical behavior of sensor and biosensor in the phosphate buffer solution. When the sensor was immersed in PBS, cathodic peak was observed. This peak occurred due to the modification of the screen-printed electrode with gold nanoparticles and tyrosinase, respectively, in the case of the biosensor. The next text includes the immersion of each electrode in a solution of potassium ferrocyanide. In the cyclic voltammograms obtained, we may observe an anodic and cathodic peak release of the oxidoreduction process of the ferrocyanide. In this table are shown the characteristics of the peak pair observed in the previous analysis. The highest peak are seen for sensor, which shows higher sensitivity in detecting potassium ferrocyanide and the synergic effect of gold nanoparticle in detection. The presence of tyrosinase on the biosensor surface is clearly evinced by the difference seen between the electrochemical behavior for sensor and biosensor. The next step was the reporting of the cyclic voltammogram at the various scan rate using the same solution. The figure shows that the intensity of the peak corresponding to the process of oxidoreduction of the ferrocyanide increases with the scan rate. You can be seen that there is a linear dependence between the current of the anodic peak and the scan rate, which proves that the electrochemical process is controlled by the diffusion of the electroactive species. As we can see in the table, the biosensor has the lowest value of the active surface because the tyrosinase immobilized on the electron surface does not take part in the process of oxidoreduction of the ferrocyanide, proving selectivity. In the next step was evaluated the voltammetric responses of electrode in ferrolytic casing solution. Both cases evince 3 anodic and 2 cathodic peaks, or different intensity and potential related to oxidoreduction process of the ferric acid at the surface of the sensitive element. In the case of biosensor, the potential of the first cathodic peak is lower. This displacement towards negative value of the potential, showing that the reduction process is strongly influenced by the presence of the enzyme. The detection at a lower potential shows that the reduction process needs a lower activation energy in the biosensor case, it being a property of the enzyme. So the biosensor has superior sensitivity and selectivity compared to the sensor in detecting ferrolytic acid. The next stage is the kinetic of redox process for the two electrodes. The figure shows the voltammograms obtained for a biosensor and the linear dependence between the currents of the cathodic peak and the scan rate. That confirmed that the redox process of ferrolytic acid is controlled by absorption. As a result, the reduction process is governed by the Lagrange equation. The value of the surface concentration of the electroactive species were calculated from the slope of the linear equation. Comparing the result obtained, we can say that the process being faster in case of the biosensor. The biosensor was used to detect ferric acid in various concentrations. It may be observed that the intensity of the first reduction peak increased with the concentration of the analyte within the concentration range study from 0.1 to 129.6 micromolar. The current response is linear in the range 0.1 and 1.6 micromolar. Limit of detection and quantification were calculated and the values are low, which shows high sensitivity of biosensor. To check the feasibility of the method proposed, the biosensor was used to detect ferric acid in cosmetic products. The voltammograms performed for all these cosmetic products show the peak due to ferric acid and the current increases with the concentration. The figure on the right side shows the voltammograms of biosensor immersed in solution of ordinary antioxidant serum of various concentrations. The representative peak may be seen for electrochemical process of the ferric acid, and the first reduction peak was used for quantification. The ferric acid concentrations were calculated for the cosmetic product under investigation, and the result is included in this table. To validate the voltammetric method, the cosmetic products were also analyzed by the infrared range spectrometric method. It can be observed that the analyte concentration in the cosmetic product obtained by the two methods are close in value, which proves that the method using the biosensor is useful for ferric acid quantification. In the case of ordinary product, for which the manufacturer indicates 3% ferric acid concentration, it can be seen that the results are close to the indicated ones, thus proving the accuracy of the two methods of ferric acid detection. In conclusion, the biosensor proved to be useful in the analysis of ferric acid in cosmetic products. The method proposed showed good accuracy, simplicity, and low cost. Besides this, the method is accurate and versatile, so it can be used in routine analysis in the quality control of cosmetic products, pharmaceuticals, and other type of tablets. Thank you for your attention.
Okay, thank you for your nice presentation. I think that it was interesting. Now the discussion is open. You may ask or make comments. I have a question. I didn't understood how do you bind the nanofibers and gold particles to the carbon surface of the electrode?
Yes, the first sensor was commercial. My work was to immobilize the tyrosinates.
But there are sensors which already are prepared with carbon nanofibers and gold particles on the electrode, on the working electrode. Yes.
Yes.
There are such you may buy from commerce. Yes.
Yes.
It's possible. I didn't know. And which is the provider? Is from Romania or from abroad?
Sorry, I don't hear.
Which is the provider of such sensors? It's a Romanian provider or from abroad, from
another
country.
Another country. Dropsense.
Dropsense. Yes, I know. It's Spanish. Okay.
Yes.
Okay. Then it's much easier like this because you applied a cross-linking method.
Yes.
In fact, which is the method because the enzyme tyrosinase enter into carbon nanotube, and there it's impossible to lose because it's cross-linked. This is a method. Okay, thank you.
Thank you.
So we now are going to the next presentation, which will be performed by Rebecca Leu-Alexa. The title of the research is 3D printable ink based on alginate and layered silicates. And the team is Rebecca Leu-Alexa, Horia Iovu, George Vlașceanu, Andrada Serafim, Ervila Alexandrescu, Silviu Preda, and Raluca Ianchiș. The team is from University Polytechnic in Bucharest and ICHECHEM. Please go on.
Hello, my name is Rebecca Alexa, and today I will speak about 3D printable ink based on alginate and layered silicates. The objective of this research was to obtain nanocomposite materials suitable in 3D printing for biomedical application. These composite materials are made of an alginate matrix and several anorganic fillers, namely Clasid sodium, Clasid 30B, Clasid 93, Clasid 20A, Clasid 15A. These types of Clasid were added in order to improve the rheological, mechanical, and biological properties of alginate and to make a comparative study in order to select the best one for the future in-depth biological studies. Spectrometry confirmed the inclusion of clay in the alginate polymer matrix. Specific peaks of clay were found in the nanocomposite samples at 1041 and 1048 and correspond to CO stretching vibration. The next peaks are 460, 566 and are attributed to aluminium OC vibration. Clear spectra of the nanocomposite samples containing organomodified clays reveal the specific peaks attributed to the symmetric and asymmetric stretching vibration of methylene groups at 2857. These groups are from the organomodifier agent, namely hydrocarbon chains of the quaternary ammonium salt. 3D printable ink based on alginate and layered silicates. The objective of this research was to obtain nanocomposite materials suitable in 3D printing for biomedical application. The research was carried out using 3D printing methods. The techniques From the X-ray spectra, we can observe that the silicates were very well dispersed in the polymer matrix by decreasing the intensity of the peaks attributed to the interbase distances. Rheological investigation revealed that the addition of any clay modifies the behavior of the neat polymeric matrix. The most obvious influence on the internal structure of the composition and therefore on the rheologic behavior is observed when closid sodium and closid 93 are added. In these cases, the solid-like behavior of the composition is maintained on the whole studied frequency range with a storage modulus higher than loss modulus. The addition of closid 30b, closid 28 and closid 15 bring by a slight modification on the rheological properties of the synthesized inks, lowering the crossover point to 4 Hz in the case of closid 15, 2.5 Hz in the case of closid 30b, and 0.8 Hz in the case of closid 28. Considering that the crossover point is the inverse of the relaxation time, this modification indicates that the composition made of alginate and closid 30b, alginate and closid 28 and alginate closid 15 needs longer relaxation times when compared with the alginate control sample, while the composition made of alginate closid sodium and alginate closid 93 maintain their solid-like behavior throughout the whole process. The steady shear viscosity measurements show that the addition of any clay leads significant modification in the viscosity of the composition. While the alginate control sample exhibit a Newtonian flow, followed by a slight shear tightening behavior, the addition of clay leads to a predominant non-Newtonian behavior with a pronounced shear tightening effect. The most noteworthy effect of the composition behavior is exhibited by the addition of closid sodium and closid 93. Mechanical analyses were performed on the nanoindenter on crosslink sample. Generally, studies demonstrated that layer silicates improve the mechanical properties of the final nanomaterials, which were strongly dependent on clay concentration and platelet distribution within the polymeric matrix. In our case, the elastic modulus show higher values for the nanocomposite samples containing closid sodium and modified nanoclay when compared to pristine alginate. These facts indicate an increased mechanical stability and a stronger gel structure with nanoclay platelets acting as an elastic solid under stress condition. Additionally, all these samples behaved as crosslink hydrogels with a storage modulus higher than loss modulus and had a predominantly elastic rather than viscous character, suggesting complete crosslinking. From the swelling and disintegration studies, we can observe that with the introduction of the nanoclays in the alginate matrix, the hydrophilicity of the alginate was changed, function of the nanoclay introduced. All nanocomposite samples presented a variation in hydrophilicity when compared to the biopolymeric matrix. With the introduction of the nanoclays in alginate matrix, in most cases, the degree of swelling decreases. This effect was attributed firstly to the chemical composition and secondly to the physical barrier effect induced by the cluster formed by clay layers, leading to the limitation of hydrogel swelling. Another reason for the decrease in swelling can be explained most likely to the presence of clay organomodifiers, which reduce the sensitivity to water of the nanocomposite scaffold. From the dissolvability studies, which were made 3 days, 7 days, and 21 days, we can observe that with the introduction of clay, dissolvability decreases. This behavior can be explained by the ability of clay to form strong hydrogen bonds with the alginate. Because the main purpose of this research study was to develop a nanocomposite printing ink that can be used in tissue engineering, 6 things were investigated to generate biocompatible scaffolds which would allow cells to attach, migrate, and proliferate. These things were based on alginate hydrogel, which presents properties like biocompatibility and biodegradability. However, alginate ink has a disadvantage of low viscosity, which further causes the collapse of the filaments and the instability of the 3D printed constructs. In order to improve alginate properties like viscosity, porosity, cell adhesion, and mechanical properties, 5 types of nanoclays were added to the alginate matrix. Thus, new nanocomposites hydrogel clay printable inks were obtained. Printability of the nanocomposites hydrogels was tested using the direct dispensing printhead of the 3D Discovery bioprinter from Regenhue. The direct dispensing printhead uses extrusion technology. Because the extrusion process takes place at room temperature, the technology offered the ability to test different materials in a cell-friendly environment. For the extrusion process, a 3 ml syringe was used with metallic and variable needle dimensions. Additionally, parameters like pressures and printing speeds were explored in order to establish the most suitable one for each material. BioCAD software was used to design 3D structures layer by layer. BioCAD is software that allows drawing object in 2D, and after the G-code is generated, the object can be viewed in 3D. The low viscosity of alginate and alginate modified with classes 30B, 20A, and 15A induce instability during the printing process, so final scaffolds show irregular filaments. In addition, because the material were not very stable, the nanocomposite filaments collapsed and the final scaffold did not maintain their initial dimensions. These things were printed only up to 5 layers. Instead, the samples based on alginate classes sodium and alginate classes 93A presented regular filaments and maintained the initial structural integrity. Moreover, these nanocomposite inks were printed up to 10 layers and, in the case of classes sodium sample, up to 20 layers. Of the material is one of the main parameters studied in hydrogel-based scaffold with important consequences on cell viability. The pores facilitate cells encapsulation, proliferation, and vascularization, and boost nutrient transportation. Another important parameter for biopolymers used in tissue engineering is the roundness of the surface because it affects the cell behavior, allows the cells to attach, spread, and proliferate in a gel mass scaffold. As you can see from the same images, all samples except those obtained from pristine alginate present roundness. The analysis of the roundness of a pore could offer a possible quantification of the printed shapes. Using same images and the Weidel equation, the average roundness of the open pore of the 3D printed scaffolds was calculated. For a perfectly round object, a value of R equal 1 is obtained, while irregular objects tend to have values less than 1. The highest calculated value of 0.7 was obtained for the alginate and the lowest 0.2 for alginate classid sodium and alginate classid 93. These noteworthy differences indicate that 3D printed constructs maintain their shape fidelity better in the presence of classid sodium and classid 93. This dried object obtained by 3D printing from the composite formulation alginate classid sodium to alginate classid 93, alginate classid 30B, 20A, and alginate classid 50A, and the control composition alginate exhibit distinct morphometrical behavior post-deposition from the extrusion needle. The fidelity of the resulted print compared to the square lattice CAD model varies based on the anorganic phase nature. The polymer matrix is reinforced with. Generally, the addition of clay seems to improve the shape fidelity of the print. This can be observed both the preservation of the 90 angles of the printing unit design and the distinction between adjacent layers in cross-sectional view. Empirical alginate classit sodium and alginate classit 93 composition seems to provide the best rheological support meant to preserve the shape of the desired print that could be attributed to the favorable interaction between the measure phase with the specific clays that tune the viscosity. Moreover, the object fabricated from alginate classit sodium composition provides the best stability as per the clear delineation of each deposition layer and the smoother surface compared to the rest of the batch. Also, in the case of alginate classit 20A and alginate classit 50A, the square pattern is also sustained, despite the fact that the first deposition layer manifests a tendency to collapse and spread post-deposition. This variation can be observed from the difference between inner and outer extremities. Also, pore distribution is drastically influenced by clay composition. The control has the most narrow pore distribution, with most of the pores prevailing in the 5-15 micrometer domains. Clay addition determines the broadening of the pore size. Alginate classit 50A pores also occur preferentially in the smaller domains, as the incidence of larger concavities decreases linearly. However, the empty space channels cover large values too. The pore distribution in alginate classit 93 formulation best emulates the Gaussian distribution, while in the case of alginate classit 20A, the incidence of larger pores is superior. Such architecture might be the best choice when dealing with cell cultures, since they must have adequate interconnected networks of sufficient magnitude in order to successfully penetrate through the scaffold for optimal population. To evaluate the in vitro cytotoxicity of the investigated alginate and alginate combined with different types of clay, we assess the MTT tetrazolium salviability and then use supernatants of normal fibroblast cells to measure the LDH release from the matched cells exposed of the undiluted and diluted extracts of the composites for 24 hours. Or the extracts were clear and with a pH of 7. According to the MTT test, the most undiluted or diluted extracts induce a higher viability than 70%, which means that the samples are biocompatible. The only exceptions are represented by undiluted alginate classit 30B and alginate classit 15 extract, which exhibit high cytotoxicities. These results are strengthened by the morphologies and densities of the cells observed under the light microscope. The cells appear modified, round and detached only in the case of the treatment with undiluted alginate classit 30B and alginate classit 50A extracts. As indicated by the LDH release assay, all the sample extracts seems to be non-toxic with the exception of undiluted alginate classit 30B extracts, which induce a release of 40% of LDH in the fibroblast culture media. This outcome implies that the lysis of cells was produced by affecting the plasma membrane integrity. Printable nanocomposites ink and further 3D printed scaffold based on alginate and several types of commercial clays were successfully obtained. The 3D printed scaffold maintained their shared fidelity and presented modified properties as function of the nanocomposite ink used in the synthesis process. Future prospects include in-depth biological analysis in order to check if our nanocomposite materials based on alginate with either classid sodium or classid 93A could act as an extracellular matrix in order to regenerate human tissues. Thank you for your attention.
Thank you for the presentation. It was with a lot of data, and I enjoyed this presentation. I want to ask if there are some questions.
Okay.
Hello, Andrei. It's Luc from Rouen.
Yes.
I'm very pleased to be with you today.
Yes.
Bună ziua.
Bună ziua.
I should have one question, please. Thank you for the nice talk. I have a question about the amount of calcium ion in clays. Did you have any idea of the possibility to have calcium ion in your clays?
That's a very good question.
Because you know, calcium ion and alginate make a very good hydrogel.
Yes, it's the mechanism for making alginate hydrogel. Did you check the quantity, the amount of calcium ions, cations?
No.
No, but
we have calcium from the calcium chloride. With we made the cross-linking process step.
You added a quantity of calcium chloride, but you didn't mention.
No, my scaffolds were immersed in calcium chloride solution.
I see. So the quantity, the amount of calcium in layer silicate is no more important if you put all in a calcium chloride solution. Yes, I see.
Yes.
But I have another question.
Thank you for your answer. Thank you.
The pH is important in such a process. Is this important for you? Did you check the influence of pH?
Yes, it is very important. I must have a pH 7 in order to allow the cells to grow and regenerate the tissue. If I have my scaffold to present, for example, 5 pH, the cells will die. So yes, it is very important.
Okay. Thank you.
Welcome.
There are other questions? If not, we pass to the last presentation in session 1, multifunctional materials and nanocomposites. Thank you too. The talk will be given by Alexandra Sândulescu, and the research is photooxidative degradation of organic substrates over noble metal modified with titanium dioxide. The team is constituted from Alexandra Sândulescu, Crina Anastasescu, Florica Papa, Monica Răciulete, Anca Vasile, Nicu Doinel Scărișoreanu, Tanța Spătaru, Monica Scărișoreanu, Nicolae Spătaru, Maria Zaharescu, and Ioan Balind. All the team is from Institute of Physical Chemistry of the Romanian Academy of Science. So please
Good
go.
afternoon. It can be seen?
Yes.
The sunlight-driven photooxidation processes of organic matter are of great importance for several practical reasons, imply low material and operational costs, are potentially able to clean water and air by mineralization of organic pollutants to carbon dioxide, are attractive alternative routes for selective synthesis of high added value oxygenated products. Material preparation. The photocatalysts employed in this study are titanium dioxide and noble metal modified. Titanium dioxide in this study are silver, gold, and platinum. High surface area titanium dioxide form of nanometric size particles were prepared by laser pyrolysis using titanium tetrachloride as a precursor material. Metal deposition was performed by impregnating titanium dioxide with hexachloroplatinic acid, silver nitrate, and chlorouric acid precursors, followed by reduction with sodium borohydride. The transmission electron microscopy analysis of noble metal dispersed on titanium dioxide prepared by laser pyrolysis shows well dispersed platinum on titanium dioxide support, figure 1A. The silver and gold particles are significantly larger than of platinum. CTEM images in figure 1B and C. Individual spherical gold nanoparticle of around 5 nanometers can be observed in figure 1C. From the X-ray powder diffraction analysis, it can be seen that titanium dioxide is a mixture of anatase and rutil. The crystal size is in between 18-21 nanometers, and that it increases with the deposition of noble metals. Experimental setup used for photocatalytic test composed of quartz reactor provided with quartz window, AM1.5 simulated light source, liquid and gas sampling system, gas and liquid chromatographs, and chillers to control the temperature of glass condenser, mains 5 degrees Celsius, and reactor 18 degrees Celsius. For each test, 110 milliliters of 50 milligrams per liter phenol aqueous solution containing the suspending catalyst powder, 0.05 grams, were placed into the photoreactor. Prior test, the suspension was kept in dark for 30 minutes under steering to attain equilibration of the experimental system. Typically, one experiment consisted of light irradiation of liquid suspended photocatalyst for 360 minutes. Results. Figure 4 shows that on titanium dioxide there is a total oxidation in the presence of the hydroxyl radical, while on metal modified titanium dioxide there is a mild oxidation in the presence of the superoxide anion, oxidation that leads to organic products, benzoquinone, hydroquinone, and 1,2-dihydroxybenzene could be observed only on metal-loaded titanium dioxide. In contrast, over bar titanium dioxide, phenol was mineralized directly to carbon dioxide. The time course of products formation during photocatalytic oxidative degradation of phenol over bar and metal modified titanium dioxide exposed to simulated solar light is present in figure 5. It can be observed that the formation of oxygenated products takes place only on metal-loaded titanium dioxide, the amount of hydroquinone increases rapidly in the first hour of the reaction. The activity order for hydroquinone formation over metal-loaded titanium dioxide is silver, gold, platinum-loaded titanium dioxide. Transient formation of benzoquinone was observed only over platinum and gold modified titanium dioxide. The amount of benzoquinone decreases progressively, vanishing completely for gold modified and remaining at low concentration in the case of platinum. The evolution of reaction selectivity to oxygenated compounds and carbon dioxide is present in figure 6. Selectivity to 1,2-dihydroxybenzene reaches a maximum at 30 minutes for all metal-loaded catalysts, 78% for silver, 73% for platinum, and 53% for gold. Highest selectivity to oxygenated products was measured for platinum after 30 minutes of reaction time, followed by gold 77% at 60 minutes, silver 61% at 120 minutes, and titanium dioxide 0%. In order to explain to experimental results, we have carried out selective radical trapping experiments. The radical oxygen species analyzed are hydroxy radical and anion superoxide. In case of oxidative degradation reaction mechanism, the photogenerated charge are shuttled to phenol by intermediation of reactive oxygen species. Hydroxy radical is a powerful non-selective oxidant, whereas anion superoxide is a weak oxidant. Detection of radicals. The formation of free hydroxy radicals was probed by monitoring the development of fluorescent umbelliferon results in the reaction between non-fluorescent coumarin and hydroxyl radicals. Figure 7 show how the amount of hydroxy radicals rises gradually in time for all photocatalysts exposed to solar light. The formation of anion superoxide over metal loaded titanium dioxide was evidenced indirectly by detection of XTT formazan, which is the product of reaction between XTT and then anion superoxide. The measurements were carried out with UV spectrophotometer. Figure 8 show the evolution of anion superoxide relative concentration over the catalyst exposed to solar light. We can observe in figure 8 the anion superoxide is not formed to titanium dioxide, while hydroxyl radical is formed in case of all catalysts. In conclusion, this study revealed the light-initiated photooxidative pathways for an organic substrate with aromatic ring, in this case phenol, over titanium dioxide charged with noble metal, silver, gold, and platinum. The analysis of the complex phenomena associated with the photocatalytic reaction focuses on the oxidative conversion reaction mechanism of phenol, including the formation of reactive oxygen species, hydroxy radical, and anion superoxide. The best catalyst for depollution is titanium dioxide, and the metal-loaded titanium dioxide are suitable for mild oxidation of phenol to oxygenated products. Thank you for your attention.
Thank you too for this presentation. Also,
I can hear well.
now you hear me. I want to thank you, yes, for your nice presentation, also very interesting. And I want to know if there are questions.
Yes.
I have a question. The amount of noble metal was the same in all cases, and which was this amount?
1% by mass percentage.
Oh, it's very good, very good. In increase in performance.
Again, I can't hear.
I told that it's very good because to increase in performance is very important, and you obtained something very interesting. Did you check if there are changes in crystalline phases of titanium dioxide? You can hear me? Yes? Okay.
Can you repeat the question?
If you know, you observe a change in crystalline phases of titanium dioxide by introducing these noble metals.
Yes, from the X-ray powder diffraction, but because the percent of noble metal is small.
Yes. It's not an important change. And the phase is rutil or what? What is?
It's a mixture of anatase and rutil.
Okay. Thank you very much. Thank you very much, and congratulations. I want now to close this session and to thank to all the participants, especially to those which made the presentation. I think that we can be very happy because we have good young researchers, and they have all conditions to develop and to become very good specialists in their field. I thank you very much, and now we will begin the third session, and I give the leadership to Professor Rodica Mariana Ion to chair this last session. Thank you. Goodbye.
Have a nice day. Thank you.
Thank you, Dr. Sârbu. First of all, hello everyone. My name is Rodica Ion, and it is my pleasure to open the section 3, Innovative Technologies and Cultural Heritage Protection, with the first paper. The presenter is Andreea Gabriela Oporan, and the paper is entitled Solid State Synthesis, Characterization and Color Properties of Cobalt Mangan Doped Charoid Ceramic Pigments. Please, Andreea.
Can you see the presentation?
Yes, it is okay. You may let the registration. It should start.
You can hear the presentation?
Up to now, no. Press start. Or even it is not possible, you can speak by yourself. Yes, maybe now.
Hello, my name is Silvana Andreescu, and I will present to you my work entitled Solid State Synthesis, Characterization and Color Properties of Cobalt Manganese Doped Charoid Ceramic Pigments. Special thanks to Irina Atkinson, Luminița Predoana, Anca Irina Gheboianu, and Rodica Mariana Ion. First, I will begin with a brief introduction, then I will present to you the materials and methods, results, conclusions, and of course, the references. The traditional ceramic industry showed an increased interest in research of new ceramic dyes because they are an integral part of many decorative and protective coatings and are used for the coloration of glazes, ceramic bodies, and porcelain enamels. These ceramic pigments are inorganic crystalline structure which should be thermally stable, resistant to alkaline and acid attacks, and must be inert to the action of molten glass. Caroit is the non-official name provided for synthetic magnesium titanium pseudobruchite and can be considered a potential ceramic pigment due to its high refractoriness, melting point around 1650 degrees Celsius, and high refractive indices, as well as its ability to host transition metal ions into crystallographically distinct octahedral sites, resulting in the obtaining of different colors and shades. Its structure consists of bands of edge-sharing octahedra extending in the B direction. The cations are distributed over two crystallographically distinct and highly resorted octahedral sites, M1 and M2. M1 is larger and more disordered than M2. Each formula unit has one M1 and two M2 sites where magnesium and titanium are accommodated with a certain degree of disorder. In the fully ordered structure, magnesium is portionated in M1 and titanium in M2. The cation ordering affects many physical properties of pseudobruchite type compounds. It may affect its elasticity and other structural changes, and also it may affect its color. Our purpose was to develop and study 4 Caroit solid solutions doped with cobalt and manganese according to the stoichiometry presented on the bottom part of the slide. The materials used to develop the pigments are presented on the slide. We used magnesium nitrate, sodium hydroxide, methanol, and distilled water to obtain the magnesium oxide. Then, we used cobalt nitrate, ammonium bicarbonate, and distilled water to prepare the cobalt oxide. And then, all the oxides were used for the synthesis of the pigments. The first step was to obtain the oxide precursor from the nitrates. Magnesium oxide was obtained via a salt gel synthesis, and cobalt oxide was mechanochemical synthesized. After that, the Caroit pigments were prepared by the classical ceramic route. On the left side of the slide is represented the technological flow for the obtaining of the magnesium oxide. First, a solution of magnesium nitrate was mixed with 100 ml of distilled water on a magnetic stirrer. Then, a sodium hydroxide solution was added dropwise to the mixture, resulting in a white precipitate that was filtered and washed with methanol. The dried obtained powder was then calcinated at 550 degrees Celsius for 2 hours. As you can see in figure 1, all the diffraction interferences belong to the magnesium oxide phase. For the mechanochemical synthesis of cobalt oxide, cobalt nitrate and ammonium bicarbonate were mixed and milled at room temperature, corresponding to the first reaction on the slide. The reaction was observed to be very fast. The color of the mixtures changed frequently until it remained the shade of purple. Obviously, the ammonia gas could be smelled during the milling. The as-milled powder was then washed with distilled water, dried and calcinated at 300 degrees Celsius for 2 hours, resulting in a mixed valence oxide. For the ceramic dye synthesis, the oxide powders were mixed and wet homogenized on a magnetic stirrer. The mixture was then dried and calcinated at 1200 degrees Celsius in alumina crucibles. You can see in figure 3 the resulting Caroit samples after calcination. The figures situated on the left side of the slide represent the X-ray diffraction spectra of the cobalt dope samples, and on the right side, There are the spectra of the cobalt manganese dope samples. The diffraction interferences confirm the effective formation and the relative stabilization of colloid solid solutions in all samples, although with some secondary phases, rutyl and glycolite respectively. It was observed that the residual phases decrease with the increasing concentration of the dopants. Next, at Fourier transform infrared spectroscopy, there were obtained the spectra indicated in Figure 8. These spectra are specific to the colloid. As you can see, there is a set of bands from 615 to 350 centimeters raised to the power -1, characteristics of the bending modes of oxygen, titanium, oxygen, magnesium, oxygen. The samples were also color characterized. The characterization revealed that the cobalt dope samples are less yellow because their B values are lower, and also A parameter increase when the dopant's concentration increase. Moreover, the colors are darker the higher the cobalt and manganese content, as indicated in Figure 10. In conclusion, there was investigated the synthesis of colloid ceramic pigments. The XRD characterization confirmed the formation of colloid solid solutions with residual rutyl and glycolite phases. In the future, it is desired to test and establish the stability and coloring performances of the pigments in a glaze so they can further be applied in the ceramic industry. These are the references, and thank you for your attention.
Thank you, Andrea. We have time for one or two questions, please.
Yeah, I want to put some questions for my colleague Andrea. I have two questions. Which are the advantages and disadvantages of the ceramic pigments, and why there were secondary phases in your pigments?
The ceramic pigments are more thermally stable than the organic ones, and that's an important feature in the ceramic industry. And also they are safer for the ceramic manufacturers. The main disadvantages of them are that the colors are not bright enough, and there's not wide range of colors to choose from in comparison to organic dyes. And there were secondary phases because doping with transition metal ions caused significant changes in terms of phasal composition, elementary cell dimensions, and the distances between the metal ions and the oxygens, causing the formation of rutyl and glycolite, which are more thermodynamically stable. Thank you for your questions.
Okay, thank you.
Okay, thank you. There are any other questions? If not, thank you again, Andrea. The next paper is compositional investigation of a medieval stone cross, 1656. The presenter is Georgiana Iulia Paraschiv. Are you there, Georgiana? Okay. Yes. Please give a share screen with your presentation because we don't see anything. Okay, perfect. We don't hear you. Please check your microphone. The sound is too low. Now we don't see the presentation. The presentation is okay, but the sound is not enough.
My name is Georgiana Iulia Paraschiv.
Okay, please.
My doctoral professor is Rodica Mariana Born. The specialists are Raluca Maria.
We don't hear you. Please try to do something with the sound, make it louder.
Better now?
A little.
The area of the Goviste has a rich, countless vestiges of a historical past. Countless vestiges of historical past. The parish church was Paraschiva, located from the voivodeship of Wallachia, was documented on July 5. According to the archival treasure, 1526, but the church is much older from the Clarkson.
Give slideshow. You don't have slideshow.
We don't hear you. The sound is very low. Stay closer with your computer.
And give slideshow to have full screen. Don't tell me nothing.
Georgiana, give share screen again and try to speak louder, closer to the computer. Okay.
Okay?
Yes.
Okay. Historical context. The area of the Goviste has a rich history, countless vestiges of the historical past. The parish church, Paraschiva, located 8 kilometers from the voivodeship fortress of Wallachia, was documented on July 5, 1705. According to the Dâmbovița archival treasury of 1526, 1848. But the church is much older, from the text carved in a stone on a stone cross being inscribed January 11, 1605 BC, placed next to the place of worship. The paper address on the major challenges of conservation state on expertise. In our study, microscopical optical and stereo methods Manual forward and another plan. Also, some textile could be seen more clearly, the uncomposed motor and textile, while some manual processes are visible, or structure with information independently located or is accompanied by but our measurements are that both sides will be
No.
in the blue, purple, red range.
Where are you staying? Because the sound is very, very low and interrupting all the time. Do you use Bluetooth or something else? Microphone or what else? No, we can't hear you. No, it's not possible to hear anything. I suggest to stop this presentation because it's not possible to hear, neither to see nor to hear, and to start the next presentation. And maybe at the end of this section, we will start again this presentation. I agree.
Yes.
Okay. So the next presentation is evaluation of new and old banknotes using hyperspectral imaging. The presenter is Dan Adrian Vasile. Please, Adrian. Adrian?
Yeah, here, right now I share the presentation. Okay. It's okay.
Yes, it is perfect.
Okay, good morning. My name is Dan Adrian Vasile, and today I have a short communication about a new research area that is under development. When I say new, I refer to approximately 50 years or old. The hyperspectral imaging technology dates from 1972, when NASA launches in space Landsat-1, the first satellite with the purpose to acquisition of Earth images. This remote sensing technique is mainly established on the principle of object reflection, with the other words to electromagnetic wave emission. This technology enables each pixel in the image to obtain the continuous reflectivity curve greater than having the interval between the bands like other general traditional imaging spectrometer. It was used to identify hyperspectral images chamber near inospec green eye with a spectral domain from 373 and 1102 nanometers and a resolution of 10 nanometers. The quality of the spectrum is influenced by parameters such as light intensity, distance between object and camera. In our case, we put this distance 60 centimeters, the quantity of light which goes to camera with to light bulb of 230 watts. This method used consisted in an analysis of a multivariate image in a reflectance spectrum. Faced to the normal color image, which has three bands, red, green, and blue, the hyperspectral image contains much more information. being composed of a very narrow continuous band that consists of hundreds of bands. We can correlate with the method pixel by pixel. We can correlate the spectrum with each points from banknotes correlate and to see the spectrum. The Hyperdata Cube was visualized with the help of MATLAB software hyperspectral image processing toolbar, and we can calculate the spectral angle between the two spectrums to determine the level of their similarity. In conclusions, the application of high-text techniques due to its non-destructive abordation for the cultural heritage science can be used to build a spectral library for banknotes from different periods and with different values. The spectral library will be used for authentication, the accuracy from cultural heritage. In forensic science, the same method that uses a spectral library can be applied to evolution and depistation, the counterfeit of mind. Thank you for watching.
Thank you, Adrian. We have time for one or two questions, please.
I have a question, if it's possible. How can you determine the level of similarity?
The level of similarity between two spectrums in SEM algorithm, spectral angle mapper algorithm, we can use a formula which means inverse of cosine of vectorial product of the two spectrums divided by the scalar products of each component of the first unknown spectrum and the reference spectrum. If we have a spectral angle lower, the level of similarity is bigger, and that's okay.
Okay, thank you.
Thank you, Andrea. There is any other question? If not, I will pass to the next paper. Calcium aluminum layered double hydroxides for heritage conservation, preparation, characterization, and kinetic stability of their dispersion in solvent. The presenter is Claudiu Eduard Rizescu.
Hi, good afternoon. One moment. Yes. Yes, can you see the presentation? Yes. Se vede prezentarea?
Yes, it's perfect. You can start.
Okay. Today I'm going to present the calcium aluminum layer double hydroxide for heritage conservation, preparation, characterization, and kinetic stability of their dispersion in solvent. Introduction. In time, buildings and the stone monuments, especially those exposed, suffer a loss in aesthetic and mechanical properties. The mechanisms that are responsible for this degradation are the transport of water in pores, freeze-thaw cycles, environmental pollutants like sulfates, nitric oxides, and even CO2 can alter the properties. The crystallization of salts in the pores, in the material's pores, and biodeterioration. To stop this destruction of stone, some treatment must be applied to stop this deterioration and improve the cohesion between the material grains. These consolidants must possess some properties. They must improve the cohesion within the grain of the building materials. They must be compatible with the building materials. They should not alter the aesthetical properties of the stone. They should not facilitate the microbial growth within the stone. thus they must have a bacteriostatic effect, and they must be somehow able to stay stable in a dispersion in material. Layer double hydroxide-like materials have attracted a lot of attention due to some of their properties. Even in the fields of building materials science, they proved to be efficient as a hardening accelerator for building materials. They can possess bacteriostatic properties. They can absorb the undesired anions within the stone materials, anions that produce that are in the composition of some salts like sulfates that are responsible for the degradation of stone. Layer double hydroxide are a group of synthetic or natural crystalline materials consisting of brucitic layers composed of divalent and trivalent metals. When trivalent cations substitute a divalent cations in a lattice, a positive charge will be obtained. This positive charge can be neutralized by an anion. The anion that are found in the interlayer space. Their general formula are described here. In the interlayer, several anions can substitute the existing species. There are a series of stability of the interlayer. The most stable anions in the interlayer is the carbonate, and the less stable is the nitrate. Different cations can enter the brucitic layer by isomorphic substitution, and depending on the type of cations that we use in the synthesis, materials with different properties can be obtained. The study aim is to obtain a calcium aluminum layer double hydroxide using some facile synthesis method adapted from literature to characterize this obtained material and study their stability, the stability of their dispersion in different solvents. The material preparation, we use two different preparation methods. First of all, we use first method consisted of a co-precipitation at low supersaturation and constant pH. Thus, the solution containing the metallic chloride salts were added dropwise along with the precipitating agent, sodium hydroxide 2 molar, were added dropwise under constant stirring, and we control the pH to be as close as 12 as possible. The obtained precipitate was aged 12 hours at 80 degrees Celsius, filtered, washed until a neutral pH, and dried in air. We obtained the first material. The second synthesis were co-precipitation at high supersaturation. Thus, we took 100 milliliters of solution containing calcium cations and aluminum cations with the ratio of 2 to 1, calcium to aluminum, and we added this solution into the sodium hydroxide, into 100 milliliters of sodium hydroxide. We added this solution fast under vigorous stirring. The obtained precipitate was also aged 12 hours at 80 degrees Celsius, filtered, washed until a neutral pH, and dried in air. We obtained the second material. Both materials were characterized using X-ray diffraction, infrared spectroscopy, and X-ray fluorescence spectroscopy. In the X-ray diffractogram of this material showed that only the diffraction line characteristic to a pure layer double hydroxide structure, or in this case hydrocalumet, are called the subgroup, were obtained. No other phase can be observed in this diffractogram, showing that the method used were highly efficient. The material were analyzed using Fourier transformed infrared spectroscopy, and the spectra obtained present some bands characteristic to these materials. First are the bonds at around 3,500 centimeter to -1, which are characteristic to the water and the interlayer hydroxide groups. Also, the bands at around 1,600 are also characteristic to the hydroxide groups in the interlayer. The bands at around 1,317 are characteristic to carbonate anion. These bands prove that in the interlayer, some carbonate groups have entered, and the amount of carbonate in the interlayer groups differ from one materials to another, showing that the synthesis method took an important role in the presence of CO2 in the material. The X-ray fluorescence spectroscopy showed that the materials that no important amount of calcium or aluminum cations were lost. The desired ratio have been achieved of 2:1 calcium to aluminum, and also the presence of carbon show us again that the interlayer is composed of carbonate anions and chloride anions in a different ratio. The obtained materials were characterized where the stability of these materials in the water and water ethanol mixtures were analyzed by the variation of absorbance over time, analyzed by UV spectrometry. The kinetic stability was in each mixture was monitored for 300 minutes, and measurements were made from 10 to every 10 minutes. The kinetic stability of these materials present good value, both in water and water ethanol mixtures. The best results obtained are in the case of the layer double hydroxide obtained by the second method, over 80% stability after 300 minutes, showing us that these materials dispersed in the water ethanol form good stable dispersion. And conclusion, both methods led to pure layer double hydroxide phases. The interlayer consists of hydroxyl and groups, chloride and carbonate. The material dispersed in water and water ethanol form stable dispersion, and the further studies are needed to understand the properties of consolidants. Thank you for your attention.
Thank you, Claudiu. There are any questions?
Yeah, I have a question.
Yes.
How the preparation method will affect the final material?
Thank you.
The type of method.
Thank you for your question. The material, the preparation method strongly affect the final material properties. We choose two different preparation methods. The first one, which consists of slow addition of salts along with the precipitating agent, it's good to obtain material with good crystallinity and big particle size. The second one, which is a fast addition of salts into the precipitating agent, will lead to small particle, but with lesser crystallinity, because it will form more crystallization center, at least theoretically. This is my question.
Thank you.
Okay, there is any other question? If not, I want to say that this hydrotalchides, layer double hydroxides, as Claudio prepared, is one of the last generation materials used in conservation and restoration of different monuments, surfaces, and the results are very promising. Already we submit a patent request, also a publication. There are many other experiments under progress. So we are very confident that such type of material will give us very good results. Thank you, Claudio.
You're welcome.
Okay, I will pass to the next presentation. Carla Cezarina Pădurețu will present us characterization of mortars prepared with different biopolymers and yellow iron oxide. Please, Carla. Carla, are you there?
Yes, good afternoon.
Okay.
Is the presentation can be seen?
No, it's not visible up to now.
Acolo de share screen din nou. De share screen, da. Acolo, nu. Așa, și selectează prezentare.
Now, is it visible?
Yes, it's perfect. Please.
Thank you very much.
Okay.
Can I start?
Yes, please.
I'm Carla Pădurețu, and I will present the paper, Characterization of Mortars Prepared with Different Biopolymers and Yellow Iron Oxide. Over time, the meteorological factors and harsh environmental conditions have seriously affected the constructions that are mostly part of the national cultural heritage. Mortars are materials used in masonry to fill the gaps between bricks, as seen in the pictures, and are usually made of sand, water, and cement or lime. If the mortar between bricks is affected, this will certainly lead to an overly affected structure in time. In order to prevent such events, a restoration needs to take place. In this study, the influence of different biopolymers on the physical mechanical properties of mortars was studied. As seen from the Great Wall of China, the sticky rice was used for the construction along with the calcium hydroxide. The materials and methods used in this study consisted of chitosan, sodium alginate, and starch as biopolymers, and the yellow iron oxide as an organic pigment. The influence of the yellow iron oxide on the properties of the mortars prepared were evaluated. The physical mechanical properties consisted in determination of mechanical properties by compressive strength, a colorimetric analysis using conditions under illuminant C and 2 degree, determination of freezing towing resistance, and analysis by optical microscopy. The compressive strength has shown a slightly better resistance when using biopolymers. The addition of yellow oxide to sodium alginate can be seen from the tables that it was slightly improved. Although for the other materials, biopolymers, the compressive strength didn't show a good improvement. For sodium alginate, the results were better. The colorimetric analysis was measured to evaluate the total color difference and the yellowness index. The charts from the left side show higher total color difference for samples with sodium alginate, as can be seen. The total color difference is measured on a scale from 0 to 100, a value less than 1, which can be seen from sand and chitosan, sand, white cement, and starch, suggests a difference that is not perceptible by the human eye. A value between 1 and 2 is perceptible through close observation, which is the case of mortars prepared with sand, white cement, yellow oxide, and chitosan, then yellow oxide with starch. The value between 2 and 10, it can be seen from the samples prepared with sodium alginate and sodium alginate with yellow oxide. The value is between 2 and 10, and this is perceptible at a glance. The yellowness index from yellow iron oxide has changed the yellowness index with maximum 15%, as can be seen in the upright chart, compared to the samples without yellow oxide. Moreover, sand has a major influence on the, as seen from the chart in center. Sand has a major influence on the yellowness index. Although chitosan and sodium alginate have close values, by the combination with sand and white cement, respectively with yellow iron oxide, the sodium alginate shows higher yellowness index values. The freezing thawing resistance has shown that the samples with sodium alginate and yellow oxide can resist up to 47 cycles, as can be seen here in the chart from left, compared to the sample without yellow iron oxide, which resisted only up to 25 cycles. From this determination, the freezing thawing, the lower limit is 25%. When there is a 25% loss of the material, it can be considered that the material has seriously been affected. The highest resistance was obtained for the samples with chitosan and starch, which can be seen they resisted up to 50 cycles, which is the minimum value for this type of analysis. In the right graphs, right charts, okay, it can be seen that they resisted up to only 47 cycles. The optical microscopy has shown the presence of aggregation. The reference sample had a size between particle size between 151 and 636, while by the addition of yellow oxide, the particle size has slowly increased. It can be seen that higher aggregates were formed. Also, chitosan has a particle size distribution between 100 and 150 on average, and by the combination of yellow oxide, higher aggregates were observed. The same case is for sodium alginate. Can be seen that they formed a very large aggregation and the homogeneous phase. As conclusions, the mortar samples showed some important changes in their properties. The compressive strength showed that chitosan and starch exhibited good durability, while the samples with sodium alginate, the compressive strength was increased. The colorimetric analysis showed that the highest values of yellowness index were obtained when using sodium alginate. The freezing thawing resistance showed that the samples prepared with sodium alginate and yellow oxide have shown a better resistance than the samples prepared with only sodium alginate. And in conclusion, the sodium alginate is not a good material that could be used for enhancement of mechanical properties, physical mechanical properties. Thank you very much. Can you hear me?
Sorry, I forgot to press mute, unmute it. Okay, thank you very much for your nice presentation.
Thank you.
There is any question, please?
Yeah, I have a little question. Why did you use the yellow pigment and not other pigment?
It is a very good question. I have tried to use green iron oxide, but the total color difference was very high from the color, as seen from the colorimetric analysis, which I have previously determined. And moreover, the yellow iron oxide was mostly used in masonry and constructions because of its warm color. has a natural, pretty much a natural color, and it could be used in lower quantities than the green oxide, which is a very powerful, gives a very powerful color, even at lower concentrations.
Okay, thank you.
Thank you.
There is any other question? If not, with your permission, I will try to connect again with Georgiana Paraschiv. Maybe she solved the problem with the sound. Georgiana, are you there? We still have time until 6 o'clock.
Good afternoon again.
Okay, now it's perfect. Please share your presentation.
Good. Let's try it again.
Okay.
It must be fine now.
Okay, we hear you very well.
That's good.
Please share the presentation.
Thank you.
Okay.
Okay.
Press there to make it bigger.
My name is Georgiana Iulia Paraschiv, and my doctor professor is Rodica Mariana Ion. My specialists are Luca Maria Știrbescu and Anca Irina Gheboianu. Historical context. The area of Târgoviște has a rich history, with countless vestiges of the historical past. The parish church, Pius Parascheva, located 8 kilometers from the voivodeship fortress of Wallachia, was documented on July 5, 1705, according to the Dâmbovița Civil Treasury, 1526-1848. But the church is much older, from the text carved in stone on a stone cross, being inscribed January 11, 1656, placed next to the place of worship. Aim of the paper. The paper aims to address one of the major challenges of conservation state of stone artifacts surfaces. In our study, the microscopical investigation, Fourier transform, X-ray diffraction, and wavelength dispersive X-ray fluorescence methods could be observed some degradation signs as 2 sides, one white colored and another black. Also, some textile could be seen, most probably due to some composite motor and textile. Meanwhile, some weathering processes are visible as follows: porous structure with cracks, alveolar formation and detachments, effusive, unevenly located gypsum deposits accompanied by microbial population development. By calorimetric measurements, it was confirmed that both sides of the investigated samples show that B is negative and A is positive. The color of the sample being in the blue, purple, red range, which could be an indicator for different weathering layers and migration process between both sides. In order to establish the importance of the cross in the histography of the monuments of Târgoviște and its surroundings, we want to pay due attention and highlight the monument of real significance. These are materials and investigation equipments: optical microscope Novex, stereo microscope, and calorimeter Konica Minolta. Results and discussion. In our study, the microscopical investigation methods could be observed some degradation signs as 2 sides, one white colored and another black. Also, some textile could be seen, most probably due to some composite motor and textile. Meanwhile, some weathering processes are visible as follows: porous structure with cracks, alveolar formation and detachments, effusive, unevenly located gypsum deposits accompanied by microbial population development. Weathering effects: cracks, alveolar formation and detachments, efflorescence and gypsum. These are white samples, and the black ones are microbial population and microbial population cracks, alveolar formation and detachments. Fourier transform X-ray diffraction and X-ray diffracted results. By Fourier transform X-ray, some major metallic oxides have been identified as calcium oxide, silicon dioxide, aluminum oxide, ferric oxide, strontium, silicium, well correlated with X-ray diffraction results, where the following species have been identified as follow: calcite, calcium carbide, perlite, and minor quantity of iron, strontium, and silicon, magnesium, aluminum derivatives. Calorimetric measurements. By calorimetric measurements, it was confirmed that both sides of the investigated sample show that B is negative and A is positive. The color of the sample being in the blue, purple, red range, which could be an indicator for different weathering layers and migration process between both sides. In order to establish the importance of the cross in the histography of the monuments of Târgoviște and its surroundings, we want to pay the attention and highlight the monument of real significance. The investigated sample shows specific bands of calcite and calcium carbide. The sample presents water too as a humidity presence proof and calcium sulfate as weathering sign. Now the conclusions. The medieval stone cross 1656 is created from calcite stone due to the porosity and fragility aspect. The weathering processes are visible by optical and stereo microscopy. Some weathering processes are visible as follows: porous structure with cracks, alveolar formation in detachments, effusive, unevenly located gypsum deposit accompanied by microbial population development. By calorimetric measurements, it was confirmed that both sides of the investigated samples show that B is negative and A is positive. The color of the sample being the blue, purple, red range, which could be an indicator for different weathering layers and migration process between both sides. In order to establish the importance of the cross in the histography of the monuments of Târgoviște and its surroundings, we want to pay the attention and highlight the monument of real significance. Other investigations are in process, revealed the presence of calcite, calcium carbide, and calcium sulfate. Thank you for your attention.
Okay, thank you for your presentation, nice presentation. There is any question?
Yes, I have a question, please.
Yes, please.
First of all, congratulations for the very nice subject and the very nice results. I have a question. Do you perform any sampling on the object, or was it, let's call it natural sampling, you know, pieces detached from the larger object?
I think I have to answer, need to answer. We analyzed only detached sample from this monument because it's a monument included in the Natural Cultural Heritage Register. So we didn't want to affect in any way this monument.
Okay, thank you. I assume that, and that's why it should have been important to tell that in the beginning of the presentation, that no, the larger object was not affected by any investigation. That was very important. Thank you.
Thank you. So, there is any other question or comment? If not, I want to thank you to all presenters, to all participants which listen our presentations. I wish you a nice afternoon and nice evening, and see you tomorrow. Goodbye.
Thank you, Professor.
Thank you too.
This concludes our day for the first day of our exploratory workshop. I wish you all a very nice evening, and let's see you tomorrow in the morning for the second day of our workshop. Thank you and goodbye.
Good evening.
Goodbye.
Good morning. Good morning, everyone. Dear guests and dear participants, for the NeXT-Chem workshop, we arrived to the second day of our workshop after nice and fruitful days, the first day, when we had two invited lectures from Professor Arben Merkoçi and from Professor Silvana Andreescu. And also we had the presentations of the participants with registered papers. And now we are honored to have the third plenary lectures sustained by Professor Luc Picton from University of Rouen, France. Professor Luc Picton is a dear friend of us and a collaborator. He is working mainly with a group of led by Professor Andreescu. And for this, I want to ask Professor Andreescu to introduce Professor Luc Picton. And thank you very much, Professor Luc Picton, for your presence here.
You're welcome. Thank you.
Professor Sârbu?
Yes, it's a great pleasure for me to introduce one of my best friends. We are friends from almost 20 years. And we have very good relations, which developed by French-Romanian colloquium on polymers. So I know the activity of Luc since a long time. I want to present some data about him. He is a professor in laboratoire polymer biopolymer surface in the University of Rouen. Now he is a professor class exceptionnel since 2020. He from 25 he got habilitation in leading research. Now he is in charge as Vice Dean with research of the University of Rouen. He has Kirsch Index 32. He published 111 articles and 80 proceedings. He is working especially in the field of chemistry and physical chemistry of colloidal complex systems in aqueous solutions, mainly based on natural polymer and especially on polysaccharides. Recently, he entered in a new field, which is very close from my field. It's working in covalent immobilization of enzyme on matrices, and especially there are some new materials which are smart polymer because they are sensitive to some external stimuli, as for instance pH or temperature. and so on. So I want to say also that Luc is a big friend of Romanian people. He received a lot of Romanian young people as in stages in Rouen. And I am very proud that he accepted to honor our symposium with his lecture. I want to tell him good success for this presentation. Please.
Many thanks, Andrei. Many thanks. I love Romania, really, sincerely. and it's a pleasure for me to be with us, with you. I apologize for my English because I'm not so fluent, but I will try to do my best. So perhaps I can put my presentation on. Did you see it?
Yes, yes.
Yeah, yes, okay. One more time, many thanks for the invitation. As you say, André, I come from Rouen in Normandy. So perhaps you know the Normandy, some pictures. Here's the impressionist pictures of the cathedral by Monet and Mont Saint-Michel, perhaps you know it. And I will talk about this morning about smart water soluble polysaccharides for adaptive properties. So it is possible to change. Yes, like that. So my lab quickly is named Polymer Biopolymer Surfaces. There is 5 groups in this lab, and I work in one of these groups called System Colloidal Complex. And in these groups, our main objective is to work on modified or unmodified polysaccharide. Our main objective is the elaboration of smart system. We can work on self-assembling in solution, and we will see some example of that. On interface functionalization, as Andrei said, we also work on hydrogel, which can be physical or chemical one, and also on polysaccharide enzyme systems. So on which polysaccharides we worked? On exudate 1, mainly the most known exudate polysaccharide is Arabic gum. Also on algae polysaccharides as alginate, less with carrageenan. We also work on a new macroalgae polysaccharide. On plant polysaccharides as flaxseed, we have a lot of flax plant in Normandy, and cellulose derivative, which are industrial polymers. And we also work on bacterial polymers as xanthan, scleroglucan, which are very rigid polysaccharide and give rise to very interesting rheological properties. We also work on hyaluronan, because hyaluronan is highly biocompatible and opens the way to a lot of biomedical application. The pullulan is our model polysaccharide, because pullulan is very simplistic polysaccharide, is linear, is neutral, there is no ramification, so we can modify it and see what brought our modification. Obviously, the advantage of polysaccharides are notably that they are renewable resource. They are highly biocompatible and biodegradable. They also offer large functionalities for modification, and they present a large potential of structure. So here is the polysaccharide hydrophilic backbone in blue here. And what we do we incorporate some charge groups or hydrophobic groups. I will show you also some example of stimuli modulated groups, mainly on the temperature stimulus. We also can graft enzyme or peptide or active molecules, and we can also conduct some cross-linking. We can also use reducing sugar at the end of the chain to graft some such groups, and then we have some block structure. The other structure or comb structure. So we can have various architectures of such polysaccharides, and such modifications permits us to control three main properties. The first one is obviously the rheological one. We can see with associative polymer that are amphiphilic ones that we can control the rheology, the shear thinning behavior, the thixotropy, and there is a lot of application in many domains as paints, cosmetics, and so on. Another property which is interesting to control is interfacial control. With amphiphilic polymer, we can control such properties, and there is a lot of application in surface modification or the control of suspension or emulsion. The third domain is the drug release. So we can use such polymers to embed some active substance and to try to control the release of such substance. But we can also use a matricial system to produce cell cultures. Moreover, we can have stimuli-sensitive characters that permit to have on-off control of such properties. So I will speak very quickly about natural polysaccharide, and then I will present you some results on functionalized polysaccharides, mainly associative one, that means amphiphilic polymers. Then I will show you some smart adaptative polysaccharides, and mainly stimuli sensitive to the temperature. And if I have the time, perhaps I will show you a recent study in my lab with the elaboration of a three-dimensional network conducted by enzymatic catalysis. So natural polysaccharides are well known for their interesting property, and intrinsically they can be neutral or polyelectrolyte. Usually we can find a lot of anionic polysaccharide. For cationic one, it's really rare. My knowledge, there is no cationic polysaccharide in the nature, but we can transform some polysaccharides into cationic one. I mean the ketosane, for example. Some polysaccharide systems show associative characters. You can find some hydrophobic interaction with amphiphilic characters. You can also find some hydrogen interaction, giving rise to the elaboration of gel structure. And there is also a lot of peculiar conformation. So there is intrinsically some very interesting properties. As for some example, viscosifying properties with polysaccharide that presents very rigid backbone with helix conformation. I think to xanthan, for example, or scleroglucan. You can also have obtain very interesting gelifying property. The best one, the more new one is probably alginate calcium system, but you can also elaborate gel with scleroglucan in addition with borax, or with the control of temperature with agar-agar, for example. Some of them are also emulsifying, and I speak just before about gum arabic, which is an exudate gum produced by some trees like Acacia senegal or Acacia seyal. We can find such trees in a sub-Saharan area in Africa. Gum arabic is largely used to stabilize emulsion in beverage, for example, like orange juice or Coca-Cola. So what we try to do is to functionalize such polysaccharide, and to do that we conduct some chemical functionalization. Here I will show you amphiphilic polysaccharides. So we are going to incorporate some hydrophobic group. Most of them are alkyl one, and we can play on the length of the alkyl chain, of course, and on the degree of incorporation. We can graft such hydrophobic group on the backbone to obtain comb structures, but we can also graft at the extremity of the chains and to obtain block structure. First of all, I will speak about comb structure, and some example of block structure will be presented with thermosensitive properties. So what is the interest to graft hydrophobic moieties on hydrophilic backbone? It is an apparition of self-association properties, because hydrophobic groups try to escape the water and to self-associate. You have two kinds of association, intramolecular one and intermolecular one. If we see the influence of concentration of polymer, obviously in the very dilute regime, you have mainly intramolecular association that lead to the collapse of the chain and the decrease of viscosity. Such polymer have been studied a long time ago in the last century with a lot of very short chain that calls polysops. And polysops were able to collapse as that, even in very concentrated regime. So in the 80s of last century, Landau tried to incorporate less chain, but chain with more long carbon. And then we can obtain some different association. And when we increase the concentration, we have first time the formation of aggregate, and when we are about the critical concentration, association becomes mainly intermolecular, giving rise to three-dimensional physical network with very interesting rheological properties. Landau was a researcher of an industrial company, and he worked mainly on as a cellulosic derivative. Such polymers are actually sales to improve the rheological property of paints, for example. Such behavior depends on the nature of polymer, the rigidity, the molar masses, for example. It depends also on the hydrophilic lipophilic balance. So the nature, the length, the distribution, the incorporation rate of hydrophobic groups, the neutral or charged character of the polymer, and also it depends on the external parameters as ionic strength or pH or temperature. So the chemical grafting is not so difficult. Here you can see the pullulan. I said you that pullulan is our model polysaccharide. It is more easy to graft on carboxylic groups. So in the first time, we incorporate such carboxylic group to the pullulan to obtain the carboxymethyl pullulan. For that, you can incorporate a monochloroacetate sodium and in a mixture of water and isopropanol, and then you obtain carboxymethyl pullulan with a substitution degree about 1. With this polymer, you can fix an alkyl chain. Here, there is an example with an alkyl chain. This is a bromoalkane. But first of all, we have to choose the good solvent, because obviously alkyl chains are not soluble in water. So we have chosen the DMSO, and to obtain the solubility of polymer in DMSO, we transform the CMP salt with the CMP salt of tertiobutyl ammonium. And then both are soluble in the DMSO, and then we can have the reaction of grafting. Then you put some natrium chloride and the polysaccharide precipitate. We obtain substitution degree from 10 to 45, about that. We can't go above 45 because after that, above that, the polysaccharides become not soluble in water. So the main behavior, and we have evidence that with physical-chemical characterization in dilute regime, as a determination of molar masses or dimension as hydrodynamic radii. And then we can see that the size of the polymer, the size of the coil is quite unchanged for low amount of grafting, but when you increase the amount of grafting, you can see the apparition of large aggregates, and then for the higher amount of grafting, you can evidence the collapse of the chain with a diminishing of the hydrodynamic radius. More interesting is the properties when you increase the concentration. Then you can have the transition of intramolecular to intermolecular association, and we can fix the radical properties from viscous behavior to viscoelastic as a weak gel behavior, and we can also obtain elastic system that we call pseudogel. This is a question of connectivity, of course. As we said just before, such behavior are controlled by the nature of the polymer, natural characters, charge characters, the flexibility, the molar masses, the concentrations, the hydrophilic, lipophilic balance, and also external condition. So Lendl, I talked about Lendl just before, as shown in the past on hydroxyethyl cellulose, HEC, modified with alkyl chain from 8 to 16 carbon, that the viscosity depends on the amount of grafting groups. The best result was obtained for the 16 carbon chain, but you see the maximum of viscosity appears for a very low amount of alkyl. And then above this amount, the polymer precipitates quickly. So this polymer was chosen to be the industrial one. I worked on such polymer at the beginning of my work in researches, and here I can show you the behavior on rheology with the precursor HEC and the industrial modified polymer HM-HEC. Solution have been fixed at 3% at 25 degrees. Here you can see the flow behavior of the precursor and of the modified polymer. And you can see that the increase of viscosity is very important. Also, the shear thinning behavior is improved with the modified polymer. Perhaps you know that you can obtain the relaxation time of the system, because you can imagine that the system is an entangled system with a dynamic of polymer inside this entanglement. We can obtain the relaxation time thanks with these curves. You take the viscosity at the plateau, then you divide by 2. With this value, you cross the curves, then you draw down to the shear rate. You take the inverse of the shear rate, and then you obtain the relaxation time. Here you can see that the relaxation time is largely higher for the modified polymer. So the dynamic is slowed by the interaction, and we have a sticky reptation. Here you can see the rheological profile in a dynamic mode with a determination of elastic modulus G prime and viscous modulus G double prime for the precursor and for the modified polymer. For the precursor, for all the range of frequencies, we are mainly viscous. And for the modified polymer, you can see that we are mainly viscoelastic. And the cross point of the moduli give also an information on the relaxation time. You take the cross point, you draw down to the frequency, and take one more time the inverse frequency, and you obtain the time of reptation. And you can see that the time is slowed down, is higher for modified polymers than for the precursor. Such polymers can also be used to the control of adsorption. Here are some results about the theory of CMP with a different degree of grafting with octyl group. And you can see the adsorption of latexes of polystyrene sulfonate. The latexes were about 100 nanometers. And then you can see that if you put in the contact of latexes or a different modified system, you have a large absorption. Here you have the precursor, which obviously did not absorb on the latexes, which are hydrophobic one. And here you can see the adsorption, and this adsorption is higher when the amount of grafting groups increase, and then you reach a plateau. We try to see if we can determine the thickness of the adsorbed layer. And it was possible by dynamic light scattering. And here you can see that the layer of adsorbed, the thickness of adsorbed layer decrease when you increase the incorporation of grafting group of alkyl groups. This is easily to understood, because when you increase the amount of alkyl group, you increase also the anchor point of adsorption on the polymer. And as you can see on this small image, you diminishing the thickness of the adsorbed layer. I said you also that we can use such system to incorporate some active substance which are often amphiphilic or hydrophobic one. So to try to see if we have the formation of hydrophobic clusters, we conduct some fluorescence spectroscopy measurement with a pyrene probe. In fact, pyrene give 5 peaks in fluorescence spectroscopy, and the ratio of the first and the third peak give interesting information of the environment. For a higher ratio, you have a polar environment, and when you see this ratio decrease, it means that you have the formation of apolar cluster. Here you can see that for CMP with 49 degree of octyl incorporation, here you have the evidence of the formation of hydrophobic clusters. So such clusters can be used to incorporate active substance. And we have worked with pharmacian researchers in Paris to see if it is possible to have the sequestration and the drug release of active substance. We have chosen, yes, the docetaxel. Yes, this molecules is largely used to in an oncology domain to have good result on cancer cells. Usually, docetaxel is embedded in a mixture of water and ethanol with a surfactant, which is here twin 80. The objective of this study was to use the CMP with a 45 degree of incorporation of alkyl groups to have the sequestration of the docetaxel and to see if the efficacy is good on cancer cells, and also to see if there is an impact of cytotoxicity, because you know that docetaxel in the commercial mixtures give rise to a lot of secondary effect and is largely cytotoxic. So our results are good because we have evidence that both system, commercial one and our system, are quite equivalent in terms of efficacy, but our system here with the black symbols are less cytotoxic than the commercial system. So it was a very good result. The problem was the quantity of the solution to have the same effect with our polymer. Needs more volume of injection compared to the commercial one. So I arrived to stimuli-sensitive associative polymer. The aim is here to develop polymers with tunable properties, notably here with temperature stimulus. So you know there is some polymer that presents interesting properties that calls low critical solubility temperature. For such polymer, you have a desidratation that occurs above a critical temperature. That means the polymer becomes hydrophobic above such temperature. A large example exists in the literature, and the most known LCST polymer is probably the poly and isopropyl acrylamide. But there is also some block copolymer, block of polyethylene oxide and polypropylene oxide that presents also such interesting properties. So we have tried to incorporate some LCST group polymer onto the backbone of our polysaccharide with a branch-head structure or block structure. I will show you some example of chemically grafting on the polysaccharide, and also a study with a physical association of LCST group on the polysaccharide backbone. Then a short example of a block structure. So what we expect of such system is, for example, a transition between a sol property, solution property, below the critical temperature, and then when we increase the temperature, we expect an association that gives rise, for example, to a gel transition with the formation of hydrophobic cluster that permit, for example, to sequestrate some hydrophobic molecules. Here an example with industrial polymers with a thermosensitive association. There is some polymer like hydroxypropyl cellulose or methyl cellulose, which evidence such property. Here we can see in dilute regime by a simplistic measurement of the adsorbance as a function of temperature, the apparition of turbidity, that you can see here, the turbidity, when you reach a critical temperature. And you can see that this critical temperature can be changed by addition of salt, for example. If you add salt, generally you decrease the critical temperature. So the choice of polysaccharide for grafting was, of course, our purulane, and we also have tried the electroassociation on alginate using the carboxylate function. The choice of thermosensitive group was the jeffamine, because jeffamines are industrial polymers produced by the Huntsman Society, and it's a polyetheramine with propylene oxide group and ethylene oxide group and amine terminal groups. This amine is important for us because it's a function that we use to graft such polymer onto our polysaccharide. You can see this polyetheramine at a cloud point about 15 degrees, and we can graft that on a carboxylate function to obtain COM structure, or on aldehydic function of the reducing sugar to obtain block structure. The grafting is now well known and good control in our laboratory. We can incorporate amine on carboxylic groups using carbodiimide activator, and then we obtain our COM structure. Here is the example of a carboxymethyl purulane graft with our jeffamine with a rate of substitution degree of 0.2. The yield is about 70%. We have largely worked on such system with my colleague Georgeta Mocanu in the Petroponi Institute in Iași. I want to think about Georgeta actually. So what is the behavior of such polymers in the entanglement semi-dilute regime? We can see here the behavior at the concentration of 5% in a buffer. Then we measure the G prime and the G double prime as a function of temperature. And you can see that for room or below room temperature, we have a mainly viscous system. So this is the characteristic of a viscous organization and solution properties. And when we increase the temperature, you can see a large increase of the elastic modulus, and also but less of the viscous modulus. And then you have a crossing point of the modulus. After that, the elastic modulus become the more important one. And then here you have the formation of a gel. So here you can see that such system can permit to control the transition of a solution to a gel, a weak gel transition. We also follow this transition by macro DSC, and you can see that effectively at this transition you have transfer of energy that we can measure and quantify by micro DSC. So after that, we imagine a system that can behave as the previous one, but without chemistry. And to imagine that, we imagine in the first time the electro association of a polyanionic polysaccharide and a polycationic LCST group. The main idea is the following, and perhaps I can put on like that. You can see? Yes? It's okay?
Yes, it's okay.
Okay. So the main idea is to fix by electrostatic association the thermosensitive groups onto the anionic moieties of the polysaccharides. So we expect that. And after that, we want to see if there is a thermo association with increase of temperature. So that we want is the formation of association with increase of temperature, but we also expect thermo association giving rise to mixed micelle and also unical micelle. So we will see if it works. Yes, it works. So to obtain success with such system, we have firstly to fix the pH in order to have the maximum of ionization of both anionic polysaccharide and cationic thermosensitive polymers. And at the pH of 7, we reach the maximum of ionization for both. So we are in the best condition to electro association. We have also seen that the electro association have to be done at a temperature lower than the temperature of association of the thermosensitive polymer. It means at 4 degrees, for example. And in this condition, you can have electro association. And when you increase the temperature, you can see that we observe a sol gel transition here. So it was a good result. Then we have wanted to see if there is a reversibility. I haven't said that, but with a chemically graphed thermosensitive polymer, we have a very good reversibility. It means that if we decrease the temperature, we return to the sol state. When we do that with electro-associated system, you see we increase the temperature, so we have the gelation of the system. And when we decrease the temperature, we stay on an elastic property. So we stay at the gel state. In fact, to have the reversibility, you have to decrease the temperature during a long time. So you have to keep the system for 1 day at 4 degrees Celsius, and then you can return to the initial state and have a new time, the gelation, if you increase the temperature. So our hypothesis is the formation of a cooperative system. I will try to show you what we imagine for this system. So it begins like I showed you just before. So with electro-association, then you have this cooperative system. You see, the cationic moieties can move onto the backbone to associate themselves and reinforce some junction zone. here. So you have probably a system with more structured hydrophobic association. And then you have to return to low temperature to disassociate the system, like that. I want also to see you quickly, very quickly, the behavior of block polymer with thermo-associated groups. So here we use one more time the reducing sugar here to fix the thermosensitive group on the polysaccharide one. And that we expect is the formation of micelle when we increase the temperature, and also perhaps the possibility to stabilize some emulsion or suspension as a function of temperature with a temperature control. So the modification is the following. We have the gefamine that I presented before, and here's the pullulan. Here you have the reducing sugar with the aldehydic function, and DMSO with cyanoborohydride of sodium. We can fix these amine groups on the aldehydic one and to form an amine here. So this reaction works. Not very well, but it works. The reaction is long, and we can elaborate some blocky system. So here, Sabrina Belbekouche, who works on this thesis on such system, varied the length of the polysaccharidic moieties. Here, from this length to the maltodextrose unit. Of course, when you change the length of polysaccharide moieties, you change also the hydrophilic lipophilic balance. More longer is the polysaccharidic hydrophilic groups, more is the HLB. As a first result, we follow the turbidity with adsorbents at 400 nanometers, and then you can see the behavior of the series of block system of copolymer, and you can see that the more hydrophilic one do not evidence any change in the adsorbents. But when you decrease the HLB, I mean, you decrease the length of polysaccharide moieties, you can see the apparition of the turbidity, and this turbidity appears for temperature which are lower as the HLB decrease. So it's a good indication of the thermo association of such copolymer. If you put this system in an emulsion system, in a water octanol, for example, you can see that with the precursor pullulan, which is only hydrophilic, there is no stabilization, and you have the fast separation very quickly. But if you put some copolymer with thermo association properties, you can see that we are able to stabilize an emulsion here. Sabrina wants to see if the stabilization can be controlled in the time and for different temperature. So she tried to see the stabilization time of water octanol emission in the presence of copolymer with the variation of HLB. And for different temperature, at low temperature, obviously, our polymer is purely hydrophilic. So there is no stabilization, and you can see in the green, the stabilization is only for 15 minutes. But if you increase the temperature and you are above the association temperature, you can observe a good stabilization of the emulsion, and this stabilization is better as the HLB is lower, obviously. So it was a good result, and that means we can control the stabilization of an emulsion with the temperature parameter. I don't know the time here. Perhaps I can show you just few last results on
get 50 minutes.
50 minutes, yes, it was largely sufficient. Thank you. So just for example of the control of chemical hydrogel obtained by enzymatic catalysis. So the idea begins of a biomimetic approach. Perhaps you know that when you take some cereal feeds and you put them in hot water, you obtain an increase of viscosity of the solution. That means there is a mucilage which solubilize in the water and give viscosity. This mucilage are mainly arabinoxylan polysaccharide. And when we observe in detail this polysaccharide, you can see that there is some ferric acid moieties that are grafted on such arabinoxylan. And in the nature, there is also some enzyme. As an example, an enzyme which is called lacase. And lacase can dimerize the ferric moieties, which give rise to a chemical cross-linking of arabinoxylan. This is very important for the feed and for the plant because that permits the retention of water and the growing of the plant. So we have the idea to imagine the grafting of ferric acid on different polysaccharides. So it was the thesis of Adi Hadrich, which has been defended 2 years ago. And Adi grafts ferric acid on non-ionic polysaccharides, pullulan, for example, and anionic polysaccharide, and we have tried carboxymethylpullulan and also hyaluronan. So Adi did a very good work because he success to graft the ferric acid on both type of polymer. Just today, I want to show you only the grafting of hyaluronan, of the ferric acid. We use adipic acid dihydrazide as intermediate, and then you can see we take the ferric acid with EDH. We condense the amine onto the acidic moieties, and then we obtain this molecules with amine terminal group. Then we can incorporate, as I shown you just before, on carboxylic group of the polysaccharide. Then we obtain ferric acid graft polysaccharide. We obtain different derivative with various amount of ferric acid content. Just a result of gelation of such system. Here you can see the modified polymer in the presence of lacase enzyme in a buffer at 5.5 pH at 25 degrees. And we put the lacase into the solution and we measure the elastic and viscous modulus. At the beginning of the reaction, you have a viscous state with G double primes much important than the G prime. And then, as a function of time, you can see the gelation with an increase of the G prime, and then you obtain a clear hydrogel here, which is a chemical hydrogel. Of course, you can control the time and the value of the elastic modulus with the control of the ferric acid incorporation rate and also with the amount of lacase. So, as a conclusion of such hydrogel, we have found that on neutral one, we can't graft a lot of ferric acid because the polymer becomes quickly insoluble in water. And the flexibility of the change needs a lot of polymer to obtain the sufficient concentration for hydrogel elaboration. For anionic one, the association is not so important because of the presence of charge, and on hyaluronic acid, it leads to very interesting biological application. We can also say that ferric acid has a lot of phenolic structure, are antioxidant and antibacterial. We have shown that, and there is also no cytotoxicity in the system. So I have finished my talk. I want to thank all the students who have participated to all of this result. Also my colleagues, and also the collaboration, and you can recognize here a lot of collaboration with Romanian colleagues. I want to thank you for your attention, and mulțumesc.
Thank you very much for your very nice presentation. If you agree, we open this for discussions. Maybe there are some questions from the attenders. So please, want to ask a question to Professor Luc Picton.
Félicitations, Luc. Tu as présenté un très bon travail. Et je voudrais te demander, je me permets de parler en français, parce que I will speak in French because I want to say to our young colleagues that it's very important to know English. It's obviously very important, but also it's very good to know other languages also. It's very useful in research activity. Donc, je vais te demander si tu as essayé d'utiliser les systèmes qui changent avec la température pour immobiliser des enzymes et de passer ainsi de la catalyse homogène en catalyse hétérogène, parce que tu as une solution, c'est catalyse homogène, et puis quand tu passes dans l'hydrogel, tu as une catalyse hétérogène. Et donc, tu peux changer et faire les réactions en fonction de lieu où la réaction se passe. Est-ce que tu as essayé d'utiliser cette possibilité de tes systèmes?
Je réponds en français, André, ou je ne sais pas.
You may answer in English because many young people don't know French.
So you asked me, André, if I try to graph some enzyme and with sensitive polymer to change from a homogeneous to heterogeneous system and to organize the enzymatic catalysis in heterogeneous states. It's a really interesting idea. Unfortunately, we have not tried that, for instance, but I retain the idea. It's a very, very elegant approach.
Yes, it's very important because enzymes are expensive, and covalent immobilization is a way to don't lose to enzyme, because normally when after the reaction you are losing to enzyme, but after immobilization the enzyme it's bonded to a polymer and it's possible to recover the enzyme. But so this also gives the possibility to apply homogeneous catalysis or heterogeneous catalysis depending of the reaction which you intend to catalyze with your enzyme. So if you agree, we can do such cooperation in the future, because we are also working on enzyme immobilization, but I didn't had till now a system which is able to pass from homogeneous to heterogeneous catalysis.
Yeah, I think it's a really interesting idea, because there is also a problem with enzyme, mainly for a biological or biomedical application, because enzyme are protein, and oftenly application needs to avoid enzyme after the transformation. So it should be a nice idea to fix the enzyme and to avoid the enzyme after the transformation.
Yes, I hope to speak more about this in October, because you know you are also invited in PrioChem symposium, and I hope that you will be able to come here and to speak in face about these ideas.
Andre, we have a nice future project together, so I think.
Okay. Thank you, Luc.
Merci beaucoup, André.
If there are some other questions, please. So thank you very, very much, Professor Luc Picton, for your very nice presentation. And we wait for you in autumn, in the end of October at PrioChem, because you were so glad to accept our invitation to participate to PrioChem, and send to your collaborators our salute and all the good wishes for them and success.
You're welcome. It will be a great pleasure to be here, and I hope in Bucharest and not behind a computer.
Let's hope to have again a face-to-face.
Yes. Okay, thank you very much. Bye.
Thank you. And now we shall pass to the presentations made by our participants to the workshop. And I invite Dr. Sanda Velea to chair this section for the second day of NeXT-Chem workshop. Please, Dr. Velea. I don't know if she's connected. Yes, she's connected. Or maybe Dr. Fiorescu, Radu, please, can you start with the first presentation? To invite the first.
Yes, thank you. Yes, until our colleague Sanda Velea will be online, I will present the following colleagues. So the first presentation is made by Diana Ioana Buliga with the title Phycocyanin, a possible solution for natural blue pigments. Diana, please.
Și n-am programul, dați-mi mie programul, că nu știu ce să prezint. Nu știu lucrările, că îmi ziceți să conduc eu sesiunea.
We don't have sound. Diana?
Yes, good morning. There is a recording made in the PowerPoint. I checked after I sent it to you. I can try and connect from the computer, but I have a connection. I have a problem with the Wi-Fi connection, and currently I am on the cell phone using data. So I'm not sure if it will work.
No problem. Probably our colleague that is running the presentation should check for the sound.
Yes, thank you.
To also share the sound. Alexandru, please.
Sorry, we have a problem with this connection now.
Good morning, my name is Diana Ioana Buliga, and today's presentation is entitled Phycocyanin, a possible solution for natural blue pigments. It is focused on the extraction of phycocyanin from Spirulina plantensis. Spirulina is a microorganism originated from the lagoons of Latin America and Africa. It is a cyanobacteria which presents helical filaments. It has a high tolerance to alkaline pH and grows vigorously in sunlight and at high temperatures. Due to the fact that spirulina does not have cellulose in its structure, it is easily digested and absorbed by organisms. Moreover, the blue-green microalga is an important source of valuable compounds, which is why it is currently used in the functional food and nutraceutical market and can be administered as capsules, tablets, or as powder added in foodstuff. Phycocyanin is a pigment protein complex from the phycobiliprotein family. The phycobiliproteins are composed of 2 subunits typically formed by 8 alpha helices. The protein backbone of the subunits has 1 or 2 linear tetrapyrrole chromophores, which are covalently bound through a thioether bond. For phycocyanin, the chromophore is phycocyanobylene, which has the chemical structure on the upper right corner. Phycocyanin is responsible for the high antioxidant activity of spirulina and for its vibrant blue color. For this reason, spirulina can be a raw material for the obtaining of a substitute to brilliant blue and an environmentally friendly coloring solution. The aim of this study was to establish the best extraction conditions of phycocyanin from Spirulina plantensis in order to utilize it as a natural blue pigment. The extraction of phycocyanin was performed using powdered spirulina purchased from a local market and a 0.1 molar sodium phosphate buffer solution with a pH of 7.5. The ratio between the vegetal material and the solvent was 1 to 15.6, and both conventional and unconventional extraction methods were used. For the conventional extraction, the temperature and the time were varied, while maintaining a constant stirring speed. The time and the sonication mode were varied in the case of the ultrasound-assisted extraction, which was performed using a sonication probe. The stirring speed and the amplitude were constant. After the required amount of time, was separated by centrifugation at 4000 rpm for 15 minutes. Following the separation of the extract, they were analyzed in order to determine the concentration of phycocyanin. Moreover, preliminary studies have been carried out regarding the antioxidant activity using the cupric-reducing antioxidant capacity assay. Both analyses were performed using the spectrophotometer. The influence of time and temperature on the extracted phycocyanin content for the conventional extraction was studied. At temperatures, such as room temperature and 30 degrees Celsius, the phycocyanin concentration increases with time until a maximum is reached. After this point, the amount of interest compound extracted tends to remain constant. At higher temperatures, the phycocyanin is extracted more rapidly, in less than half the time necessary to obtain the highest extracted content at room temperature. At 40 degrees Celsius, a similar value is obtained. However, working at higher temperature increases the risk of degradation. For the best extraction conditions, the antioxidant activity was computed using the Cuprac assay. A high value of 21.6 milligrams of equivalent Trolux per milliliter of extract was obtained, but this value can vary according to the cultivating and processing conditions. The influence of time and sonication mode on the extracted phycocyanin content in the case of the unconventional extraction was studied. When a sonication duty cycle of 1 second on and 1 second off was used, a higher amount of the targeted compound was extracted. A maximum was reached after 15 minutes. Following this point, a decrease due to phycocyanin degradation occurs. For the continuous mode, lower concentrations were obtained because the intense sonication can lead to the degradation of the targeted compound. In this case, the maximum amount of phycocyanin was reached after 20 minutes, and it had a value with 15% less than the one for the previous case. For a more clear view, the influence of the extraction type on the extracted phycocyanin was also studied. For the conventional case, higher temperatures ensure a quicker extraction of the interest compound, reducing the time by more than half. from 270 minutes to 120 minutes. The ultrasound-assisted extraction provides up to 20% higher extraction yields in a much shorter time span. However, an intense sonication of the extraction mixture can result in the degradation of phycocyanin. To conclude the presentation, it is very important to remember that spirulina is an important functional food with a possible future in the dyeing industry. The aim of this study was to establish the best extraction conditions for phycocyanin from powdered spirulina. The extraction was highly efficient using the ultrasound-assisted extraction, where important quantities were extracted in shorter periods of time than compared with the conventional extraction, due to a better mass transfer. However, for a long extraction time, the targeted compound degradation occurred. For the conventional extraction, the best extraction conditions were at 120 minutes and 40 degrees Celsius, while for the ultrasound-assisted extractions, the best conditions were at 15 minutes using a 30% amplitude and a duty cycle of 1 second on and 1 second off. Here is a short bibliography. Thank you very much for your attention, and I am more than happy to address any questions you might have.
So, sorry for my sonorization. If my colleague from NeXT-Chem team have some questions, because we have the same activity in the field of microalgae, especially for extract from porphyridium to obtain these products, very interesting. My young colleague, if you have some problem for discussion. So we can talk the next period about real cooperation between our collective to complete this research in this field. It's very interesting, your presentation, and will be for our interest to develop a new cooperation with our team. Yes, thank you very much for your presentation.
Thank you.
Thank you. And now, the next presentation, I ask my colleague, my young colleague Naomi, to make the next presentation for this session.
Yes, good morning. The next presentation belongs to Maria Cristina Ghețu from University of Bucharest, and it's called Cascade Biotransformation of Monoterpenes using co-immobilized enzymes. So, Maria, you can share the screen.
Okay, thank you.
Thank you too.
Can you see the presentation?
Yes, please.
Okay. My name is Maria Ghețu, and I'm here to tell you about how we managed to create a biocatalyst based on enzyme co-immobilization with applications for monoterpenes valorizations. Here is the outline of the presentation. I'm going to start with a short introduction, followed by the experimental setup, short process optimization, few applications, and of course, conclusions. As an introduction, I would like to tell you something about monoenzymatic processes, which represent a combination of several enzymatic reactions. A good example are metabolic processes. Then cascade reactions that in our case represent multi-enzymatic processes comprised of at least 2 consecutive reactions, and they have the great advantage of not needing of isolation of the intermediates. Then the lipases and epoxide hydrolases, which are the enzymes we employed in our study. The lipases catalyzed reactions on ester bonds and have a higher preference for water-insoluble substrates. And the epoxide hydrolase, which catalyzed the hydrolytic ring opening of the epoxide, resulting in the corresponding vicinal diols. And of course, our substrates, the monoterpenes, which are a class of natural compounds widely occurring in plants. And it's worth mentioning that their oxygenated derivatives are used as flavor and fragrances for centuries. Therefore, we propose a bienzymatic 2-step cascade process that converts the monoterpenes into flavor and fragrance products. Here we have the reactions for limonene. And in the first step, the lipase indirectly catalyzes the transformation by oxidating the fatty acid in our system, in our case, the octanoic acid, using hydrogen peroxide to octanoic peracid. And this formed peracid then converts limonene into limonene oxide. And in the second step, the hydrolase directs the transformation toward the formation of the S isomer of the limonene diol. For the experimental, we start by using Novozyme, which is lipase B from Candida antarctica immobilized in hydrophobic beads, and it's commercially available. And CH55, which is a thermophilic hydrolase, which was provided to us in the form of an enzymatic solution by Dr. Monti. The immobilization, it's pretty straightforward. It's based on the absorption of the CH55 hydrolase on the surface of the acrylic beads, which already contain the absorbed lipase. And the preparation, it's pretty simple. We just added few microliters of the enzymatic solution of CH55 hydrolase over few milligrams of Novozyme beads. We also used gas chromatography as our method of analysis. We used GC with flame ionization detection to properly determine the conversion of the substrate, the limonene oxide yield, and the diol yield, and also the enantiomeric excess. And in order to properly quantify our results, we constructed calibration curves. And we also used GCMS to identify our products. We are going to start with the process optimization using a monoenzymatic system containing only the lipase, and we tested few different possible candidates for our lipase. More exactly, few different free enzymes versus immobilized lipases. All of them are commercially available. And overall, it can be observed that the immobilized enzymes perform better. And the best results were obtained for the Novozyme. Therefore, we use Novozyme as our possible future candidate. Still working on a monoenzymatic system, we varied the reaction temperature in order to see its influence and if we can increase further the conversion. We varied the temperature between 25 and 50 degrees Celsius. And overall, the best results were obtained for a temperature of 40 degrees Celsius. However, as it can be observed, as the temperature increases, the limonene oxide yield decreases, which was to be expected due to higher energy input. Therefore, we testing different acid trapping regions in order to increase the epoxidation yield. And from all the trapping regions, the best results were obtained for trisodium citrate. Therefore, we increased the reaction temperature to 40 degrees Celsius and added trisodium citrate in our system. And moving on to our bioenzymatic system, we compared our biocomposite containing both enzymes immobilized on the acrylic beads versus the immobilized lipase combined with free hydrolase. And overall, we managed to obtain the best results for the combination of Novozyme beads with immobilized CH55 with a high conversion and obtaining almost enantiopure diols. Therefore, we took this biocomposite and tested few applications such as recycling. We tested our material for up to 3 consecutive reaction cycles. The material was washed after each cycle in order to remove the possible remaining substances. And overall, we obtained the average conversion degree of around 40%. And the formation of the S isomer of limonene 1,2-diol is favored after each cycle. And another possible application for our material is the bioconversion of alpha-phelandrine to its corresponding vicinal diols. Here we have the mechanism. And here are the results, which are very promising. Therefore, in conclusion, we developed the biocomposite based on the absorption of CH55 hydrolase on the surface of commercially available Novozyme. Our biocomposite showed outstanding performances in converting limonene compared to Novozyme beads alone or coupled with hydrolase in solution. Our biocomposite managed to conserve most of its activity after each cycle. And we tested our material for alpha-phelandrine transformation, and we obtained promising results. Thank you for your attention.
Thank you, Professor, for your very interesting presentation, and very interesting for maybe for a next cooperation between our team, because we have a long and very good cooperation with the team of Professor Călinescu. Thank you. If our colleagues have questions about your presentation, please. I understand no questions. Thank you for your very interesting presentation. We rest to discuss about potential cooperation with our team. Thank you very much.
Thank you.
Let me introduce now for this next 3 presentation, because it's 3 or 4 sorry, 4 presentations. The next presentation is from our young colleague. The first presentation will be done from Grigore Pshenovsky. It's about our complex project about the new and original system integrated in biogas microalgae from our complex project. It's some very interesting results of our cooperation. Please, Grigore, present your recent
I think
research.
it's okay. You can see my presentation.
Yes, please.
Okay. Hello, ladies and gentlemen. My name is Grigore.
Louder, please.
Today my topic is about treatment of liquid digestate from anaerobic digestion by electrocoagulation, flocculation, batch mode.
Altceva, băgasem în altă parte.
Just a second, I can't switch the. Just a second, I have some troubles. Okay, so introduction, anaerobic digestion in multi-stage biological process using organic waste management practice, which simultaneously limits the waste and produces biogas, a clean and important biofuel for energy generation. But however, a big disadvantage is the large amount of digestate resulted as a byproduct from this process, and the treatment is required because it's a high amount of carbon, it's a high amount of nitrogen and phosphorus. That's why it's over the limits. And we can't throw this digestate. So the study of this topic is about the performance of the electrocoagulation flocculation process for treating liquid digested resulted from a pilot scale system of anaerobic digestion of biomass mixtures and farm waste as potatoes, beets, carrots, and stuff like that, combined with the wastements from animals, poultry and pig manure. And by reducing the content of phosphorus and nitrogen from liquid digestate. So electrocoagulation flocculation in a batch system was studied as a treatment for turbidity reduction of nitrogen, phosphorus, carbon decreased by using as a process factors electrode distance and applied voltage onto the electrodes, as well as the time. As materials, the experiments were performed in laboratory scale installation for biogas production from indigenous feedstock. As you can see, the image is this pilot installation that have a volume of 30 liters. And in the table, in the right table, you can see the characteristic of the digestate. Commonly, it's a dark brown color with a high value of turbidity, carbon, nitrogen, phosphorus, and others. So in the next stage, we extracted this digestate, put into cylinders and overnight to precipitate. And this image we'll discuss later. We made 14 experiments. So experimental design, it's a total of 14 electrocoagulation experiments were performed at different values of the main parameters coordinate the process as follows. As the distance between the electrodes during the experiments was set at 1, 2, 3, 5, 6 centimeters respectively. The electrodes were subjected to two different potentials, so 6 volts and 12 volts. The experiments were performed at different times, some lasting 30 minutes, 60 and 90. In figure A, you can see the scheme of the batch electrocoagulation reactor, and the second figure B, it's a voltage source. For each experiment, the temperature, pH, and conductivity of the solution subjected to the electrocoagulation process were monitored by measurement every 10 minutes. The evolution of the main parameters controlled, such as temperature, pH, and conductivity during the experiment, is presented in table 2. So every 10 minutes with measurement, the main parameters, temperature, pH, and conductivity of the solution, of the digestate. So to explain the main parameters, with every increase of temperature, pH is also increasing, but conductivity is going low because these salts from the solution are attracted to the electrodes made from aluminum. Analysis method, the efficacy of the electrocoagulation process was evaluated according to the degree of decrease of the following parameters: color, turbidity, total organic carbon, and nutrients such as nitrogen and phosphorus. The next parameter was the temperature, pH, and conductivity were measured each 10 minutes during the experiment using a WT2 multiline P3 pH. It's simple to handle, and that's the best solution because it measures the pH and conductivity of the solution. The next step is to measure the turbidity using a sensor from reactor, photobioreactor. It's only a sensor that we use to measure the turbidity. The next one is a total solid, and total solid was gravitationally measured after overdrying overnight at 105 Celsius until constant weight. It was just dried, and the solid content is the total solid content. And the next parameters as phosphor, nitrogen, and carbon was determined with the same apparatus. It's called the multi-NC 2100 analytic GENA. It's made, it's a dispositive made from Germany, and it's a best solution to determine the parameters of the phosphor, nitrogen, and carbon content. As a result, in synthetic results of the experiment, in all of these 14 experiments, the best results we can see in experiment number 1, 2, and 10. So the OD, it's present the best result in experiment number 1 and 2. Also, the best result of nitrogen decrease is in experiment number 2. The decrease of the phosphorus is in experiment number 9, and the carbon, the best is number 10. As we come back to the first picture of the experiments, the best results you can see. Also, the clean solution of the water in 1 and 2, but the best results of carbon, that is also very important, is experiment number 10. So to make a more clean results, the every parameters was generated into percent. So the first, so the best decrease also it was 96% of the turbidity. Nitrogen result was 69, phosphorus 73, and the carbon was 76. The next result we generated into graphic. Also the influence of the aluminum anodic consumption of turbidity on the nitrogen, phosphorus, and the carbon. But to explain what's happening with the every increasing of the voltage, the results are going more better. So we increase the voltage, the distance between electrodes. So with the every increasing, the results are coming better. But there is a disadvantage. It needs more consumption of the energy. So this is a disadvantage. Let's see, the more electricity, the more cost, and it's kind of bad. So as conclusion, electrocoagulation flocculation process in a batch system was studied as a treatment for turbidity reduction of phosphorus, nitrogen, carbon decrease by using a process factors, electrode distance, and applied voltage onto the electrode, as well the time. Applied voltage and especially the process time present a positive influence on turbidity, but the electrode distance and the combined interaction between electrode distance negatively affect the parameter. To explain again, a small distance between electrodes is good. If we increase more this distance is going to bad results. So The maximum turbidity reduction, about 89%, was obtained on time 90 minutes, and applied voltage of 12 volts, and the electrode distance is kind of medium, 3 centimeters. And liquid digester can also be used for the further application. The benefit from is high content of nutrients, such as microalgae cultivation or hydroponic cultures. It has been attempted to grow microalgae in this waste stream, as microalgae can consume the excess of carbon, nitrogen, and phosphorus. So also to explain, if we decrease this content of nitrogen, phosphorus, and carbon, it's also the best way to use in irrigation or for fertilization for plants and other culture and other agriculture. So thank you again for the attention. Thank you for my colleagues for this experiment, for the help, and I'm very glad. Thank you.
Thank you, Grigore. If you have the questions about this presentation, I want to talk just not for use this device in the treatment of liquid digestate, but for the harvesting of microalgae, because you know our project it's about biogas installation and about the microalgae culture, because this microalgae is used for decontaminate the liquid digestate from biogas installation. And in the same time, the big problem was harvesting of microalgae. You know, this is dimension is colloidal dimension of this microalgae make difficult this process for sedimentation, settling, and harvesting this type of products. Is a very important device because it's the original concept and handmade it in our institute. If you have the questions about these results, we can ask my colleague, please. Thank you, Grigore. We haven't questions. Our colleague haven't questions. And in this situation, we can ask our colleague Alexandru Vlaicu to present your results in the work about intensification technique for biodiesel production. Please, Alex.
Good morning, my name is Vlaicu Alexandru, and I will be giving a presentation about assisted biodiesel production in continuous flow systems. For this work, an in-depth comparative study was carried out, focusing on unconventional techniques, specifically hydrodynamic cavitation, ultrasonic cavitation, and microwaves, for intensifying biodiesel synthesis, while also taking into account the energy efficiency of said processes. Although process intensification techniques are well documented, especially for batch systems, our work has been centered around the viability of using continuous flow systems to produce fuel for small and medium farms, where such equipment could provide sufficient fuel for on-site agriculture machinery using renewable or waste green materials which are produced on-site. Fatty acid methyl esters through hydrodynamic cavitation were synthesized using a reactor consisting of two symmetrical plates facing each other, one stationary, the stator, and one connected to an engine, the rotor. Due to the specific geometry of the plates, the small indentures on each of the plate and the very small space between them, strong enough forces are exerted on the fluid, leading to the formation of bubbles, which collapse through cavitation. For ultrasonic cavitation, two types of equipment were used. The first with a probe system, which is placed inside the reactor through the top section. The reagents are mixed and pumped into the cylindrical shaped reactor through the bottom. The temperature is monitored in the outlet and maintained with the help of a thermostated cooling agent, which passes through the exterior jacket of the reactor. The placement of the product's outlet allows for a reaction volume of 60 milliliters to be achieved. The second type of ultrasonic processor which was used is the clampon, in which ultrasonic waves are generated over the full length of the metallic pipe. The reactor itself consists of a glass tube with the reaction mixture passing through it, while the cooling agent passes through the space between the glass and the metallic tube. Sufficient mixing of the reactants wasn't assured by the clampon system, requiring the addition of a premixer. For the microwave-assisted process, the glass reactor is placed inside the waveguide of a monomode applicator. The reactants are pumped towards the base of the reactor, and the products exit through the top once the reactor is filled. The microwave power provided to the system is automatically and continuously adapted to maintain the chosen temperature, which is monitored in the outlet, while being simultaneously recorded by the software. For every intensification technique, as well as conventional processes, various concentrations of sodium hydroxide dissolved in methanol were used. In the transesterification of sunflower oil, the molar ratio of methanol to oil which was used was 6:1, and all the catalyst concentrations were calculated based on the amount of oil in the reaction. The beneficial effect of using intensification techniques is especially visible for lower concentrations of catalyst. As the catalyst concentration increases, the gap between the assisted and the conventional process decreases in all cases. The probe system showed the best results at an amplitude of 45%, for which the ultrasonic power and the grid power are 70 and 100 watts respectively. Fatty acid methyl esters concentrations above the standard requirement for biodiesel were achieved in under 30 seconds, this also being the most energy efficient intensification technique. The results which were obtained at the amplitude of 70% showed that this type of reactor is limited in terms of amount of ultrasonic energy, and would suggest that there may be an optimum power for this type of reactor. For the clampon system, we can see that simply passing the reactants through the premixer is insufficient to achieve a high conversion rate. A percentage of fatty acid methyl ester of only 76% was achieved in the premixer after 3 minutes. Although concentrations higher than the standard required for biodiesel are easily achieved, the required reaction times and the specific energy consumption for this type of device are slightly higher than that of the probe system. We can see when comparing amplitudes that there are no significant differences between the 40% and 60% amplitude for this type of reactor. Results indicate that microwave reactor uses more energy than an ultrasonic reactor. This gives us some indication of relative energy requirements for laboratory equipment. Although microwave and ultrasonic systems are somewhat different when larger scale instruments are considered. The microwave and hydrodynamic cavitation assisted processes are the least efficient in terms of specific energy consumption out of the studied processes. Although the hydrodynamic cavitation reactor wasn't able to achieve the minimum Fama concentration in this conformation, other forms of hydrodynamic reactors can be used in these processes and are much easier to be applied at industrial scale. Therefore, it still remains one of the more versatile types of intensification techniques which can be used. Continuous low acidity translocation processes were developed and compared in terms of fatty acid methyl ester concentration and specific energy consumption. These types of process intensification methods are beneficial for the production of fatty acid methyl esters, with the acoustic cavitation systems being the most promising for small-scale production of biodiesel. The improvements over the conventional process were more likely due to the higher mass transfer between the two non-miscible reactants via the formation and the collapse of asymmetric cavitation bubbles. Thank you for your attention, and if there are any questions. Good morning, my name is Luc Alexandru, and I will be giving a presentation about
So, thank you, Alex. We haven't questions about this very interesting subject about biofuels, but it's about new technique in this process, and it's very interesting for this. If you haven't questions, we can talk about the next presentation. It's a young lady from our team, Luisa Merzi. It's about catalyzed. We use catalyzed in this project in the field of hydrotreating of solid digestate and for another use. Thank you, Alex. We talk about the next presentation.
Good morning, everyone. Yes. The aim of this presentation is bifunctional aluminum oxide-based catalyst for the hydrotreating hydrocracking reaction of bio-oil. Some of the topics that I will talk about are the catalysts that we use for hydrotreating hydrocracking reaction, their synthesis and characterization, and the hydrotreating hydrocracking installation. Hydrotreating is one of the major processes for the cleaning of petroleum fractions from impurities such as sulfur, nitrogen, oxycompounds, chlorocompounds, aromatics, waxes, and metals using hydrogen. Hydrocracking is a catalytic hydrogenation process in which high molecular weight raw materials are transformed and hydrogenated into low molecular weight products. Hydrotreating units are needed in the refinery to clean the flows of materials harmful to catalysts. That is why they are located before the hydrocracking installation. Catalysts used in hydrocracking are bifunctional, composed of a metallic part that promotes hydrogenation and an acid part that promotes cracking. The acids used in our case is alumina. Hydrotreating removes impurities in the feed, such as sulfur, nitrogen, and metals. Cracking will break bonds, and the resulting unsaturated products are consequently hydrogenated into stable compounds. The catalysts were prepared by an impregnation method. The impregnation solution was produced by dissolving the salts of the catalytic precursors in the solvent. In order to obtain good homogeneity of the metals deposited on the support, successive impregnation and drying of the catalyst were performed. Drying has been carried out at room temperature for 24 hours and in an air recirculation oven at 160 degrees Celsius for 4 hours. After final drying, the catalysts were calcinated in an oven for 6 hours at 450 degrees Celsius. For the hydrotreating and hydrocracking process, catalysts used are porous matrix of aluminum impregnated with combination of some transition metals. The microporosity of catalysts was evaluated from beta analysis, recorded at 77.4 kelvins. The nitrogen absorption-desorption isotherms showed a type H3 isotherm. The shape of the bed curve is characteristic of mesoporous structures. The desorption and absorption branches meet at some intermediate relative pressure, giving a closed hysteresis loop. The textural characteristics of the bifunctional catalysts indicate obtaining a mesoporous structure, which is confirmed by the specific surface area, the total pore volume, and the average pore diameter. Hydrotreating hydrocracking reactions were performed in a pilot plant in a continuous flow system consisting of a fixed bed catalytic tubular reactor with a descending flow of liquid and gas produced by PR Instrument Company USA. As we can see in the figure 5, the hydrogen and the liquid enter the reactor through the tap. The tubular reactor is provided with 3 independent heating zones. The catalysts are positioned in the appropriate area using the metal spacers and the required volume of glass bolt. The reaction products are cooled in a double pipe heat exchanger and end up in a gas-liquid separator. The liquid phase is collected and analyzed, and the gas phase is released under a controlled atmosphere. The first stage of treating hydrocracking technology consists in loading the tubular reactor with a capacity of 500 cubic centimeters with 2 hydrotreating catalysts and the hydrocracking catalyst corresponding to each heating area. For the first hydrotreating process, the catalysts used are copper palladium deposit on aluminum at 180 to 220 degrees Celsius. In the second hydrotreating process, we used also a copper palladium-based catalyst at a temperature range of 217 to 320 degrees Celsius. And the lower heating area was filled with nickel platinum-based catalyst for the hydrocracking process. The performance of the hydrotreating hydrocracking process has been assessed by determining the liquid phase efficiency and the composition of the resulting liquid product. The composition of the final product resulting in the pilot plant by hydrotreating hydrocracking processes of the conditioned pyrolysis bio oil was determined by gas chromatographic analysis. In conclusion, the synthesized bifunctional catalysts were prepared via an impregnation method, and it was proven to be highly efficient. The performances of the hydrotreating hydrocracking process were evaluated by determining the yield in the liquid phase, the acidity index, and the unsaturation index, as well as the calorific value of the liquid product obtained. The chromatographic analysis reveals a reduction in oxygen content and an increase in linear branch or cyclic saturated hydrocarbons content. Thank you for your attention.
Thank you, Lisa. If there is comments or questions about this presentation, if you have a comment or questions, I ask my colleague Alex and Naomi to make the next presentation. Please, Naomi. You.
Yes, next Sabina Ion from University of Bucharest will talk about grafting natural lignin with aniline using biocatalytic approach. So yes, you can share your screen and let's hear it. Good luck.
Hello, can you see the screen?
Yes.
Thank you. Good morning. My name is Sabina Ion. I'm a PhD student at Faculty of Chemistry, University of Bucharest. And today I'm going to present you a very interesting topic regarding a component of biomass, which is lignin. But first, did you know, I have a technical problem, sorry. Did you know that annually over 50 million tons of lignin are produced as a byproduct of the paper industry, but only 2% from this amount is used in the plastic industry? Also, lignin and wood fibers from the pulp and paper industry are used to create bio-based plastics and biocomposites. Lignin was mentioned for the first time by Kendall in the 19th century as lignum, which means wood. It is a complex heterogeneous biopolymer with a 3D structure based on the phenylpropane units called monolignols, with or without one or two methoxy groups in ortho positions. The general perception of lignin is that of a renewable resource derived from three monolignols, namely paracumaril alcohol, coniferyl alcohol, and synapyl alcohol, in different proportions depending on the plant species, climate, or geographic area. An important aspect of lignin represents its properties. It is a green polymer insoluble in water, which transports liquids throughout the plant and provides resistance and rigidity to the cell walls. Moreover, it is an elastic polymer with thermic and mechanical resistance. Several important applications of lignin include chemicals such as phenols, active carbon, binders, or dispersants, but more important, lignin can be used in the energy field as electricity, heat, solid fuels, ethanol, or bio oil. So the main purpose of this subject was the derivatization of lignin using grafting bioprocess involving amino groups based on the biocatalytic oxypolymerization with peroxidase enzyme. This process involves a radical mechanism which includes an electronic transfer between the enzyme and oxidation reagent, generating unstable enzyme forms, substrate radicals, and in the end generating the grafting polymer. There are four types of peroxidases used in the grafting process of lignin. PADA 2,1-B, BB8, which were modified in the lab of Professor Alcalde from Spain on the hydrophobic part of the protein in order to improve the enzymatic activity, stability, and the robustness of the protein material. Also, commercial peroxidase was tested in the system called horseradish peroxidase type 6. The grafting process starts from several types of lignin in methanol, aniline, PBS buffer, peroxidase, and hydrogen peroxide as oxidation reagent at 40 degrees for 24 hours and stirring. The obtained reaction mixture was separated by centrifugation into two phases, a solid phase and a liquid phase. And different techniques were used in order to characterize the polymers. So the used techniques were UV spectrometry, Follin-Ciocălteau analysis, Fourier transform infrared spectroscopy, nuclear magnetic resonance, temperature programmed desorption of ammonia and carbon dioxide, thermogravimetric analysis, scanning electron microscopy, and conductivity. First of all, the enzyme screening evidenced that horseradish peroxidase exhibited the highest catalytic capacity of aniline insertion into Clarkson lignin, followed by alkali lignin, as you can see in figure B. More than that, 11% of peroxidase enzyme was the optimum concentration. The descendant trend from 22% concentration of peroxidase could be explained based on the enzyme agglomeration and the blockage of the catalytic sites. Follin-Ciocălteau measurements on the original engrafted polymers evidence a lower concentration of free OH hydroxyl groups on the grafted polymers due to their participation in the grafting bioprocess. FTIR analysis of Clarkson lignin and grafting Clarkson lignin with aniline presented differences in the spectra, and new bands specific to amide and amine groups appeared, which means a confirmation of the grafting bioprocess. Moreover, chemical shifts from proton spectra of original and grafting polymers confirm also the presence of a new signal at 5.01, 5.99 ppm, corresponding to the generation of protons from polyaniline. The temperature-programmed desorption of ammonia and carbon dioxide results presented a higher basicity and a lower acidity for graphic lignin in comparison to the original one. So this is also another confirmation of the successful process. Using a thermogravimetric analysis, we were able to demonstrate the increased stability of the graphic lignin introduced by the bioprocess. More than that, SEM images of original and grafted polymers evidence a smooth surface for parent lignopolymers and the roughened morphology for the aminographic lignopolymers. The differences were correlated to the type of lignin used in the process. So the graphic lignin exhibited a specific semiconductor property due to the variation of free hydroxyl groups. In conclusion, a new strategy for lignin derivatization was developed. The signal corresponding to the protons from polyaniline were noticed by NMR, and new bands were detected by FTIR after the derivatization process, leading to an amino-derivatized lignin more stable than the original version, with lower conductivity and improved basicity. Some uses of this polymer could be ion exchange reagents, cationic surfactants, flocculants, coagulants, heavy metal absorbents, or even we can use this graphic polymer in order as a support for the protein immobilization. So I would like to thank to my colleagues, to the entire research group, to Professor Vasile Pârvulescu, Professor Mădălina Sândulescu and Bogdan Cojocaru, and thank you all for your attention.
Thank you too for your presentation. If there are questions. Okay, so if there are no questions, thank you too. Let's move to the next presentation. Ioana Popa-Tudor is the next participant with new approaches for obtaining lignin from lignocellulosic biomass. Good luck.
Hello, welcome to my presentation entitled New Approaches for Obtaining Lignin from Lignocellulosic Biomass. Pure spent grains is a byproduct of brewing. There are several stages in the processing of PSG. In order to facilitate subsequent extraction processes, chemical pretreatment of PSG is required. We treated PSG with a mixture of toluene ethanol in a 2:1 mass ratio. The samples are here treated at 90 degrees Celsius under stirring for 6 hours. The determination of the amounts of cellulose, hemicellulose, lignin, and proteins were performed by applying several chemical treatments that were determined in duplicate and triplicate. The main benefits of lignin are protection of hydrophilic components, cellulose and hemicellulose, protection against distortion and compression, gives mechanical strength to plants. Lignin can also have many uses, including lignin-based preservative to protect wood, involvement in the construction of thermal and acoustic insulators, as well as a substitute for raw materials from fossil fuels or other petrochemical resources. A new approach is the use of lignin in the medical field. It is a good precursor in the synthesis of certain drugs with a protective role with the intestinal flora. As a synthesis, a gentle heat treatment was applied, reaching the eutectic point being around a temperature of 50–60 degrees Celsius. As for tests based on betaine hydrochloride, they initially require careful solubilization, which is carried out in the presence of the polar solvent. Then the mixture is further subjected to thermal treatment until complete homogenization. Glycine-based tests required a higher temperature of 80 degrees. Several types of deep eutectic solvents have been synthesized. As it is known, it must contain two main components, hydrogen bond acceptors and hydrogen bond donors, respectively. Choline chloride, betaine hydrochloride, glycine were used as hydrogen bond acceptors, and as hydrogen bond donors, lactic acid, polyethylene glycol, urea in various molar ratios. We present to you through a short demonstration obtaining the eutectic compound. The two components are subjected to heat treatment together and a new homogeneous compound results by the formation of intermolecular hydrogen bonds. The main advantage is represented by a significant drop in temperature. FTIR analysis revealed the presence of hydrogen bonds. In the slide are presented the extraction and solubility processes of lignin by applying DES. The two processes are quantified by two methods, gravimetric and UV-Vis spectrometric methods. Following the calculation, it was found that the highest lignin yield was obtained when the test based on betaine hydrochloride and lactic acid was applied in a molar ratio of 1:5, reaching the value of 69.9%, followed by the solvent with the same content but in a molar ratio of 1:2. Lignin solubilization yields were high for 5 days. The highest yield was recorded when the quaternary DES was applied, followed by the ternary DES, both with polyethylene glycol composition, which led to a slight increase in lignin solubilization. Surface tension and density were also determined. There is a close correlation between the surface tension of a solvent and its density. Compared to water, these have a significantly higher density and lower surface tension due to the formation of hydrogen bonds. As conclusions, we can mention the synthesis of several solvents, the application of characterization techniques, extraction and solubilization tests showed higher yields on certain DES. As prospects for the future, it is necessary to deepen the above-mentioned processes by applying several techniques for characterizing of the PSG. Thank you.
Thank you to Ivana. If there are any comments or questions, please.
Hello,
Okay, thank you, Ivana.
Sorry, thank you for this very professional presentation. And please, let's Alex to make the next presentation for the work. Yes?
Thank you very much.
Alex is not present, yes? Sorry.
I'm just looking for the next presentation, so I'm here.
Alex have a problem.
Okay, so the next presentation belongs to Denisa Ioana Tudor, which is called Activity of Time Essential Oil on Plant Pathogens and Plant Seed Germination. Good luck, Denisa.
Today, I will present the study Activity of Time Essential Oil on Plant Pathogens and Plant Seed Germination. Synthetic pesticides are considered a real problem with increased interest in the development of biodegradable and non-toxic compounds as are essential oils. Essential oils are used in agriculture, medicine, and food industry. They are antimicrobial, antiviral, insecticidal, and antifungal. Due to the fungicidal activity, essential oils are an alternative in combating phytopathogens of agricultural crops while being safe for the environment. The aim of this study was to test antifungal activity of the volatile compounds of time essential oil on 2 strains of phytopathogenic fungi and evaluate the phytotoxic activity of the oil on the germination of plant seed. Testing antifungal activity of time essential oil was tested in vitro on Fusarium graminearum and Isoctonia solani, which were grown on PDA medium at 28 degrees for 5 days. A piece of fungi with growth medium was taken and placed in the center of petri dishes on the PDA, and sterile paper disc was placed on the lid. So, this method was used for testing the volatile compounds of time essential oil. After that, 10 microliters of different concentration of essential time oil was added over it. The phytotoxicity test kit for liquid samples containing 3 species of plant seeds, Sorghum saccharatum, Sinapis alba, and Lepidium sativum, was used to determine of time essential oil at 0.1% concentration. It was evaluated proportion of germinated seeds, roots and shoots length, roots proton pump, seedling total reactive oxygen species by staining with nitrobutadiazolium and stereomicroscope analysis. These measurements were performed with the aid of an image analysis program. Different percentage concentration of time essential oil dissolved in dimethyl sulfoxide were tested: 0.5, 1, 5, 10, 25, 40, 50, 75, and 90. An inhibitory effect was observed on Fusarium graminearum strain, starting at 50% oil concentration, and on Rhizoctonia solani 40%, as can be seen in these images. This graph shows the index of inhibition. For phytotoxic activity, here's the visible difference between plants treated with deionized water and those treated with 0.1% time essential oil for each seed species. Time essential oil in this concentration shows a significant inhibitory action on seed germination, mostly in Lepidium sativum, in Sorghum saccharatum, and less in Sinapis alba. The greatest phytotoxic action was manifested on the length of the plant root, the most affected being sorghum and the least is Sinapis. Measurement of the rhizosphere pH after 24 hours for placing the plants in the broom-cresol purple medium indicated a major discoloration in the oil sample, which means activation of the proton pump. The method of staining plant roots with NBT in the oil sample indicated an accentuated intensity of the color, especially at the top of the roots. This means a high level of reactive oxygen species in treated seedling. In conclusion, the volatile components of time essential oil slowed the growth of both fungal strains studied, demonstrating a high antifungal effect. According to the results obtained, time essential oil at 0.1% concentration has an inhibitory action on the plant seed germination, mainly affecting the appearance of the primary lateral roots and image their architecture. And thank you for your attention.
Thank you to Gheorghe Denisa. If there are questions. Okay, so I think we are going to take a break now and see you at 12:30 p.m. Thank you.
Hello, if everybody returned from this short break, I would like to continue today's session with the second part. The first presentation will be biostimulant activity of yeast protein hydrolysates on vina radiata shots. The presentation will be given by Otilia Gabriela Barbieru, and I would like to ask my colleague to start sharing the screen.
Hello everyone. My name is Otilia Barbieru, and today I will be talking about yeast protein hydrolysates and their effect on mung beans. Let's start with biostimulants. They represent a set of novel agricultural additives used as a large scale nowadays in both ecological agriculture as well as in the conventional one. Recently, these were regulated through an European Council document, which offers a set of instructions on their use. They also define the main categories as well as the conformity measures for these products. The main categories are represented by humic and fulvic acids, seaweed extracts, various extracts such as plant extract and protein hydrolysates, as well as plant growth promoting microorganisms. The categories that hold the largest portion of the market are humic and fulvic acids, as well as seaweed extracts. However, the ones of interest for us are represented by protein hydrolysates. Protein hydrolysates can be obtained either chemically from an animal source, such as bones, viscera, or other connective tissue, or enzymatically from various plant sources or agro-waste. The most frequently used type of protein hydrolysates is of an animal source. Several studies on plants treated with protein hydrolysates indicate that these have quite a number of beneficial effects, both on plants as well as the rhizosphere. They improve the uptake of nutrients and promote plant health. Also, some physiological modifications were observed, such as increased shoot and root length and root branching. One interesting substrate that could be used to obtain a protein hydrolysate is spent brewer's yeast. Brewing beer yields several types of waste, among which large amounts of yeast slurry. This slurry can be dried and used for different applications. Yeast contains large amounts of proteins, around 45% in weight. But proteins are not the only compounds of interest. Natural glutamates are also relevant, as they can be used for plant cultures, since they tend to chelate metallic ions and could serve as an easy method to enrich soils in certain micronutrients. Lastly, yeast extracts are rich in vitamins, especially the B group, which are essential in plant growth. The aim of our experiment was to see the effect of yeast hydrolysates on germination and the first stage of development of our plants, even when they are under stress. In order to obtain the protein hydrolysate out of yeast, several steps were taken beforehand. The yeast slurry was dried using a drum dryer, then it was leached using a combination of high-pressure homogenization and enzymatic treatment, which proved to be quite efficient. The supernatant containing the soluble proteins was spray-dried into a powder. The powder was subjected to a treatment with alkalase, serine protease, also known as subtilisine. The reaction mixture was incubated at 50 degrees Celsius for 4 hours under shaking. The yeast hydrolysates were used afterwards to treat the seeds. We used mung beans, which were surface sterilized using sodium hypochlorite and ethanol, and then washed several times with sterile water. The seeds were soaked in a solution of yeast protein hydrolysates for an hour before being transferred in petri dishes lined with sterile gauze. They are placed in a cool and dark place to germinate for 3 days and watered every day. Half of the seeds were treated with sodium chloride, 200 millimolar, in order to induce saline stress. The images show a comparison between the salt-treated plants and the untreated plants for all 3 conditions. As it can be observed, the salt-treated shoots are underdeveloped. In the case of our test, we could observe that treating the seeds with yeast protein hydrolysate seems to improve the germination rate, especially in the case of the salt-stressed seeds. In the case of biomass accumulation, we can observe that different parts of the plant react differently to different concentration of yeast hydrolysate. Root length presents similar growth in all 3 growing conditions, but yeast hydrolysate could not mitigate the effect of salt. Shoots of the salt-treated plant seem to have a higher resistance to stress if they are treated with undiluted yeast hydrolysate. Finally, in the case of weight, the undiluted yeast seems to have more of an effect in cancelling the saline stress. In conclusion, we prepared the yeast protein hydrolysate using an enzymatic method, which we tested on mung beans, which were subjected to saline stress. Some modifications were observed in plant development in the case of stressed plants. Next up, we plan to test these hydrolysates for their phytohormone-like activity and maybe even test them at a higher scale, such as a greenhouse.
Thank you for the nice presentation. If there are any questions for Ms. Barbiero from the audience. If not, we will move on to the next presentation, which will be held by Mr. Daniel Preda. The name of the presentation is the activity of the lignolytic enzymes from the spent pleurotus substrate obtained by enriching the solid state fermentation process. Let's listen to his presentation.
Hello, My name is Daniel Pereda, and I will present to you a few aspects about the influence of selenium on activity of linolytic enzymes from the spent pleurotus substrate obtained by enriching the solid-state fermentation process. Solid-state fermentation is a fermentation process that takes place in the absence or very low amount of free water, leading to significant quantities of products. This process has aroused interest due to its economic benefits and to its implication in the green chemistry concept employing industrial residues. The process is based on the mixture of Pleurotus ostratus mycelia with wood chips that was already boiled, sterilized and treated with calcium carbonate. Initially, we maintained the room temperature at 25 Celsius degrees for the blank probe and for the selenium enriched probes. All the experiments were done in triplicate. By the time we wait for mycelia to grow, the light was turned off. When we saw a small intention of mycelia to exit the bag, we turned on the light and decreased the temperature to 18 Celsius degrees. Some interesting facts were observed on the selenium enriched medium, selenium-2, such as a small delay on the fruiting body process, and also some irregularities and bigger fruiting bodies than selenium-1 and standard medium. The mushroom were preserved by lyophilization, and the CPS was used for protein extraction at pH 3.8 in a water bath with agitation at 25 Celsius degrees for 4 hours. In this slide can be seen the 4 types of enzymes responsible for degradation of linin, such as D-lacase, which is an oxidoreductase, linin peroxidase or versatile peroxidase, aryl alcohol oxidase, and manganese peroxidase. It can also be observed the schematic mechanism of each enzyme. Lacase activity takes place with the involvement of the 4 copper atom cluster that reduces copper 2 to copper 1. The oxygen is also reduced to water, regenerating the enzyme. Linin peroxidase oxidizes a linin compound such as veratryl alcohol to veratryl aldehyde in the presence of hydrogen peroxide. Aryl alcohol oxidase transforms veratryl alcohol by the action of the oxygen that's reduced to hydrogen peroxide to veratryl aldehyde, and manganese peroxidase oxidizes a phenolic substrate with the help of hydrogen peroxide and in the presence of manganese salt. In this slide, we observe the influence of selenium on lacase activity. The enzyme activity was measured at 420 nanometers, where the oxidized APTS absorbs. The blue curve is responsible for the lacase activity of the blank probe, the orange curve for the lacase activity of the selenium-1 probe, and the gray curve for the lacase activity of the selenium-2 probe. It also can be seen that selenium decreases the lacase activity. The results of the other linolytic enzymes are still underway. In conclusion, selenium salts can be used to transform CPS into a valuable subproduct enriched in selenium for nutraceuticals applications. And also the influence of selenium on the linolytic enzymes need an in-depth investigation. Thank you for your attention.
Thank you for your presentation. If there are any questions from the audience, we are listening to them. If not, we are moving on to the next presentation. The effect of a strigolactone mimic in phytopathogenic fungi development, which will be held by Ms. Ioana Alexandra Bala. We are listening forward to her presentation.
Hello, my name is Ioana, and today I will present a few considerations concerning the effect of a strigolactone mimic on phytopathogenic fungi development. Among living organisms, there are plants that can interact with some above and below ground species and release chemical compounds such as strigolactones. More than 25 SL and SLI compounds have been identified so far in the root exudate of several plant species. They all share a conserved tricyclic lactone structure containing rings referred to as ABC rings, linked via a nano-ether bridge to an invariable alpha-beta unsaturated furanol moiety named D-ring. Initially, upon their discovery, strigolectons were thought to have two main functions, acting as plant hormone and regulating several aspects regarding plant development and various stress-related functions, as well as signaling molecules in the rhizosphere. SLS can stimulate the branching and metabolism of presymbiotic hyphae in arbuscular mycorrhizal fungi. We have used a group of compounds lacking the ABC scaffold, able to stimulate germination, mimicking SL in activity, but not in structure. In this study, we tested the bioactivity of a new strigolecton mimic on Coletotrichum acutatum and Sclerotinia minor development. The objectives and the aim was to prepare the mimetic solutions and to test them on fungi in order to evaluate if they inhibit or stimulate the effects on structural development on fungi. The fungal strains that we used were represented by Coletotrichum acutatum and Sclerotinia minor. For cultivation methods, we used PTA medium, following the standard procedure, which include preparation, inoculation, and an incubation at 28 Celsius degrees. After 5 days, we observed the colony appearance. Following that, we inoculate other plates with the selected strains on different media which contain different solutions of SLS, GR24 and synthetic SL mimic 5 incorporated in agar medium. After 3 days, the developed fungal colonies were observed and the petri dishes were examined under a microscope. The observations were focused on the diameter and also on the arrangement and number of hyphae. The number of hyphal branches of different orders from second till the maximum observed in each case was recorded on each primary branch, moving back from the end of the youngest hyphal tip. Statistical analysis was applied on the data using SPSS. Both GR24 and SL synthetic mimic 5 compounds induced an increase of branching activities. GR24 treatment with various concentrations increased the hyphal branching and determined the formation of fourth-order branches, which were lacking in the controls as presented in Table 1. The responses of tested fungal strains to compound SL mimic 5 were quite similar to the response to GR24. The presence of these compounds in the culture media appears to inhibit the growth of both fungal plant pathogens, as shown in figures 1 and 2. In conclusion, our results suggest that the mimetic compound have the same effect as GR24, inducing hyphal branching stress response and inhibiting phytopathogen growth. Thank you for your attention.
Thank you for the presentation. If there are any questions from the audience, if not, we will move on to the next one. The presentation is called In Silico Evaluation of the Ability of Soy Proteins from Glycine Hispida to Release Antioxidant and Antihypertensive Peptides, and will be held by Mr. Grigore Mihai-Țăstan. Looking forward to this presentation.
My name is Grigore Mihai-Țăstan, and my presentation is called In Silico Evaluation of the Ability of Soy Proteins, Glycine Hispida, to Release Antioxidant and Antihypertensive Peptides. In literature, bioactive peptides are considered agents for promoting human health against non-communicable diseases through antihypertensive or antioxidant actions. We wanted to see the possible bioactive peptides with antihypertensive effects, also known as ACE inhibitors, and antioxidant effects released by two high-quality proteins in soy using an in silico approach. For this study, the FASTA sequences of alpha and beta subunits 1 of beta-glucine were collected from the UniProt KB database. a series of in silico approaches, biopeptide ranker, peptide calculator, toxin prediction, and allergen FP were employed to evaluate the potential of alpha and beta subunits 1 or beta-conglycine from soy as a potential source of bioactive peptides. The in silico digestion was done using 3 enzymes, subtilizine, papain, and pepsin, and the most bioactive peptides resulted in this phase were analyzed for their physicochemical properties. When we subjected the alpha and beta subunits 1 or beta-conglycine to computational digestion of 3 enzymes, we found variable numbers of resulting peptides due to the unique characteristic of each enzyme and the site they action on. In the case of antihypertensive activity, pepsin generated the highest number of bioactive peptides in both proteins, followed by papain and subtilizine. We note that of the 2 proteins in soy, the alpha subunit generated the largest number of antihypertensive peptides as follows. Pepsin released 47 peptides, papain 26, and subtilizine 11. The beta subunit showed 34 peptides released by pepsin, 23 by papain, and 9 by subtilizine. The antioxidant activity was low in both proteins, but the beta subunit of beta-glycine released a greater number of antioxidant peptides. For the beta subunit, subtilizine and pepsin released an equal number of antioxidant peptides, 3, the podium being occupied by papain with 5 peptides. In the case of the alpha subunit, subtilizine released 4 antioxidant peptides, followed by pepsin with 3 and papain with 2 peptides. When evaluating the sensory characteristics of the peptides obtained, we found that the alpha subunit, compared to the beta subunit, under the influence of pepsin, has the ability to release a greater number of beta peptides, which also shows ACE inhibitory effects. And in the case of papain, the alpha subunit released more beta peptides compared to the beta subunit. Subtilizine showed an equal number of peptides for both subunits. When we look at the physicochemical properties of the most biological active peptides, we can see that most of the peptides have an isolatory point at pH around 3, and they have low solubility in water. Also, most of these peptides can be found in both proteins, and they have beta characteristics, are non-toxic and non-allergen. The highest score, according to peptide ranker, showed the peptide PF 0.92, but it has low solubility in water, so it is not a good target for more in-depth studies. The next high-scoring sequence, 0.86, is the RF sequence, which showed an isolatory point at pH 10, and is the only peptide which is predicted to have good solubility in water. This results from the fact that the side group of arginine have a strong acidic character. The PHF peptide showed the highest score among antioxidant peptides, 0.77. According to allergen FP, this peptide is probably allergenic, so it is not a good target for further analysis. In conclusion, the alpha and beta subunits 1 or beta-glycine are a good substrate for the generation of antihypertensive and antioxidant peptides, and the in silico approach is a good method for accumulating information on these structures with beneficial effects on health. Thank you for your time.
Thank you for your presentation. We are looking forward to any questions from the audience. If not, we're gonna move on. The next presentation is called Ultrasound-Assisted Extraction of Phenolic Compounds from Quince Leaves using Box Mechanism Design, and will be held by Lumința Dumitriu, who is a PhD student at the University of Agronomical Sciences and Veterinary Medicine of Bucharest. Looking forward to your presentation.
Hello everyone, my name is Lumința Dumitriu from Team 4 Bioproducts. Today I want to talk to you about optimization of ultrasound-assisted extraction of phenolic compounds from quince leaves using a box Benken design. Quince fruits are characterized by astringency, bitterness, firmness. For this reason, they have a low demand for fresh consumption. However, quince fruits are widely used in the production of canned food, as jams, marmalade drinks. Byproducts of quince industrialization like quince seeds, pulp, peels, and leaves contain different amounts of bioactive compounds, such as phenolic acids, flavonoids, organic acids, polylactic zaccharides, and others, which can be used in various fields. Quince leaves are used as infusion in traditional medicine for their properties, like antipyretic, antitussive, and for the treatment of various skin dishes. Recently, the total phenolic content from leaves was reported as much higher than was found in pulp, peels, and seeds, which may indicate that this part of the tree can be much more interesting in the term of health-promoting constituents. As well, the leaves are a good and low-cost natural source of phenolic acids and flavonoids, which are antioxidants with different biological properties. Response surface methodology is an approach to evaluate the relationship between responses and independent variables, determining the optimal parameters and giving information about the predictive responses. Phenolic compounds from quince leaves was extracted by ultrasound-assisted method, and the optimization of extraction parameter was performed using response surface methodology from Design Expert software. The Box-Banken experimental design with 3-level and 3-factor was able to obtain the optimal condition for the highest yields of total phenolic compounds and the antioxidant activity of the extracts. The 3 factors selected for this experimental design were ethanol concentration, the extraction time, and the ratio of liquid to solid. The antioxidant activity of extracts was evaluated by 3 calorimetric methods, namely ABTC, DPPH, and FRAP. The optimal extract was analyzed by HPLC to identify the phenolic acids. The results of experimental design are summarized in Table 2. The experimental design consisted of 70 experiments, 5 of which at the center point were used to evaluate the experimental error. Figure 1 represents graphically the interaction between variables and the effects on the total phenolic content. The effect of the ethanol concentration on phenolic content is significant, and the value of total phenolic content increases with the decrease of ethanol concentration beyond 63%, then decreases to 96%. So, the ethanol concentration is inversely proportional to the phenolic content. At too low ethanol concentration, the polarity of the solvent is too high and the viscosity is increased, while at too high ethanol concentration, the polarity of the solvent decreases too much for efficient extraction of polyphenols. These slides show the interaction between variables and the effects on the antioxidant activity by ABTC, FRAP, and DPPH methods as a function of ethanol concentration, extraction time, and relation between liquid and solid. The ethanol concentration influenced the antioxidant activity by all methods, but in the case of DPPH, the ethanol concentration presented a lower level of significance compared to other two methods. The phenolic acids from the optimized extract of the quince leaves were identified by HPLC as caffeic acids and chlorogenic acids. To conclude, we can say that the highly significant factor for polyphenols content and the antioxidant activity was the ethanol concentration, while the other two were less significant for the extraction of the maximum total phenolic compounds and the maximum of antioxidant activity of the extracts. Also, the optimal extract was analyzed by HPLC to identify the phenolic acids, and the main phenolic acids were chlorogenic acids and caffeic acid. At the end, I would like to thank to my team 4, and this work was supported by Project POC and by Project Segment, subsidiary project number 2609, NutriGood.
Thank you for your presentation, Ms. Dumitriu. If there aren't any questions, we will move on. The next presentation will be held by Ms. Eliza Gabriela Mihailă, a PhD student at the Polytechnic University of Bucharest. And the name of her presentation is Green Solvents based on choline chloride for CO2 capture. We'll listen to her.
Hello, I'm Eliza, and today I will be talking about green solvents based on choline chloride for CO2 capture. CO2 is the most important greenhouse gas due to the large quantity in the atmosphere. The greenhouse effect is the way in which heat is trapped close to the surface of the Earth. These heat-trapping gases are compared to a blanket wrapped around the Earth, which keeps it toastier than it should be. There are both natural and human sources of CO2 emission. Natural sources include decomposition, ocean release, and respiration. Human sources come from activities like cement production, deforestation, as well as the burning of fossil fuels like coal, oil, and natural gas. Due to human activities, the atmospheric concentration of carbon dioxide has been rising extensively since the Industrial Revolution and has now reached dangerous levels not seen in the last 3 million years. The greenhouse effect accelerates the climate change with dramatic consequences. Extreme weather, perhaps you remember the tornado from Dobroja, or this year's flood from Moldavia, desertification, loss of biodiversity, and the emergence of new diseases. We've all seen what means a new virus for the humanity. World disasters are at a large scale, well, at a huge scale. I don't know if Bruce Willis will be able to save the day, but I'm sure the science will try its best. So, science brought us CCUS, carbon capture, utilization and storage. It prevents carbon dioxide from being released into the atmosphere. The technology involves capturing CO2 produced by power plants and then recycle the CO2 for utilization as raw material for fertilizers, bricks, chemicals or food industry, or compressing it for transportation and then injecting it deep into a rock formation at a carefully selected and safe site where it is permanently stored. Capture rates of around 90% of total CO2 emission produced by power plants, heavy industries and refineries are technically achievable using 3 main methods. First, pre-combustion capture separates CO2 from gasification and reforming processes in which a gaseous fuel or synthesis gas, syngas, is formed, consisting mainly of hydrogen, carbon monoxide and carbon dioxide. The carbon is captured from the syngas before it is combustion in the gas turbine. Second, oxy-combustion capture. Oxy-fuel combustion is the process of burning a fuel using pure oxygen or a mixture of oxygen and fuel gas instead of air. Since the nitrogen component of air is not heated, fuel consumption is reduced and higher flame temperatures are possible. Oxy-fuel combustion produces approximately 75% less flue gas than the air-fluid combustion and produces exhaust gases consisting primarily of CO2 and water. The last method is post-combustion CO2 capture. It refers to the removal of CO2 from power station flue gas prior to its compression, transportation, and utilization or storage discussed earlier. A number of different techniques are applicable. Almost all of them are adaptation of acid gas removal processes used in the chemical and petrochemical industries. CO2 can be separated using sorbents like solvents or zeolites, selective membranes, cryoseparation, or chemical looping. Many of these techniques existed before World War II, and consequently, post-combustion capture is the most developed. For my work, I have chosen the post-combustion absorption process in chemical solvents. For this application, I have selected the deep eutectic solvents because they present several advantages. They are easy to prepare, reusable, present low toxicity, and they are made from cheap raw materials. Eutectic solvents are substances formed by hydrogen bond donors between one hydrogen bond acceptor and one or more hydrogen bond donors. They have applicability in electrochemistry, metallurgy, extraction, chemical synthesis, lubricants, catalysis, and obviously in CO2 separation. The solvent processes must be environmental friendly, biocompatible, customizable just by tailoring their properties, and used in mild separation systems. The state-of-the-art CO2 capture method is the post-combustion, which separates CO2 from the combustion gases using amine solvents. This work aims to bring new green solvents, deep eutectic solvents with amines, solvents that are more stable, reusable, and safer than the classic aqueous amine solutions. Three types of deep eutectic solvents with amines were prepared, with choline chloride as hydrogen bond acceptor and each three amines as hydrogen bond donors, mono, di, and triethanolamine. 4 molar ratios were selected. The deep eutectic solvent preparation didn't require special conditions. After weighing the substances, they were mixed using a heating plate at moderate temperature, 50-60 Celsius degrees, with a vertical condenser system until clear liquids were obtained. Several analyses were used for characterization. We can observe the molar ratio didn't have significant influence in the pH measurements. Density, refraction index, and the electrical conductivity seems to decrease with the amine molar ratio increasing, while viscosity mostly increases with the increasing ratio of amines. Absorption tests were made in a simple installation which started with a pure CO2 tank at 500 ml per minute flow rate bubbled through the solvent while mixing with a magnetic stirrer. Distortion test had a similar installation but with air or nitrogen tank. All the experiments were made at 2 different temperature and normal pressure. The normal temperature absorption experiments showed that the deep eutectic solvents with diethanolamine had better CO2 capture capacity, followed by monoethanolamine deep eutectic solvents, and very low results for triethanolamine. The experiments continue with distortion also at normal temperature and pressure. As expected, the distortion wasn't complete, meaning that some parameters should be modified. 2 cases of deep eutectic solvents were selected: choline chloride with monoethanolamine at molar ratio 1:8 and choline chloride with diethanolamine at 1:10 molar ratio due to their highest absorption rate. The temperature for this experiment was 30 Celsius degrees. For monoethanolamine, it can be observed that 25% more CO2 was captured just by raising this parameter. Quite the same trend was in the DEA case. The CO2 saturation was reached in only 30 minutes for monoethanolamine and 4 times faster for diethanolamine solvent, 45 minutes versus 3 hours. Continuing from the absorption at normal temperature and pressure condition, distortion at 120 Celsius degrees was studied. This temperature was selected because it's the amines aqueous solution distortion temperature. Both solvents lost their CO2 in just 5-10 minutes, and after continuing the heating, the solvent was partially evaporated. Conclusions: deep eutectic solvents were prepared easily. An optimal composition was studied experimentally. These solvents can be a new greener solution for CO2 absorption. Their advantages consist of cost-effective solvent price, easy preparation, easy reuse, and regeneration. Based on the previous observation that amines and dipeutectic solvents can absorb CO2, we developed a combination between them and improved the CO2 absorption capacity. CO2 from various sources can and will be captured somehow. The following question is, what should we do with it? Nature inspired us one important solution: bring inorganic CO2 into organic forms, such as microalgae. They are a sustainable answer because microalgae grows faster than the trees and have many utilizations. Remember CCUS and also the closing the loop economy? This month, the International Energy Agency published Net Zero by 2050, a roadmap for the global energy sector, a study of how to move up to a net zero energy system while ensuring stable and affordable energy supplies. The report also examines key uncertainties such as roles of bioenergy, carbon capture, and behavior changes in reaching net zero. Oh, I almost forgot. Elon Musk launched in February this year a big contest. X-Prize carbon removal is aimed at tackling the biggest threat facing humanity, fighting climate change and rebalancing planet's carbon cycle. The prize is motivational. This $100 million competition is the largest stimulant prize in history, an extraordinary milestone. He guaranteed he will not pay in bitcoins or Dogecoins. Thank you for your attention.
Thank you for your presentation, Ms. Eliza. If there aren't any questions from the audience, we will move on to the next presentation, which will be held by my colleague Naomi Tritan. She is a PhD student at the Faculty of Biology at the University of Bucharest. Her presentation is titled Morphological Characterizations of Viral Cells Treated with Biogenic Silica Nanoparticles. And let's listen to her presentation.
Hello everyone, my name is Naomi Tritan and today I will talk about morphological characterization of viral cells treated with biogenic silica nanoparticles. The current global situation did not have much of an impact on the rice market as the demand of rice increased all across the globe. It's well known that rice husk is a valuable source of biocompounds as it contains mostly organic compounds like cellulose, lignin and hemicellulose, followed by inorganic compounds like silica. About 600 million tons of rice husk are generated each year worldwide, and biogenic resources rich in silica have gathered great interest in the biomedicine field because of their availability, low cost, and eco-friendliness. Silica nanoparticles represent one of the most efficient forms of controlled release of silicic acid. It has been shown that silicic acid can promote wound healing and connective tissue repair. Particles are known to behave differently when they come in contact with biological material. Thus, the aim of the present study was to detect any morphological changes induced by silica nanoparticles from rice husk in the viral cell line in order to show their suitability to be used in different applications such as medical devices. In order to study cell morphology, viral cells were incubated for 24 hours at 37 degrees Celsius in Dulbecos-modified Eagles medium supplemented with 10% fetal bovine serum. Subsequently, the cells were exposed to 10 micrograms per milliliter silica nanoparticles dose. Two controls were used. One of them represents the cytotoxicity negative control where the cells were not treated. In the case of the cytotoxicity positive control, 5% dimethyl sulfoxide solution was used. Cell morphology was evaluated after 24 hours and 48 hours using fluorescence microscopy. Thus, live datassay uses calcine, AM, and ethidium homodimer to differentiate viable cells colored in green from dead cells colored in red. Determination of cell viability by fluorescence microscopy allows the examination of cell morphology too. Cytoskeleton was observed by labeling the actin filaments with phalloidin conjugated with alexafluor 488, and the nuclei were marked with DAPI. There is no change in the number of viable cells and in the cell morphology after 24 hours incubation with silica nanoparticles in comparison with the cytotoxicity negative control. After 48 hours of silica nanoparticles treatment, there was an increase in the number of metabolically active cells at 10 micrograms per milliliter silica nanoparticles dose in comparison to the untreated cells. Moreover, no dead cells can be seen, suggesting the capacity of silica nanoparticles to sustain cell survival and proliferation. Fluorescence microscopy images reveal no significant changes in cell morphology after 24 hours of silica nanoparticles exposure. Thus, the cell showed an elongated polygonal shape, which is typical for the epithelial phenotype of the viral cell line. The cytoskeleton is highly organized in a fibrillar structure with abundant actin filaments represented by arrows, which improves the mechanical stability of the nucleus. Due to the increase in the number of metabolically active cells, after 48 hours of incubation, the presence of a greater number of nuclei colored in blue can be observed in comparison with the untreated cells. There can be seen bundles of actin filaments represented by arrows which determine the shape of the cell, and the constantly changing arrays of actin filaments have an impact in the cell movement and mediate specific activities within it. To conclude, no significant morphological changes were found in the tested cell line, and this is the first step in proving that these nanoparticles are potential candidates for various biomedical applications. And thank you for your attention.
Thank you for your presentation. If there are any questions from the audience? If not, we will move on to the next presentation, which is called Bioremediation of Hydrocarbons polluted waters using microalgae. And our next presenter is Ms. Daria Gabriela Popa. We are looking forward to hearing your presentation.
Hello, my name is Daria Popa, and today I will be presenting you studies regarding bioremediation of hydrocarbons polluted waters using microalgae. As we already know by now, oil and gas industries generate approximately 250 million barrels of produced water per day. More than 40% of these waste streams are introduced into the environment, significantly contaminating it with hydrocarbons. Due to biological processes using microorganisms, which have been proven to be useful to remediate polluted waters with petroleum hydrocarbons, produced waters have the potential to be a harmless product rather than being a waste. In particular, the bioremediation using algae, also called fecal remediation, is an eco-friendly and low-cost approach due to algae's ability to utilize certain pollutants as nutrient sources. Fecal remediation offers cost-effective, non-intrusive, and safe cleanup technology in which the potential macro or microalgae are used to treat a large group of pollutants. Therefore, in this study, we evaluated the microalgae's bioremediation capacity of heptane as a hydrocarbon pollutant for wastewaters. An important first step in bioremediation processes is to choose the proper microorganism which can decompose the water pollutant in question. Taking this into account, we selected 3 microalgae which are known for their potential to remediate hydrocarbons polluted waters, originating from the Norwegian culture collection of algae, Norca. Chlorella species Chlorella xorokiniana and Desmodesmus communis are green unicellular microalgae. During the test, cultures were grown under controlled conditions in a growth chamber with white LED light of 100 microeinstein intensity at a temperature of 25 Celsius degrees with an orbital agitation of 130 rotations per minute, that being necessary for an equal distribution of the pollutant in the growth media. The hydrocarbon pollutant chosen was heptane. The impact of two heptane concentrations of 0.1% and 0.01% was tested compared with unpolluted cultures as controls. In order to determine microalgae growth under pollutant stress conditions, we measured the optical densities for both controlled and heptane supplemented samples with an UV visible spectrophotometer at a 750 nanometers wavelength. For further confirmation of culture growth, we used a hemocytometer for microalgae cells counting. Regarding heptane residues in microalgae growth media, the GCMS method of analysis has been used. Studies were carried out during 14 days with 3 key moments of microalgae growth parameters measurements on the 3rd, the 7th, and 14th day of incubation. Experimental data showed that the growth of Chlorella sorokiniana and Chlorella species was the highest at the lower heptane concentration. Taking Chlorella sorokiniana by example, the optical density recorded at the end of the incubation time has been 2.5 times higher for the sample supplemented with 0.01% heptane than control without heptane. Even up to the concentration of 0.1% of heptane, growth measurements haven't presented any inhibitory effect on the 3 microalgae tested. Furthermore, Desmodesmus communis registered the best growth at the highest concentration of 0.1% heptane, with the optical density being with 70% higher than control ODs. When cells number were counted using the Marienfeld hemocytometer, it was observed the same ascendant trend of microalgae growth as previously shown by optical densities. We can say that Chlorella species seem to have an appetite for lower concentrations of heptane, while Desmodesmus communis cell numbers showed the highest values for the higher concentration of heptane. Degradation activity of the microalgae was determined by analyzing the residual hydrocarbon from the medium using a GCMS method with chloroform as a solvent from heptane extraction with a 2:1 volumic ratio. Here we can see a comparison between the blank of 0.1% heptane in the first day and 2 representative samples of both lower and higher heptane concentrations. While in the blank the presence of heptane could be recorded, the retention time being at 1.61 minutes, the samples after 14 days of microalgae growth showed an absence of heptane. The effect of heptane on Chlorella species, Chlorella sorokiniana and Desmodesmus communis growth rate was studied. All 3 strains showed increased optical densities and cells number values after 14 days of incubation, indicating high biomass productivity. GCMS chromatograms indicated that after during incubation period, heptane has been either consumed entirely by microalgae or at least to the point where it couldn't be seen by GCMS analysis. Thus we can appreciate that microalgae used heptane as a source of carbon for their growth. All that being said, it can be concluded that microalgae, Chlorella species, Chlorella sorokiniana and Desmodesmus communis could bioremediate produced waters contaminated with same types of hydrocarbons as heptane. Regarding future perspectives on this topic, I intend to cultivate new microalgae strains, measure additional growth parameters in order to determine growth rates, supplement microalgae growth media with other hydrocarbons, water pollutants, while analyzing their residues by other methods such TLC. Thank you for your attention.
Thank you for the presentation. If there are any questions from the audience, if not, we will move on to the next presentation, which will be held by Ms. Andreea Constantin, who is a student at the University of Agronomic Sciences and Veterinary Medicine of Bucharest. Her paper is called The Antioxidant Activity of Honey with Propolis Extract. We're looking forward to hearing her presentation.
Good afternoon. My name is Andreea Constantin. I'm from Room Honey Group, and today I will talk about the antioxidant activity of honey with propolis extract. Both honey and propolis have been recognized as functional. Propolis is a resinous mixture produced by bees by collecting parts of plants. Propolis' main bioactive components are polyphenols. Polyphenols are next-generation prebiotics. This study aimed to realize a new product based on honey enriched with polyphenols from a propolis extract and compared its antioxidant activity with a commercial mixture of honey with propolis. The polyphenols were extracted from propolis by ultrasound-assisted extraction with 75% ethanol solution and the ratio powder to solvent 1:5 for 30 minutes. The extract was split equally in 2 samples and concentrated using a semi-automated evaporation system at 40 Celsius degrees. One of 2 samples was resuspended in honey and the other was resuspended in 75% ethanol solution. The extract in honey was solubilized in a ultrasonic bath, mixed thoroughly, and the polyphenols were left to diffuse overnight. The antioxidant activity of the samples were assessed using 3 spectrophotometric methods, 2 based on radical sequencing activity, ABTC and DPPH, and 1 based on reducing antioxidant power, CUPRAC. The total phenolic content was determined with following chocolate. Our results show that the solubilization of the polyphenols extracted from propolis in honey can increase the antioxidant activity of honey more than that of the simple polyflora honey or commercial mixtures of honey with propolis. Our results show that the solubilization of the polyphenols extracted from propolis in honey can increase the antioxidant activity of honey more than that of the simple polyflora honey or commercial mixtures of honey with propolis. Thank you for your attention.
Okay, if there are any questions. If not, we'll move to the next presentation. Response surface methodology for maximizing the biostimulant effect of sodium alginate coating on mung bean seeds by Mr. Idong Lee.
Hello everyone, our group have been studying the biostimulant effect on mung bean seeds recently. Now, what is a biostimulant? A plant biostimulant is any substance or microorganism applied to plants with the aim of enhancing nutrition efficiency, abiotic stress tolerance, or crop quality traits, regardless of its nutrient content. The purpose of this work was to test different compounds or classes of compounds, for example, sodium alginate, which could be potentially biostimulant among bean seeds. Possible biostimulant effects can be estimated in a number of ways, such as the measurement of morphological plant growth characteristics, for example, stem length and radical length, or biochemical assays related to biostimulation, for example, plant growth hormones such as auxin, gibberellin, and alpha amylase. We are also going to try different delivery methods. Various conditions can be tested and could lead to a synergistic interaction between these conditions. One such situation could be the use of fluorescence extracted from Cistoceria barbata. All the tests and the experiments are designed to find the best compounds and the best delivery method. As I mentioned before, we are studying a brown seaweed, Cistoceria barbata. This is a kind of seaweed which is not much valorized. It's naturally available on the Romania coast at the Black Sea and was shown to include a relatively high amount of fluorescence as compared to other species. The fluorescence has antioxidant activity and biostimulant potential. Cistoceria barbata also contains alginate, so we are trying to extract its alginate and use it for coating. We use the mini glass fluidized bed system to coat the seeds. The solution is made up of sodium alginate and glycerol with certain concentration. The first experiment was to test the effect of commercial alginate and agar, which were directly put in the gel. We counted the number of viable seeds and measured the radical length of each seed. By using the response surface methodology, these two factors, agar and alginate concentration, were analyzed. For probability results, we found that the more agar is added, fewer seeds germinate, and the alginate's main effect is not significant, but the quadratic term shows an optimum concentration. We conjecture that the positive biostimulant effect of alginate could be diminished by another negative effect associated with a higher alginate concentration, which related to gel viscosity or water availability. For the radical length result, we found that the measure of radical lengths from day 3, 4, and 5 do not produce significantly different responses with the varying agar and alginate concentrations. This could happen because of the inevitable variability of the seed radical in the first few days since germination. However, starting with day 6, some effects start to develop. The equation shows that a higher agar concentration in the gel leads to a higher radical length, which is not correlated with the number of seeds germinated, as shown above for the first response, which was analyzed. This is possibly explained by the fact that the length is measured only for the seeds which managed to germinate. The interaction terms has a negative effect if alginate and agar concentrations have different signs in terms of code values. This means a positive effect if a higher alginate concentration is used with a low agar concentration or a low arginate concentration is used with a high agar concentration. This term hints at a possible ideal viscosity for this gel, which would depend greatly on both factors. In the second experiment, we used coated seeds to prevent the sodium arginate from dissolving in the aqueous gel medium. The seeds coating should be cross-linked with a divalent salt such as calcium chloride. So in this experiment, we tested two factors, which were the concentration of calcium chloride and the cross-link time. Same measures were taken in this experiment as above. As a result, the factors evaluated had no effect on the germination of the seeds. The coefficients of the two main terms and their interaction terms explain the relative importance of the effect with the limit of the two variety factors. The concentration of calcium chloride had a positive effect on the growth. This is not a good effect, which suggests that the seeds need to be washed after the cross-linking process to prevent any interference with the growth process. Seeds put in two different growth chambers. According to the results, the microclimate series economic lax chamber is better than the Argitron. In conclusion, we have determined a suitable concentration of agar solution. We noticed that the calcium chloride had a side effect, and it should be washed after the cross-link, and the microclimate series economic lax chamber will be the growth chamber which we will use in the future. We are doing fluorescence extraction and extract characterization. Also, we are doing arginate extraction and purification from the seaweed, trying to find the optimum extraction method. The next step is to put the fluorescents with arginate coat and test their biostimulant effect. Thank you for your attention.
Thank you for your presentation. If there aren't any questions, we will move on to the last presentation of today's session before the plenary session. The next presentation is called Optimization of 3D Bioinks Composition for Biodegradable Fish Baits from Industrial Byproducts. And the presentation will be given by Ms. Ioana Silvia Hosu. We're looking forward to your presentation.
Good afternoon, everybody. My name is Ioana Silvia Hosu, and today I will be talking about the optimization of 3D bioinks composition of biodegradable fish baits from industrial byproducts. My work was performed at NeXT-Chem. So the optimization of 3D bioinks composition for biodegradable fish baits from industrial byproducts, it is necessary as the industrial market requires this kind of biodegradable fish baits. The reason is that, for example, contamination of fresh waters and salt waters with the microplastic and classical plastic-based baits, or even that using organism-based baits such as ones for sport fishing goes against the sport itself. Also, for industrialization, the automatization and scaling up the production of biodegradable baits, it is a necessary goal. So, coming to our goals, in fact, we propose here the optimization of biopolymer-based composition suitable for 3D printing of fish baits and the usage of ingredients that can be extracted from byproducts of industrial processes to encourage bioeconomy. So, to achieve our goals, the optimization of the composition has to consider first the printability of biopolymers found in a secondary byproduct. The printability is mainly related to the rheological parameters, and these rheological parameters are related to several factors such as ingredient, concentration, printing temperature, or shear rate. All these factors are also related to viscosity or the ability to create a hydrogel. And the whole printability goes hand in hand with the precision of the printing parameters to have a 3D printed object. These printing parameters are nozzle diameter or printing speed or extrusion rate or nozzle height. Here, as we said, our goal is to optimize the composition to have printable biopolymers. To do so, we created a model based on a mixture simple lattice model in Design Expert software using 5 components. This model that we propose is a valuable one, as p-value is below 0.001. So we proposed 26 samples to statistically validate our model, and the components were pectin, chitosan, carboxymethylcellulose, carbonate, and acetic acid. Using these samples, we obtained the following results with this software, and the total desirability of our purpose concluded that chitosan and pectin have to be in high amounts to obtain 3D printing objects. If we take these factors one by one, the height of the object requires high chitosan and pectin amounts. And this is expected as pectin increases the viscosity of chitosan and helps the stability of the layer by layer composition of the 3D printed objects, making it more stable. If we go to plasticity as a factor that we desired, pectin in high amounts is also desirable. In detriment, in contrast, in fact, with the carboxymethylcellulose that has to be in lower amounts, because this carboxymethylcellulose is already known to reinforce the 3D bioinks. If we go to the fungus development, we can see that both pectin and chitosan have to be in lower amounts. So in the higher amounts, so the bioprintability doesn't involve fungus creation. So our conclusion of our work is that optimization was successful for all the factors involved in the modeling. The factors such as height of the object, elasticity, and antifungal properties. As some of the samples help us manually 3D printed objects that were stable and can self-sustain themselves, and other samples that in fact didn't reach our purpose, as the plasticity was too high or fungus was created in the sample. Other factors such as resistance of the object in the water or crystallinity of the calcium carbonate must be studied forward. I would like to thank the funding project POC contract number 81 from 2016, and especially the subsidiary project 1488 of this project. And I would also like to thank you for your attention and have a good evening.
Okay, thank you very much, Yora. If there are any questions from the audience, if not, we are ready to move to the plenary session. Steve Charco from Metal Toledo, USA, with the title Safety by Design Reaction Calorimetry in Chemical Development. Before I will give the floor to Steve, a few words about him. Steve Charco graduated first class honors in applied chemistry from Brunel University, London in 1983. He started working with Metal Toledo in 1994 as a product specialist for laboratory instrumentation, and in 1985 participated in the introduction of reaction calorimetry to the chemical industry. In 1999, Steve moved to the USA to support the AutoChem division of Metal Toledo, returning to Europe in 2001 to take up a role with the Chemi Informatics software group involved with laboratory automation and digitization. Today, Steve is a senior technology and application consultant with the reaction engineering group of Metal Toledo AutoChem, responsible for reactor automation with a special focus on reaction calorimetry and process safety for the chemical. So, Steve, if you are on, please.
Hello, yes, thank you very much. First of all, can I check that people can see my PowerPoint screen?
Yes,
yes. Okay, perfect. Then hello and welcome to this short presentation about safety by design and why calorimetry provides you with relevant information. So thank you for joining today. And after a short introduction, we'll discuss the value of calorimetry when designing a process that is robust and safe at scale. My name is Steve Chapko, and as you just heard, I'm a technology and applications consultant, primarily for calorimetry with METLA Toledo. So this will be our agenda, introduction, and then just some words about the drivers and the challenges that we face in the industry today. Some comments about how to scale up a reaction safely, and then to bring in the value of calorimetry, closing with just a couple of short examples from the laboratory. So, developing new compounds and introducing them into manufacturing, that requires understanding both of the chemical routes, the molecular routes, but also the physical process with all its parameters. And both the scale-up aspects and the safety-related parameters are needed to ensure that the process is safe at scale. So if we take a simplified approach to the whole procedure, we can start with the chemical synthesis, where the molecular behavior, the chemical route are really the key factors. And usually this is done at a pretty small scale, test tube scale. It's often enough to understand the chemical nature of the product and the reaction that it's needed to produce that product. And then the next stage is the scale-up stage. And here we have to first of all understand the critical process parameters, things like temperature and addition rate and stirring speed and so on for a typical semi-batch reaction. And usually at some stage there is a need to perform a more detailed process safety evaluation, where we learn what are the critical parameters in respect of safety and uncover any aspects that may not be scalable or that may present a problem to scaling up the process to a larger scale. And generally, the earlier that we can recognize those critical conditions, then the easier and the faster it is to adjust the process and properly implement the new process to make sure that it's safe at scale. Or to put that in other words, it's no good finding out that you have a problem the first time that you run the process at scale, because then you've committed your time, you've committed to equipment which may have to be changed. So it's really important to do these studies as early as possible on the small scale and anticipate the problems really before they arrive. In terms of what's driving this, the chemical and the pharmaceutical industry in general are always driven by quality, cost, and speed. And time to market pressure is always growing. There's always a requirement to have faster development to bring products to the market sooner and sooner. But of course, at the same time, we're all faced with the need to reduce cost, to keep the costs down as far as possible, and at the same time, of course, to try and improve and maintain the quality of the products as much as we can, all in the order of getting a competitive advantage in the marketplace. And the problem really that we face is that the processes and tools of the past don't really support the requirements of today and the future. So the technologies and the procedures really have to continuously be adapted and improved to meet the requirements that we have. So what are we actually trying to do? We're starting off with a process typically in a lab, in a test tube or a round bottom flask, and we need to start learning as much as we can about that process. And one of the basics is to understand the chemical and the physical behavior of the reaction. And that includes knowing when the reaction starts, when it stops, if there is maybe a delayed start or an induction period, or if there are some physical effects such as precipitation or crystallization along the way. And if we can, we might want to determine some mechanistic information, something about the kinetics of the process and so on. But then as we scale from the lab reactor to a larger scale reactor, we need to consider the issues related to the scalability and the safety. And that includes additional information such as understanding the rate of heat release, the optimum dosing profile of reagents, and also the heat removal capabilities of the larger scale reactor and specifics related to things like mixing and mass transfer. And that's the time when we also would look at the safety related issues as well. So if we look at what we can learn from the, let's call it the round bottom flask experiment, we can have a look at the temperature profile of the process. We could maybe measure things like pH. And we might qualitatively also see things like when a reaction starts and when it stops. So we can already start to get a feeling for those kind of things when we're working in the lab scale. In fact, looking at the temperature profiles, if we compare the temperature of the reactor and the surroundings to give a temperature difference curve, that's already an indication of the strength or the power of a reaction. So even at this stage, based on these observations on a small scale, we can start to identify some conclusions about the scalability of the process. And it may be that some of these things are already enough to eliminate the process as being non-scalable. But then what we need when we come to the scaling is some quantitative information. So we have to take into account things like reactor geometry, viscosity of the materials, the capability of how we mix the materials together, and how any heat that's evolved is produced and how that can be taken away from the reactor. So we need additional quantitative information to give us a clear picture of the scalability of the process and to identify any potential risks and allow us to resolve them. So we need to do more detailed work, more detailed than we can do just in a round bottom flask or a test tube, and that's to allow us to determine quantitatively some of these parameters, such as the ideal dosing rate, the rate of heat output, the specific heat capacity of the mixture, the reaction enthalpy, so in other words, the power of the reaction. So these are all the topics that are covered by calorimetry. So this is where calorimetry starts to become involved. It's really once we move from the very small scale lab reactor into the early stages of scale up, we start to apply calorimetry to determine quantitatively some of these parameters. So at the very beginning, we do the work just to look at the synthesis, and as I say, we can see the impact, the progression of the reaction when reactions start or stop. We can see physical events if there's a crystallization, or maybe we can detect different polymorphs being formed, or if there are phase separations. And we can do a rough and ready estimation of the reaction power by looking at the heat flow trend, heat flow curve. And with a little bit of mathematics, we can relate that heat flow curve to the power of the reaction in terms of watts per liter. And if you know approximately the cooling capability of your chemical plant, you may see already whether a process is too powerful to be scaled up to the plant. So even from the small scale, we can start to get useful information of this type. But that's only qualitative information. And as we go to the larger scale, then we need to perform reactions under more controlled conditions. And that's something that's usually performed in an instrument called a reaction calorimeter. And this will allow you to run the chemical process under, as far as possible, process-like conditions, which means that what happens in your reaction calorimeter bears a strong resemblance to what will eventually happen on the production plant. And that allows you to measure fundamental parameters such as the enthalpy of the reaction, the heat transfer properties of the material, heat capacity of the chemistry, and so on. So what we're doing there is putting putting quantifiable numbers to the qualitative information that we saw in the round bottom flask. And that then allows us to begin to understand how the heat release and how the progress of the reaction occurs over time, and whether it's related to things like dosing or change of temperature or stirring speed and so on. And then really, not quite a subset, but running in parallel, we start to measure the safety related parameters. So things like the maximum heat output rate of the process, if there is any accumulation of reagent, which effectively could store up problems for further in the process. And this allows to make the go or no-go decisions to decide whether a process can be carried forward safely. And that's really where the full process safety study comes in. This is often carried out by, let's say, a specialized team of scientists or engineers, very often part of the process development and scale-up function. But here you would typically fully investigate every aspect of the process, looking not just at the desired process, but also what could happen if things start to go wrong, what happens if the wrong reagents are mixed, or reagents are mixed in the wrong order, what happens if you lose control of the reaction, for example, if there is a loss of cooling or loss of control of the reactor process. And this is also quite a data intensive aspect. We're trying to measure and quantify everything and put numbers to it so that these reactions can be compared and assessed. And in case anybody's wondering, okay, why would you spend time to do that? Why is that so interesting? I'll just share one picture, and believe me, there are thousands that I could have used. But this can be the consequence of a loss of control of a chemical reaction. In this particular case, it's not that long ago. It was 2007 in the USA, and there was a batch reactor where the cooling failed, and the reaction ran away and then subsequently exploded. And you can see from the picture the catastrophic damage and, unfortunately, loss of life and injury in this case. And these events still happen all around the world. We regularly hear reports of chemical explosions And the vast majority of those are due to not having sufficient understanding, not having sufficient data about the process that was being run. So it's actually a very important aspect, a very important topic. Whenever you're looking at scaling up a chemical process to manufacturing scale, that you really have to have this data and handle it in the right way at every step of the way to make the right decisions to make sure the processes can be safe. So I'd like to share a couple of simple examples with you, just so that you can see what this data might look like and what we can conclude from that. So if we take the first example, very, very simple chemistry, actually a simple esterification, nominally run at just over room temperature, 40 degrees C. And the reaction consisted simply of adding propionic anhydride into an excess of 2-butanol. So what we expected was a fairly straightforward dosing controlled reaction. In other words, we expected that the reaction would start as we started mixing the reagents, and that everything would proceed in a fairly linear manner. What we actually saw was that during the addition of the reagents, the reaction didn't really begin at all. So the whole addition, which took about 15 minutes, that's the green curve, the MR curve, the mass curve. Over 15 minutes, the reaction didn't really happen. But then once the addition was complete, suddenly the reaction began and the temperature started to increase. And in fact, it increased beyond the cooling capability of the system in which it was being carried out. And that meant that the temperature in the reactor very quickly over the course of a few minutes increased from 40 degrees up to a maximum of 96 degrees. So a really huge and fast temperature increase that couldn't be controlled because we couldn't cool it anymore, and we'd already added all of the material into the reactor. And then once the reaction came to completion, the reaction got less powerful. The heat that was stored, the hot reactor cooled down, so the heat that was stored was released back into the ambient. And then we regained control. And this is what would typically happen if you ran this process just in a jacketed reactor, just in an ordinary lab reactor. So what are the results that we would see there? You get a feeling for the profile of the reaction, but you have no numbers to put to it. You don't know how much energy we're talking about, and you don't necessarily know what would be the effect of changing the temperature of that reaction or changing the rate of the addition of the chemical. So you're missing all of the numerical information at that stage. And that's why you would then want to run that process in a reaction calorimeter to put numbers to those trends. This is just basic information. And then the reaction calorimeter for the same process would allow you to calculate how much energy flows across the reactor wall. That's the orange curve in this slide. And that in turn, if you integrate the area under that curve, that actually shows the total amount of energy that's released over the period of the experiment. So we can see in this example, we released 123 kilojoules of energy. And this was from only about half a liter of material. So a very powerful reaction, considering the volume that was involved. But really the key to safety and scale up is to understand the rate at which that energy is released. And to do that, we measure the accumulated energy, which was called here ACU. And this is energy that is stored in the reaction by virtue of the fact that, first of all, it heats up. And then when it cools down, that energy is released back to the environment. So if we look at the orange curve there, what we see is that it's clearly in two parts. While the system is heating up, the accumulated energy is increasing. And then while the system is cooling down, that accumulated energy is released back to the cooling system. So whenever the temperature changes, heat's accumulated and released accordingly. And in order to fully understand the process, we have to know the heat release pattern. And so we have to actually combine those two terms. We have to combine the heat flow that we see coming across the wall and the energy which we know is stored in the system as a function of increasing temperature. And that gives us what we might call the true heat release pattern. This is really how the energy is released from the chemistry. And if we look at that curve, what we can see is that this reaction has quite a long induction period. So in other words, most of the anhydride had already been added before the reaction started. But we can also, more importantly, see that heat was being produced at a rate of almost 1,300 watts, 1.3 kilowatts at the peak of that reaction. So even though we already knew that the total amount of energy was 123 kilojoules, the reaction calorimeter tells us what the true heat release pattern was and tells us that in this particular case, at that peak moment, we were producing 1.3 kilowatts of heat from about 400 mils of solution. So we really need to know that, because when you scale that up to a manufacturing scale, you might find that your plant simply can't cope with that amount of energy, and it would then result in loss of control. And in the worst case, that could take us back to that terrible photograph I showed earlier of the consequences of a reactor explosion. Let's look at a different example. Again, fairly simple chemistry. This was a sodium borohydride reduction of methyl isonicotinate. And the reaction here, again, it was at moderate temperature, 30 degrees, and it was simply a case of adding the sodium borohydride to the solution. And again, we look at the heat release curve, the blue curve this time. And in our experiment, that maximum rate of heat release was a little over 60 watts, 63 watts. And if you would make the calculation of watts per liter, that would work out to be somewhere around about 120 watts per liter of energy being produced. And as a rule of thumb, a typical plant scale reactor can take away about 30 watts per liter. So we can see straight away from this one experiment that that spike at the beginning is producing energy at about 4 times the rate that a typical plant reactor could safely remove. And we could also see, given that the addition was made right at the beginning of that reaction, we can see that the reaction takes quite a while before it's complete. So that means that we have an accumulation of unreacted material. In other words, the mixture is already in the reactor, but it's not yet reacted. And that means potentially that we could call it a loaded bomb scenario. We have energy that's just waiting to come out. And if there was a cooling failure at exactly the time when all of that material was sitting in there unreacted, that would lead ultimately to an adiabatic temperature increase of the reactor, in this case of about 53 kelvins. And that would take the maximum temperature of the process to about 83 degrees C, which actually is above the boiling point of the solvent. So what that means is that if you lost cooling on that reactor, you would suddenly have your vessel producing heat, boiling the solvent, and most probably then releasing clouds of flammable solvent vapor into the atmosphere, which of course gives rise to the risk of explosion and fire. So this tells you that the process has a very large exotherm at the beginning, which exceeds the normal heat removal capacity. and that there is a large accumulation of unreacted material, and also the reaction takes quite a long time, which from a financial point of view could be quite costly, because it's tying up your plant equipment for a long time per batch. So those are the conclusions you would gain from that experiment. And your objectives then to develop the process further and to make a safe scale-up would be to look at reducing that maximum rate of heat output, reducing the accumulation of reagent, and reducing ultimately the batch time to make that process more viable. So those are the numeric values that you might get. This would be from a single experiment. So you can see that from performing a single experiment in a reaction calorimeter, there's a huge amount of quantitative information that's available. If we look at one more example, a nitration process, this was a fairly classical piece of organic chemistry, nitration of benzaldehyde using nitrating acid, mixed acid. This is carried out quite cold, just about 3 degrees C, because of the unstable nature of the product. And what we see is a very strong reaction with essentially no accumulation of reagent. If we look at the heat flow curve from the calorimeter, we can see that the reaction starts pretty much as soon as the addition is begun. And then as soon as the addition is complete, the reaction is finished. So there is almost no accumulation there. This is, in one sense, an intrinsically safe process, because you have control over it. As soon as you stop the addition of the reagent, you stop the reaction. So if something were to go wrong, then from a manufacturing point of view, you can control the consequences of that by stopping the addition of reagent. So again, calorimetry can show you the way that the heat is released, how much heat is released, and what you can do to stop that heat being released if you need to. Again, one experiment, a huge amount of quantitative information that can help you make those decisions. And then I think the final example, again, fairly well-known chemistry. We look at a Friedel-Crafts acylation. This one takes place in two parts, actually. The first part is the acylation step itself, which is carried out at about 5 degrees C to minimize byproduct formation. And then the second step is the hydrolysis, which you can perform at room temperature. And if we look at the heat flow curves from those two parts of the process, the first one actually looks very similar to what we saw in the nitration, whereby although it's a powerful reaction, we see it's very dosing controlled. So as soon as we stop adding reagent, we stop the reaction, so we have control over it. There's no induction time, there's no accumulation of reagent. The heat release rate is controlled by the dosing rate. However, we have to be aware that HCl is produced during the reaction. So there is gas being evolved, and the evolution of gas may be a threat because you have to be able to scrub or vent that gas. Otherwise, you risk pressurizing your reactor and bursting the reactor due to pressure. If we look at the hydrolysis part, fairly straightforward reaction. It's rather slower. And what we see is that there are some mixing and accumulation issues because we're forming a slurry. The nature of the chemistry is such that we form an aluminium hydroxide to form a gel, and that doesn't stir very well, and that can cause problems with removing the heat from the process because the mixing becomes an issue. So this is really useful information that would help you safely design the process for scale up, because you could consider the mixing and how to ensure that the mixture is properly agitated in order to make sure that you remove all the heat. So my conclusions are that when looking at the safe scale-up of processes, we have to consider not only the chemistry, but we also need to consider the heat transfer, the maximum rate of heat output, specific heat capacity of the materials, of course, the overall enthalpy of the reaction, the thermal conversion, in other words, how much of that reaction has happened at any one time. In other words, that gives us information about the accumulation of energy, and as a result, the so-called adiabatic temperature rise and the maximum temperature of the synthesis reaction, which can be derived from those parameters. That information we get from reaction calorimetry, and Metler Toledo, of course, is very much at the forefront of this technology. We do have a range of reaction calorimeters available, starting at the 100 mil scale, so fitting very much in the organic synthesis lab, and moving up to the 2 liter scale, which you would find in a process development and a process safety lab for full safety studies. So that concludes my presentation. I hope that was of some interest for you. And if anyone needs more information about reaction calorimetry, then please contact our colleagues organizing the seminar, and they'll be happy to help you. Thank you very much.
We thank you for the interesting presentation. I would like to leave Mr. Perkoçi to say a few words. I saw that you came online.
I have a question regarding other possibility to monitor the reaction. Florian here. I'm the scientific director of the NeXT-Chem, and I would like to know about other possibility to monitor the reaction besides calorimetry. If it's possible to put others device to monitor the reaction and to take information on your system.
Yes, absolutely. One of the most common devices that people would add would be a reaction analysis probe using mid-infrared technology. So this would allow us to take mid-infrared spectra in the reaction mixture in situ in real time. And that allows us to monitor the relative concentrations of the key species in the process. So typically we would see the concentrations of the starting materials, and they would diminish as the reaction proceeds. So we would see the concentration trends coming down. Ultimately, we would see the concentrations of the end product growing, so starting at 0 and growing as the product is formed. But critically, we would also see any transient intermediates that might appear during the process and then get consumed again. And this is critically important if we're trying to understand the exact mechanism of the process. So we know our starting materials, we know our end product, but we may not necessarily know how we get from one to the other. And in situ reaction analysis with mid-infrared is an extremely powerful tool because of the sensitivity and the specificity of the infrared signal. You can easily identify specific reactive groups and molecular structures. So you can build very comprehensive pictures of the relative concentrations of those materials during the course of the reaction. And with a minimum of calibration, you can make those quantitative values as well. So if we go through looking at the manufacturing scale, there are many people that would use that technology to make a final process check on whether they've made enough of the right material, whether the concentration is correct. So it's a very widely applicable, wide-ranging technology, which is often introduced into lab reactors and into process scale reactors to monitor the course of the process. You can also do that with Raman spectroscopy. We have corresponding tools for Raman spectroscopy, which is complementary to infrared, because very often if a molecule is not responsive in the infrared region, it is responsive to Raman. So you can build up a picture using Raman spectroscopy. Sometimes people also use UV as a tool for investigating the chemistry. And in other cases, particularly in cases looking at crystallization or particle systems, there are a range of tools available that allow to measure and to quantify and indeed to visualize particles as they're being formed. So if we considered the pharmaceutical crystallization sector, knowing the shape, the size, and the changes in the shape and size of the crystals as they're growing in situ, that's a hugely important step to understanding and controlling the final solid form of the process. So certainly our lab reactors are designed to accommodate any and all of those techniques simultaneously. So whilst performing the chemical process and measuring the heat flow, you could also be measuring the concentrations by infrared, by Raman, measuring particle formation and so on.
Okay, thank you very much for your answer and for the very interesting presentation.
Thank you.
Now, if are any other questions? If not, we are a little bit ahead of schedule. So I propose for everyone to take a break of approximately 20 minutes, and we'll meet back here at 2:40 p.m. For the closing ceremony and also for the awards. We have several awards for the best papers presented yesterday and today. So we'll see in a few minutes. So hello everyone once again. We are closing the final ceremony of this workshop, the awards that will be granted to some of the best works presented these days. Before we go to these awards, I would like to thank some of our colleagues involved in the organization of this workshop. Our young colleagues, Marinela Dumitru, George Teodorescu, Naomi Tritean, Alexandru Vlaicu, Ana Baroi și Adrian Vasile, who organized the three sections of the workshop, as well as my colleagues, Irina Chican, Alexandrina Ivan, and Ștefan Mocanu, who worked behind the scenes to ensure the building of this workshop. I would like to ask Professor Andrei Sârbu if he's online to say a few words in these final moments of the workshop. Please, Professor Sârbu.
Yes, thank you very much for this last opportunity to express my impression about the symposium running. I appreciate that it was a successful edition, an increase in comparison with the previous two editions. The most important was that we succeed to extend the area of coverage, including guests from other institutes. I mean, master students and PhD students. I appreciate that we are on a good road and that the quality of presentation was at a rather high level. I didn't saw any presentation under the standard, but many which are over the standard. And I think that we can be glad that we have these young people in institute and in general in the research. And from the behalf of Romania's Chemical Society, I want to congratulate the winners and also to congratulate all participants for their works. Thank you.
Thank you, Professor Sârbu. And now for the moment expected by all participants for the awards, I will ask our scientific manager and the father of this workshop,
Okay.
Professor Florin Oancea, to present the awards.
Thank you very much. Thank you very much for giving me the floor. Yeah. It was a very difficult job for us to select the winner, the young scientists which have the best presentation, and to give them prizes and special mention. As Professor Sârbu said, we highly appreciate the quality of all the presentation. Each of you are winners because was a clear enhance of the quality. Already we were opening for others participants outside of the NeXT-Chem, and was a clear interdisciplinarity and transdisciplinarity in the all presentation done these 2 days. However, we should select the best. And after long discussion, we decided to give prizes to the following. Andreea Afilipoae. It's a prize which is attached to a voucher for participation to the PRIOChem. And this is a special award for Andreea Afilipoae. Maria Eliza Pușcașu, which also is getting a special award prize together with a voucher for the participation to the NeXT-Chem. Yeah, registration fee support voucher are not only it's a concrete paper which value the fee of the registration for PRIOChem when 2021. Then another special award go to the Luminița Dimitriu. Also together with a voucher for the participation to the PRIOChem 2021. Besides these 3 special awards, which I repeat were very difficult to be chosen, because were also others young scientists like Elena Cojocaru or Rebecca Leu Alexa or Eliza Mihaila or Daria Popa. So almost all of you have very nice and very good presentation. But we give 3 awards, and we decided also to give 3 special mention to Julia Elena Neblea. Honorable mention. To Claudiu Eduard Lizescu. And another honorable mention to Ancu Țădin. Once again, we have a very difficult job to select from very good presentation. Congratulations to all the participants. Congratulations to the young scientists which got prizes, special award and special honorable mention. And I hope to see you in the autumn with a new and exciting scientific presentation. Thank you very much.
Thank you very much. And with these conclusions of Professor Oancea, unfortunately, our general manager cannot be with us today, so I have to close this workshop. I thank you once again to all the participants, to all our guests who made very interesting presentations. And I hope to see you all in October at ProChem 2021, and why not next year at the 4th edition of NeXT-Chem workshop. Thank you very much and goodbye.
Thank you. Bye-bye.